Okay, so maybe may I ask the audience to guess which one will be British and which one will be American? The one on my own right, far right, will be what? The American. Yeah, you're correct. You can see the British look more stern. Next slide. All right, so Movement Disorder Society is interested in advancing knowledge of clinicians, scientists, and allied medical practitioners. And very quickly, I will encourage you to be a member. Is interested in. I've always heard that nothing in life is free, even in Freetown. Allied medical. You can trust me. If something is free, I take advantage of it. Very quickly, I will. Membership fee for Africans for now. People, scientists working in Africa is still free. It won't be free throughout. So trust me, you can visit our site and membership register as a member. We also have a journal called Clinical Practice Movement Disorders, and that journal is also available for your publications. And it has a fairly good impact factor, not necessarily very, very high, but you can also publish there, and that will count for you. Next slide. All right, so what is research? Research is finding answers and you have questions being asked, okay? For example, if at the Babcock University guest house, they found out I didn't use the shampoo, it may not be because their shampoo is not good, it's because I don't have hair on my head to shampoo. So that is finding answers to questions. So neuroscience is quite vast and it accommodates everybody. It accommodates those who want to look at humans, those who want to look at animals, vertebrate animals, and even invertebrates. All right. So we're involved in research training. We look at grants and awards, and then research finding and presentations. So we look forward to, when you have done research, what you have done must be made known so that you can ask other questions or you can network. Okay, next slide. Now, research output in Africa, we find out that, yes, Africa is, on the global map for research. However, there's been a steady rise in publications affiliated to some of the continent's countries. And statistically, the highest output of publications come from South Africa. And I'm sure Professor Manga is part of those who contribute to things coming out of South Africa. Also, Egypt is second and Nigeria is third. This was as of 2016. I'm not sure it has changed much since then. Now, of the overall publications from sub-Saharan Africa, almost 70% also have non-African based authors, according to Bhaskaran. So in other words, yes, research output is coming from Africa, but many Africans are not telling their stories. And this is where I've come to challenge us that we need to improve our own output. Yes, there are quite a number of grants that you will not be able to access except you have other collaborators from the Western world. That's fine, but we have to begin to tell our own stories. Next slide. All right, so this is just a pictorial appreciation. As of that 2016, South Africa produced about 31,000 publications that were in the repository. Egypt had about 17,000 and Nigeria about 11,000. So we need to improve on this, and that's why Neuroscience Society of Nigeria is well positioned to stimulate us to do more quality research and get more output. Next slide. All right, so the Movement Disorder Society will support research. Again, neuroscience is so broad. There's neurology, there's neurosurgery, neurobehavior, there's neurobiology, there's so many areas, but we are more interested in those who are looking at movement disorders, all right, at the MDS. So some of the common disorders, Parkinson's disease, a hypokinetic movement disorder, then some hyperkinetic dystonias, then very serious disorders such as multiple system atrophy, progressive suprabulbar palsy, Tourette's syndrome, dementia, Huntington's chorea, myoclonus, and other forms of chorea. Now, you will be amazed. I don't have enough time, but there are videos. I can send you the link that some of the people who are doing great things in movement disorder today in research are people who are themselves were diagnosed with movement disorder, some with Tourette's syndrome, some with attention deficit hyperactivity disorder. They themselves, because of their condition, made up their minds to be researchers in movement disorder. Next slide. All right, so at MDS, we have over 11,000 members. That gives us a very large network. So if you are a part of this body, you will have a large network of people in neuroscience, especially related to movement disorder. So we have neurologists, we have pediatric neurologists, we have physical therapists, we have nurses, we have speech therapists, and we have basic scientists. And I always continue to make sure that basic scientists are not relegated to the background. Yes, in terms of membership, we're fewer, but clearly, if you make an effort, we definitely have room for enough basic scientists to join. So we've been having congresses since 1990, and we also have several regional meetings, and we organize training both in person and virtual. So some of the trainings we have meet the expert sessions. Then we have video and self-learning online materials. So if you join and become a member, you'll have access to some of these online materials. And even if you're a basic scientist, it helps you when you look at the clinical conditions to appreciate how much your research is going to help those who are suffering from these conditions. Then we have the visiting professor program, in which experts from other sections, all right? So we have African section, we have the American section, we have the Oceania section, we have the European section, and the Pacific Oceanic section and all that. So you can bring in professors to come and run programs in Africa under the visiting professor program. Also, we have the virtual professor, where the professor doesn't have to come in physically, but through virtual mentorship or training, they do that. And then we also have the ambassador program, where you can invite members of the MDS to come and speak about MDS. So the society applies to MDS to bring an ambassador to talk to them. So I am promoting MDS this morning, but it's not under the ambassador program. Next slide. Next slide, please. All right, so that is the education committee. We are drawn from different parts of Africa. In the center with the beard is Professor Jonathan Kahn, who is going to start this August. I think next Congress, so he'll be taking over as the leader of the African section, and next to him is the former leader of the education section, Professor Ali Salash, and that's myself, and I'm also taking over this August as the leader of the education committee of this African section. Next slide. All right, so the MDS outreach programs, this aims to provide financial support and expert faculty for local educational activities and meetings in the field of movement disorder. So outreach programs are limited to individuals residing in and the programs taking place in no-fee countries, all right? So Africa is one of the no-fee countries. So to apply for this, if you are a researcher and you've moved to the US, you will not qualify. But if you are coming back to give back to your continent, then you can apply for that. So MDS supports is available in terms of financial and in terms of providing faculty, experts, speakers. So we take care of the honorarium of the experts as well as the travel expenses. May I just announce that the travel expenses is economy flights, not business class. All right, so we also provide technical support and access to paid Zoom accounts. And then we endorse programs. We allow you to use our logo, and you can then, we pay you something for the endorsement. Next slide, please. Next slide. All right, so having said all this, we want people to apply because again, Professor Wiyele will soon be here. I think he's arrived with his team. Yes, people have been applying and some have not gotten through. And I just want to let you know that there is a need for us to be more thorough in our application process, because again, we're competing against applications from other parts of the continent. One of the important things about the application process is a strong letter from a mentor. All right. So as a younger researcher, if you're applying for any of these programs to be trained, get a strong recommendation from your mentor. All right. Maybe one or two we usually ask for. And then also you should write a strong intention statement. What do you intend to do after you've been trained? Okay. We have labs that we accredit, and I'm happy to say that one of the labs that are accredited is that of the NSN president, Professor Wiyele in Ilorin. And so I want to also call on the experts that many more labs can be, can apply to be accredited, and we'll be glad to follow. I'll be glad to follow that up. So interested applicants complete a single simplified application, including a program description and identification of the program's needs. Completed application will be reviewed and approved by the relevant MDS committees. And so you must allow for about 3 to 4 weeks after you've submitted your approval, and then we will communicate back to you. So to request for an application or for more information, you can go to, you can give a mail to our mail address, education@movementdisorders.org, and you get a response. Next slide. So we have visiting trainee grant program. You want to visit a lab of an expert. First of all, you communicate with the experts that you have identified abroad. And so we can support you for up to 6 months to have an educational training experience in movement disorder and establish clinical and research networks across the African continent. So you can write to Professor Salash in Egypt. You can write to Samia in Morocco. I think she's in Morocco, in Tunisia. And then there are other experts also across the African continent, or even outside the African continent. If you have contacts and they say, come to our lab, we just include all their acceptance of your application, and then we will treat it. Next slide. So we also have the clinical visiting training grant. Again, this may not apply to many of us, but if I think there are one or two clinicians that also are attending this conference. So they are those who are in physical therapy. So this focuses on those who want to increase their knowledge in movement disorder. Basically, this one is more for those who are in clinical practice. So it helps them to improve their diagnosis and management of movement disorder patients. And the goal is to bring clinical skills back to their home country. So that we expect a stepping down. Once we've trained that person, you go back to your home country and you step down the training. Next slide. All right, so research visiting training grants. All right, this focuses on applicants who wish to develop a research program on movement disorder topic, working on an existing research project, or to interact with other researchers with the goal to continue movement disorder research upon their return to the home country. Next slide. So eligibility for grants, they are open to resident doctors, they are open to fellows, and those trained in neurology training program, a recent graduate of the program who have returned to practice in their home sites within 5 years of completing the training. Status must be certified in writing by the applicant's employer or past supervisors. Applicants must reside in an African country and be a member of MDS. So non-members in the African region are also eligible for a free, no-fee membership. And then you must apply for membership in advance of application submission. So my plea is apply for MDS membership. So you write a personal statement outlining the aims of the training at a host center. Your curriculum vitae must be in English, and please abbreviate it. I went to Lagos, and from Lagos, I traveled by road to Calabar. Those are not necessary. At the most, your CV should be 2 to 3 pages max. The letter of recommendation from current supervisor or departmental chair, the letter of the host, the approval from the host center indicating acceptance into the training program. Next slide. So our international secretariat is in Milwaukee, Wisconsin. And so if also you travel and you want to talk to any of our administrators, that's the secretariat address. And then, of course, if you need any information, just send the email to info@movementdisorders.org. All right, so just to emphasize again that this is international, as the word international means. So we are spread right across the world. Africa is shown there. You can see the Oceania, Asia, and then you can see the European countries, and you can see the North and South American countries. Okay, so calls are made annually for courses to be organized by directors. So for the experts too, you can apply to us to organize a course. I have organized 2 courses myself. Usually we try to promote the courses because we do, our own research back at MDS. We find out how many people attended the courses, from what countries have they attended. We do an impact analysis, and so all that we do electronically. So we encourage faculty to apply to organize courses. When you apply, you must pin down the faculty that you want to use and we encourage that every faculty that must be used must be an MDS member. And we also insist that some of the faculty must be from outside the African continent. Our administrators can help you if you're having difficulty in locating faculties outside the continent. We have a repository and be of help. So guidelines are sent out from the Secretariat and they help in the process. So one of the things I want to encourage the senior ones, please, if we have like an email pool, as soon as the calls come out, I'll make sure I forward them to you so that you can apply. The one that you can see by the left is your right, is the one we organized in 2020, myself and another person I'm proud of, which is also well known to all the very seniors here, Olasumbo Afolayan. And it was a well-attended online course. So MDS is shifting a little bit away from in-person to more of the online courses. So the course materials that are used are the exclusive property of the Movement Disorder Society. So sometimes we do bank these courses and they belong to the MDS. So when you're putting materials for these courses, don't put things that are patented or things that have intellectual property, because we always put that in the forms that you have to fill. All right, so we also have what we call the Developing World Education Program, DWEP. This supports local movement disorder education meetings and courses in underserved areas. And MDS funds faculty participation. Applications are made to the International Secretariat. It's reviewed first by my own committee, and then selection is based on clarity and completeness of program proposals. Next slide. So we also have a leadership program. This has nothing to do with neuroscience per se, but to develop leadership qualities, all right? Because even as a scientist, you are supposed to be a leader. And usually it takes place, is a 12-month program encompassing the development of leadership skills through a mentored development and didactic skill format. And usually there's a training workshop that takes place immediately prior to the Congress, okay? So you are trained and then you go to a place also to learn, and there will be online platforms for the training. But it's something that is worth trying. It's very competitive. We probably just approve 2 people per year, but my take is always everything in life, if you don't try, then you don't get. Next slide. Okay, so the LEAP facilitates personal leadership growth and effectiveness of early career movement disorder specialists. It enhances leadership skills of early career specialists through education and group leadership principles, promote engagement of early career specialists in leadership in MDS, and integrates early career leaders into appropriate leadership positions within MDS. Next slide. Next slide. Okay, so this also may not apply too much to basic scientists, but we do every year organize a school for young neurologists. So I was in Tunisia last year, as a member of the education committee. So the next one will be taking place in Lagos in December, and then it will go to another country again. Thank you. All right, so I'm almost through. Next slide. Next slide, please. All right, so we have a family. This is the outgoing president of the MDS International and myself. And those are some of our administrative staff, Annette and some others. And these are the leaders of the African section. the former president before Cardoso, Twinsky, I think that's her name, and then Professor Njide Okudubadejo, Dr. Omolara Ojo, and myself. Next slide. So on that note, I have tried to summarize what we do at MDS that can build research capacity in neuroscience in Africa. Last slide. I think that, okay, so my appreciation goes to the MDS, to Professor Uyole, Babcock University. We also want to thank all the management and the people, and also appreciate the local organizing committee chairman. I think the last slide, I want to thank you all for listening.
Thank you very much, sir. We now have time for questions. If you have questions, please can we raise up your hand for Prof? Questions? Questions or comments or some clarifications? Yes, one. Any other? But can we have a mic, please, to give to him while he speaks? Or we don't have anyone. Hushers, please come and give him this one.
Thank you very much, Prof, for a very wonderful presentation. I am Ichipifuko Patrick Chukuyenim from Delta State University, Abraka. Mine is on how to actually reactivate my membership. I once registered after the Bro School Ibadan. I was once following the activities of the MDS, but somewhere along the line, got engrossed with my PhD studies and work, and I lost track. So the question is, how do I go about ratification of my membership? Thank you.
So I don't know whether you were given a membership number at that time. Yes, so what you do is just go back to the website, type in your name, and just say you want to renew your membership, okay? Under membership renewal, it will renew automatically. For now, because we are a no-fee country, there's no problem with renewing your membership. However, like I said, I don't know how long the no-fee will continue. But basically, all the low and medium income countries are no fee for now. So just go back to the website and you will renew your membership. Okay, that's it.
Thank you very much, Prof. I want to have a applause for Prof. In case you have any other clarification, you can always see him. Is there another question? So there's one question more, sir. Is it only for graduates and above, or can undergraduates apply?
Well, I don't want to bust your bubble. You can join as an undergraduate, but many of these things are usually more useful when you are in postgraduate. You know, undergraduate, you're still restricted by your school calendar, but it's easier when you are at the postgraduate level. Don't worry, just be patient. You'll get there. All right.
Thank you very much, sir. So we'll move on to the next lecture, to be given by Dr. Meshach Ijomone. I humbly request that you come to the podium and stand while I give your citation, sir. Please, can we give him a round of applause? Well, I could not pronounce the word. I don't want to massacre it. So Dr. Meshach Ijomone is an NIHK 43 emerging global leader and an associate professor at the University of Medical Sciences, Ondo, Nigeria, where he leads the Laboratory for Experimental and Translational Neurobiology. He obtained a BSc Human Anatomy in 2006 at the University of Port Harcourt and MSc and PhD in 2021 and 2015, respectively, at the Obafemi Awolowo University. He held a conjoint International Brain Research Organization, International Society of Neurochemistry Postdoctoral Fellow position at Nsn College of Medicine, New York, USA, between 2017 and 2018. His research activities have focused in neurotoxicology, neurodevelopment, and neurodegeneration. He aims to understand how the brain is affected by an interplay of genetic and environmental factors that trigger neuronal perturbations and or neuronal death at critical stages of development and across the lifespan. Dr. Ijomene has held numerous awards, fellowships, and grants. Notably, these include the Ibro ISN Research Fellowship in 2017, Ibro Return Home Fellowship 2018, ISN Can 1 billion grant 2019 and 2021, Young Ibro Award 2019, Alexander von Humboldt George Foster Research Fellowship for Experienced Researchers 2020, Ibro Early Career Award 2021, amongst others. Thank you very much. He was named an affiliate of the African Academy of Sciences in 2021, having been listed among 40 outstanding early career researchers in the continent. Furthermore, in 2022, he won the US National Institute of Health's K43 Emerging Global Leader Award, a 5-year grant to study gene-environment interactions in autism spectrum disorder. Following the award, he was appointed a visiting assistant professor at the Nsn College of Medicine, USA, where he collaborates on the K43 project. In 2023, he won the ISN Career Development Grant to leverage the WORM model for unraveling complex interplay of genes, environment, and the gut microbiome in Parkinson's disease. In this same year, he has also received the Professor James Kimani Lecture Award from the Society of Neuroscientists of Africa, and recently the Royal Society UK Colin Blickmore Bostry Award. With joy in my heart, I welcome Professor Dr. Ijomene to give his lecture. So please, if you have questions, just write it in a piece of paper and pass it to me so that we can manage time. Thank you very much.
Good morning. Doc is always keeping my full name. I think I'm going to give her a course on how to pronounce my full name. Always. Okay, so thank you. I'm really glad and privileged to be here. Thank you to the National Executive Committee, to the LOC chairman for inviting me, to my plenty mentors here and senior colleagues. I know they are proud of me because I'm going in their footsteps. So thank you, Professor Remusu, Professor Lokpade, Professor Wolele. I'm really privileged. So today, for time, I may have to skip some things in the slides, but I think what I really want to do is just poke you a little bit on why we do the C. elegans and maybe poke your interest on why you should try the C. elegans as a research model. Just poke enough that you probably dream of it later time in your career, particularly for the young ones, and find an interest in it. So unraveling the mysteries of the nervous system with the C. elegans. Next slide, please. Technical, are they? Hello? Okay, so, excuse me. We model human, how would I put it? There are many model organisms we use to study human biology, particularly in our case, neuroscience. And what are these model organisms? Basically, systems, lower systems or animals that we try to use in research to understand how our biology is. And there are several of them, from your African frog to the zebrafish to the jellyfish, the mice, rat, fruit fly, and so on and so forth. Well, we do more of the C. elegans. And most of these, over time, we've come to understand that the mouse is actually the most prolific model organisms in biomedical space. Although here in Nigeria, I think we use more of rats, but either way, mice and rats, they are both rodents in the same space. However, we're beginning to understand that we may need what I call simple, okay, they are non-malarial, but basically simpler systems. The understanding is that some of basic human biology are quite complex to understand. And if we can break down this complexity by looking at them in much more simpler systems, like you have the fruit fly, or C. elegans were able to tease apart or were able to tear apart very complex human problems or human processes and then understand them in simple format. I think someone said yesterday, you build from down up. And after understanding them at that simple basic level, we can now begin to build towards the complex human brain that we have as humans. Next slide. So today I'm just going to run through a brief thing. Like I said, for time I'll skip. So I try to talk about something called the tiny worm, how we use worm to model developmental neurotoxicity, what we currently are doing in our lab, which is what we are being paid to do for the next 5 years, trying to look at gene environmental interplays using the worm model. And something I think just give us funds for this morning that we are just trying our hands and see what will come out with, which is basically trying to leverage the worm for gut-brain axis studies. Next slide. So what we mean the tiny, well, next slide, please, just keep some of this on. So this guy is Sidney Brenner. He's a South African scientist. We call him the father of C. elegans. In 1963, he was the one that established or first used or described the C. elegans as an experimental platform. And I think he received the Nobel Prize for that in 2002. We tend to say so far there have been 5 Nobel Prizes with the C. elegans. I can't name all of them now, but there are 5 of them. I think the most recent one was Martin Shafi, the person who discovered the GFP. He actually used C. elegans to discover the GFP that we are trained all over in mammalian species currently. So we call it the tiny one because the worm is actually just less than 1 millimeter. In reality, you don't see it with you will just see something moving somewhere, but you don't see it with the naked eye. We tend to study them using a stereo microscope or a dissecting microscope. Next slide. Just keep going. So why, oh, your Zoom is blocking my titles, technical. Okay, but let's continue. Why do we use C. elegans? First thing, they're transparent. So basically, when we look at the C. elegans using the regular stereo microscope, you could see all its internal organs from the outside. The eggs, the gut, okay, maybe not the neurons in the way they appear, but they are basically there. So it's a transparent organism that we can see. So that's one of the reasons we're trying to look at it. So you could monitor what we call live in vivo studies by under the microscope by using the C. elegans, and it's one of the major advantages they have over most of the common model systems, small model systems that you have. They are easy to grow. He rushed my stuff on the image by the right at the bottom part are how we basically grow them. We grow them in regular plates. I think everybody in the lab now knows how to make the plates and knows how to prepare their food. They mainly eat bacteria. And they have a very short life cycle, just 2 days about, and a very short lifespan, which is 21 days. So if you want to do a little bit high-tech experiment, you have what we call a quick turnaround time. You don't have to wait like for your mice to, you understand what I mean, for 3 weeks and months, as the case may be. Your average C. elegans will die within 20 to 21 days. That's their lifespan. Next slide. And then we all have a fully sequenced genome. So we know all the genes. I think there are about 20,000 what we call protein, non-coding proteins plus RNA in the C. elegans. And it's actually the first organisms or first multicellular organism that its genome was fully sequenced in biology. And over 70% of these genes have human homologs. That means there is a, how would I put it? There is a comparable gene in the human. Sometimes for C. elegans, 2 things could happen. You could have one gene that in the higher species would create 2 other functions. So one gene will serve 2 functions that a human would have. Or in some cases, you could have several genes coalesce and form 1 function for the 1 gene in the human. So basically, but we have similarities in that context. Next slide. Okay, so, and it has a very simple nervous system. So the first diagram there, I actually tried to put a red stuff about the number of neurons compared to the other model organisms. It's about 302 neurons and 56 glia compared to Drosophila, which is closer, about over 100,000, I think, if my count is correct, it's around 130,000 currently for Drosophila, for zebrafish over 100, for Mars over 700, and 7 million as the case may be. And another interesting thing about the C. elegans, it was the first organisms that its connectome, or let me put it this way, its neural connectomes was fully completed. That means within the C. elegans, we know every neurons. There are 302, so we know them already. But we know every synapse, every dendritic spine junction, all the full connections and synaptic junctions you can find in a neuron have been completely mapped. I think that was done in 2019. Actually, it was done by some guys from Albert Einstein Medical College, where I'm a visitor. I'm actually very proud of them. I'm saying that work is probably going to win a Nobel sometime in the future when we understand the full, or something close, when we understand the full impact of what they did in 2019. Next slide. And what are some of the things we could study basically within the C. elegans? We could basically study their growth. You could take a few warm eggs someday, allow them grow for 2 days, treat them with something and study how they grow. So basically it's one of the modern organisms that are rapidly, how would I put it, that is having a rapid increase in studying aging. Because in 20 days they will die. So people try to use it to study how they age, or rather how they old. So you could study how they grow. You could study what we call movement. It's a very good model organism for movement disorders. That's why I'm joining movement disorders at the end of this meeting. Thank God he mentioned at the end of, I've already started before he finished his talk. So I'll join because you could easily monitor and look at their movement under the microscope. And they are very basic small stuff every or anybody can count or monitor. Their feeding level, their ability to reproduce. The other day in the lab, I'll give this joke. My students are all learning right now to do what we call synchronization. That's create, we call it, since they all hash different ways, we have to make them the same age by removing their eggs from the adults. And they're all learning. And I had a very funny experience recently. So Victor, one of them, he did one the previous day and he was very happy. He did the process correctly, actually. He did the synchronization process correctly. And in his mind, he has done it very well, very nice. Only for me to come the next day, and then I looked at under the microscope and I could count the worms. They're just like about 50 or so. And it was, I was like, okay, don't worry, calm down. Because when we do it properly, we actually expect you to generate over 100,000 worms from one of them. So there are that many, because one of them produced just about 350 eggs. So if you have like 100 worms were synchronized, you know what you should get. So they are easily to reproduce. Their sizes are very small, and there are a whole lot of studies around their gene expressions and all the rest, how their genes relate, or rather how their genes are expressed in different conditions. Next slide. The other things we're also trying to do with them is behavioral changes. I actually just listed one. There are so many things because sometimes they also have ability for cognition. I know it looks somehow because they don't have a brain, in quotes, they just have a very simple nervous system. I don't want to use the word they can think, but maybe they can think because they have a kind of cognitive function per se. So we call that, we could either use it what we call mechanosensation or chemosensation. So over time, if you put them in an environment that is very unfriendly to their chemical senses, maybe you put them in ethanol, and we always do that experiment sometimes, and you starve them. After a while, when you put them back in that ethanol situation, they've learned to associate ethanol with starvation. So they tend to run away from the ethanol. So in their simple basic terms, they think, or they have some form of cognition. And one of the very critical, how would I put it, advantage they also have is the ability to monitor neurodegeneration, particularly with the GFP tagged worms. So basically the worms, you can see them from outside under the microscope. So you could treat something and you could actually monitor how the neurons degenerate over time, and sometimes even how they heal back. So sometimes we do that by, we could treat worms with a very good toxicant one day. So 2 hours after, we'll look at it. 6 hours after, we'll look at it. 1 day after, we'll look at it. 24 hours after, or rather 48 hours. So you're systematically monitoring degeneration. They're actually good for what we call time-lapse experiment, trying to understand degeneration across several time stages. And you could study them developmentally across a whole, either whole organisms or very specific cells. Okay, next slides. He skipped something, I know. I think the next thing I wanted to talk about was So basically for our lab, mostly what we do is we try to use them to look at neurodevelopmental toxicology. Most of what we do in our lab currently, you can go on, I know you skipped something, so don't worry. We try to look at them in neurodevelopment, we try to look at how toxicants impact their development. So with these C. elegans, like I said, they grow in 2 days, or rather they hatch in 2 days. Basically what we do is we treat them at a very young age, Lava 1, and then try to see how that impacts their how it impacts as they grow up to adulthood. So that's what we mostly do, because we understand that most of toxicants that are around us, even when the humans, the adults, are exposed to them, you could be fine, but same levels of toxicants exposed at either prenatal stages or to children have one huge impact that then affects their lives as they grow on. So that basically brings us to what we are really interested in the lab. So most of what we do in the lab are basically using metal exposures, different form of metal from nickel, manganese, as the case may be. Next slide. I think I have a few results. So I think, okay, I'll skip this. Next slide. So these, there's a reason I put this here. This actually was the preliminary data that formed our NIH grant that we won last year. Everything here there was another figure, but that was very small. So this was the basics. And what did we basically do here? In both cases, you could see, in one case, we actually treated worms with manganese at the L1 stage. In another case, we treated them with nickel at the L1 stage. And then we're able to monitor their behavior 2 days later when they are young adults, or we're able to assess their ability for the neurons to degenerate, whether it's cholinergic, GABAergic, or dopaminergic neurons immediately after the treatment. And basically from this particular study, the whole summary was, we tend to see that both manganese and nickel, they could impact a kind of developmental toxicity on the C. elegans. And more importantly is that whatever toxicity they cause at that early stage in their life, that L1, reflects in the behavioral deficiencies that we see when they are adult. The adult is 2 days later. So you're not waiting for thy kingdom come. So next slide. I'll put a little about review, because this is one of the ones I'll skip. You pick it up there. A little bit of review we put together about several, I think these are mostly methods that we wrote this, I can't remember the year, I think 2017 or something like that, 2018. Having a summary of some of the works, both us, our lab, our group, our network, and other people have done using the C. elegans to model developmental neurotoxicology. Next slide, you can skip these. You can skip these. Thank you. So what we currently doing in our lab is we're trying to understand what we call gene-environmental interaction. So basically, we understand that most of the neurodevelopmental disorders, neurodegenerative diseases, apart from gene idiopathic. For example, Parkinson's is seen to be highly idiopathic, that a major part of what causes Parkinson's is things we don't know. So most likely environmental factors play a role. And what I got interests me in the those environmental factors, although our target for environmental factors is metals, but people use a whole lot of other things. How do these environmental factors, in this case, environmentally toxic metals, impact the genes or react or interact with the genes to trigger some of these perturbations we would see in the neurons? And that's what we try to do in the lab. And for the C. elegans, a little bit. And now I'm talking now, you're looking like it's a little bit very simple most of the time. So next slide, let's see what I have there. Okay. So the C. elegans can be played around. So many genes to move, so many genes to knock out. And the beautiful thing about the C. elegans is we have two major resources. One is in Japan, one is in US, whereby we have, how would I put it? We have C. elegans strains in their thousands or in their millions. So if for one day in my lab, I get one strain and I mate it with another strain and I generate a new strain, I'm expected as a C. elegans researcher to actually be nice enough to donate or send it to because someday I may not be there, something could happen, my lab gets, something happens, they would always have a repository of it to either share back to me or to any other researcher. So basically what we do is that we just take some of these worms that I already have some of these genes either knocked off or overexpressed, and then we expose these ones to some of our metals of interest and then see what we expect. Next slide. Okay, I think this was one work about, we're trying to do something around the EROK, MAK pathway. It's actually a pathway that is involved in inflammation. That is what we know it for mostly, but recently beginning to understand that it's actually a crucial pathway for how neurons develop. And for this particular study, all we did was, what did we do? We just got worms with EROK parts, some proteins from the main proteins, the last two there, PMK1 and MPK, which are equivalent to the human EROK and the human P38 gene. We knocked them off from the C. elegans and then treated them with nickel and vanadium, if I remember this work correctly, yes. Although I think I put only the nickel in the story. I'm partial to nickel. We know the people who are partial to vanadium. So I put only the nickel one here. So what we found out is that for the most times when we remove these two genes, PMK1 and MAPK1, the C. elegans tend to survive better. This is a study we're still doing. And sometimes, in most cases, sometimes it could impact their development in that they don't grow to their full size, while in some other cases, it could cause them, they grow better in their full size compared to the controls. I think from what I have here, in most cases, the PMK1 and the PMK3 were able to limit the damage such that they will allow them to grow to their full size compared to their control, whereby they were not growing to their full size. What it means is after 2 days, we expect them to be about 1 millimeter. So we're able to look and see if they're actually measuring about 1 millimeter or they are still short. Or they don't have, we expect to see eggs in them because they are young adults. We don't, so it's very easy to monitor. We didn't see eggs. Then he had not matured after what? After 2 days. Next slide. So this is what we are currently doing at the moment per se. This is just one slide because this is what the other Germany, we're doing in a collaboration as well with where my spouse is doing her Humboldt work. So I'll just do a summary. For this work, we took out two genes that are important for autism, neuroglin and neurexin. In C. elegans, they are NRS1 and NRG1 respectively. And these genes are actually genes that control cell adhesion molecules that help synapse to communicate, help synapse to break down, and help synapse to get deleted. So it improves or involves plasticity. And a whole level of gene profiling has recognized that these two genes are very important, or rather, how would I put it? They are part of one of the very important things that when mutated could trigger the pathogenesis of ASD. So basically, I'll just summarize a few things. So for these two genes, I'll leave the other one. You see the figure by the left, basically we did a behavior whereby the behavior is dopaminergic neuron dependent. So we call it a dopamine function behavior. So yes, thank you. So even though we've not done the whole work, we're still on it. From what we have seen here is that when we remove neurexin from the worms, we tend to see that the worms actually, what's the word to say now? When we remove neurexin, the worms worsen when it comes to doing the dopamine stuff, doing the dopamine behavior. So they don't do what we call body bends properly. But when we remove neuroglin, we don't know what's it. No, this is a work in progress. We're just giving you what we have currently. When we remove neuroglin, we see that it improves. We are still asking ourselves what could be the connection or how the two genes are probably interacting within the C. elegans. So that's part of the work we're currently doing. Next slide. Next slide. Okay. Now what we will be doing soon when I think this is the work I have for a PhD student. So more recently, we begin to understand that the brain, how would I put it now? The brain, many of the brain diseases are linked to the guts in an axis called the gut-brain axis. So we say the gut-brain axis is a bidirectional communication between the central nervous system and the enteric nervous system in the gut. And within the gut-brain axis, the most common thing is that all humans and almost all organisms have a whole lot of hosts of microbes within their gut. We call it the gut microbiome. And some understand that this gut microbiome actually influences how the brain functions. In fact, I think I will say this, researchers are beginning to have understood that for Parkinson's, people who develop Parkinson's actually way beyond when they develop Parkinson's, they have what we call stomach disorders from, okay, when I said this, I got scared because I tend to have regular diarrhea, but I'm not having Parkinson's. So, but over time, they tend to have stomach disorders at the very early stages, even before they find out that they have Parkinson's, maybe 20 or 30 years later in life. So the link is probably there's a link between what happens within the gut and how that influences the brain. So that is our interest. Continue. Next slide. Let's see what I have there. Okay, so this particular paper is another product of interest. So within this C. elegans community, when people in the C. elegans community started looking at the gut microbiomes, what they did was take human bacteria and then, because C. elegans eat bacteria, and then feed it to the worms. And we see how that interacts basically. So we call that something like humanized gut microbiome of the worm. But this paper, I don't know why I've forgotten the year, but it's very recent. I think it's 2021 or 2022. What this guy did here, they went into the wild, harvested a whole lot of C. elegans population. They are micro neuroscientists. So I don't understand the genes, the crazy technique they used to do it. And they went back into the lab and they isolated to an extent the natural bacteria within the worms. So they were able to identify 9 bacterial species that are natural to the worms. That is the bacteria that grow naturally in the gut microbiome. And like we do in the C. elegans, they harvested all of them and deposited them with the CGC. So what is our lab interested in doing? With a collaborator we have in the UK, what we are trying to do is get these bacterial strains, introduce them to the next slide, introduce them to the worms, and then see, in short, this is something we are about to do, so no result. Introduce them to the worms and then see how worms basically behave. We're trying to answer 2 basic questions. We're trying to understand how gut microbiome can modulate gene and environmental interactions, something we have termed multifactorial gene, environment, and gut microbiome interactions in the brain. And in this case, basically what our collaborator in the UK has been able to do, and some of the strains is going to share with us, is that he has caused those bacteria to express fluorescent proteins, either GFP or ROFP or yellow FP. The one we have here is yellow FP. So what we do is we feed these to the worms, and then we're able to study them under a fluorescent microscope, like we're expecting one to see how they react. But even beyond that, we're also hoping that even if we just feed them to the worms, even if they are not taxed, we can study behavior and study neurodegeneration as we would normally do with our methods, as the case may be. And we are trying to ask that question, is it possible that if we influence the gut microbiome population, that means if we don't know which ones are toxic and which ones are not, maybe there are about 9 currently, so it's a long-term study. So you first start by studying them one after the other, and then we'll now start doing mixes. If you mix bacteria A to bacteria B, what happens? So we're trying to say, if we influence the gut microbiome, would that in any way mitigate or worsen disease pathogenesis? You see, that's a very big question mark. One of the questions we are hoping to ask in the year. So without this next slide, I think I'm concluding. In all this, what I've been able to show is make you understand, the C. elegans is opening a diverse field in how we can use more simplest or simple organisms to understand the brain. And there are many avenues for that. And like somebody said, although it appeared a little bit illogical to use these simple systems, however, they have a very good translational value in that we can be able to do risk assessment of toxicologies, drug discovery, and basic biological mechanisms in the humans. And think, next slide, it's next slide. Okay, I think I'm done. Next slide is thank you.
Thank you very much, sir. You agree with me that has been a very, very interesting and intriguing lecture. stimulating also. I think so. I'm going to work with you. I'm interested in worms now. So we can pass the question to manage her time. So we have this question here. How is this C. elegans? It's not elegant. So it's elegant. And secondly, how is it cultured? Their habitats.
Okay. Okay, so for now, we are starting to be the repository for C. elegans in Nigeria, per se. I think we just sent our first stream to someone in Lagos who needed the stream. Our lab is not yet big yet. We are hoping that by next year, we'll have a very bigger, we're expecting a very bigger incubator. It's easy to cultivate, but it took us, I think the first time we started was with a grant Ibro gave me in 2019, if I can remember. So this is 2023, right? So it's one project I didn't want to do with my money, if you understand what I mean. You know, in this context of the world, we do research with our money. So what I've been doing is I've been building little by little till this NIH came last year. So we feel like in the next 3 to 4 years, we'll go a little bit stratospheric. So they are easy to culture. When you, maybe sometimes you could, we welcome people, maybe not this year again, because we're a little bit full this year. We come next year, you could learn. And if you think it's something you can set up where you are, because it's fairly straightforward, although you would need power. That's the main thing, because they survive between 20 to 25 degrees. So that's, our lab has one, so in short, it have to be, we can't afford to go a day without light before our C. elegans cry in our head. Given in rainy seasons, they could be fine, but you know, sometimes our weather really gets really crazily hot and it's not too good. So, but they are easy to culture. We culture them in regular plates. The plates you use, something similar to what they do for Drosophila, agar, but not the kind of agar you use for Drosophila, not the kind of chemical, but they are vertically easy to culture. Oh, I thought I saw someone at my back.
Another question here. Can C. elegans model be used to study mood disorders such as depression and anxiety? And secondly, are they social animals? Can they be used for social interaction studies?
The first one, I'm not too sure, because this they can make these people, the C. elegans researchers can make, I'm not too sure about anxiety and depression. I'm not too sure. But for social interactions, yes, definitely. C. elegans are actually very social animals. And I don't know whether to call them animals or whatever it is. They are very social animals. And so that's why we are leveraging them for autism research, because basically one of the hallmarks in autism is social interactive deficit. So they are quite social animals. It may not be like you see with the rats, but believe me, they gum, they embrace, they kiss, and we can study those.
Lastly, how are the metals or toxicity induced in this C. elegans? And secondly, is it through their feed or exposure through air as affected by human?
Okay. No more, right? Thank you. Okay. So far I know there are two ways, even though I'm biased to one way. There are two ways people put them in, not the feed. The feed is bacteria. You don't want to kill the bacteria. People put them in the plates, the agar that we put them in the lawn that will grow them in. So they make it together with that agar, that medium. But basically what I do and what we prefer to do in our lab is we treat them in, how would I put it? We put them in solutions. Our treatments are mostly acute, 1 hour, 1.5 hours, maximum 2 hours. So we put them in that solution for it. Understand that 1 hour, to you, 1 hour looks small, but 1 hour in the life of a C. elegans is not small. It's very mighty. So we put them in 1 hour and just leave them in the fluid. At least they can survive. Most of what we do are when they are just hashing. So they can actually survive about 8 hours without food yet when they are hashing. So that is when we target their treatment. But people also do it in adulthood. You just expose them, put them in fluid, in small epinephrine tubes, pack a whole lot of C. elegans, make sure you calculate your dose correctly, and you leave them there rotating for a few, depending on the timing you want to use. and that's how we treat them. But sometimes people put them in the medium that they used to grow them.
We have like 3 more. That's the last batch. Is there limitation on the use of C. elegans for research?
Definitely. In short, the first limitation and the limitations will always ask, people will always ask you, how do you translate to humans per se? And of course, the fact that there's goodness in simplicity. The fact that it's simple and you could tease apart and just study one thing, but you are losing a little bit of synergism, which you see in more complex, you get what I'm trying to say, more complex animals. So we have limitations, but for the, at least for the one models, the main thing is you are able to understand basic processes, and then you can work, you can build up. And particularly with using, trying to play around genes, you knock off these genes, you knock in that genes, what really happens? And I think I should add, they are very easy to transport. I have a colleague who was trying to ship his, one of the NIH people we had 2 or 3 years ago, trying to ship his mice or rats, those mutant rats, into the country. Oh boy, no, we agree. Oh, after so many, I don't, it's a program. 2 years into the program, he had not been able to state the work because it's not that he doesn't even have the money. NIH are giving him the money. But with all the, I don't know what they do in that Lagos and all the stuff. But with the C. elegans, I didn't say it outside. You just put it in your bag and it's in my lab. Thank you.
So this second question is similar to what you have just said. And so we don't have to take that. The last one, is there a way to quantify the population of the different types of neurons, like microglia, etc.? That's if they are different types like humans, and then can they be used to study visual neurobehaviors when exposed to colors? Can they use to study, how do they respond?
Let me start from the second one. As far as I'm concerned, they don't have anything remotely close to an eye or color visions, basically, you know, at least to my knowledge. You may find out otherwise later, but to my knowledge, they don't. So basically what we do is their sensation, either mechanosensation or chemosensation. That's what they do. The other one. Yeah. Oh, there are 302 neurons. Believe me, we know all the type of neurons. We know the number that are cholinergic, the number that are GABAergic, the number that are glutamergic. Which other one did I make?
Want to know if they are microglia like humans and
if they are
named like that, microglia, astrocytes.
Oh, for the glia cells, for now, I don't think, we just simply call them glia cells. They are 56. Unlike humans where you have all the other higher, where you have a whole lot of microglia compared to neurons. In short, glia biology in C. elegans is still a very basic new field. We are still trying to understand what those glia do because they are just 56 to 302 neurons. And for now, they are just called all glia. Mostly that's what we tend to call them. I'm not very sure if they've been specifically divided into astrocytes, microglia, as the case may be.
Okay, ma.
Thank you very much. There's this nutritional component that maybe another round of research. There's what they call gastroparesis. There's this lady, Loretta, from England, that was unable to digest food for about 8 years. If she consumed the food, it's not digested in the gut. That's in relation to the gut-brain component of your study. So probably there's a way we can also use this particular organism to see why she was unable to digest her food. So basically, it's liquid food through the vein, and she's still alive, but not through physical food, but liquid food. So I think nutritional study can also, so maybe we can speak on that.
I wouldn't know what to say about that per se, but I think, basically the C. elegans is really very, very simple system. So some of those digestive stuff that you have, in short, is not that complex. It's going in and go out. It's very easy. So I'm not too sure about how they probably would model that in the C. elegans. I wouldn't tell you that, because mostly what we try to do is model the microbes around the gut. At least that is what I know. But thank you. Something we can, maybe one day I'll just wake up and sleep and wake up again, and I would have
Another round of applause for Dr. Unido. Thank you very much, sir. So that's brings us to the end of the first plenary session. We'll move on to the second plenary session. Thank you very much for your attention.
Oh. Good morning, distinguished ladies and gentlemen. It appears that we have been sitting for a few more minutes than we should. May I request that you please stand wherever you are and we'll just take a stretch. Just stretch to the best of what you can. It appears that even those who were standing to talk want to stretch. Please, don't let me call you by name because I can call everyone seated here by name. So some people are actually sitting down thinking that I won't. Yes, please stretch. Aha. Okay, so we will attempt this one. You stand in a spot and turn as much as you can. If you can go 360, fine. Otherwise, 90, 180, 120. Excellent. The moment you feel okay, you can sit down. How come everybody is feeling okay at the same time? All right. Welcome, everyone. And I am sure that the few hours that we have been together in this auditorium has been a very reflective one and a very learning-centered experience. We will go immediately into the second plenary, but just before that, I will request senior colleagues that are here that we have quite a handful of our students who are still learning. So please make yourself more available so that they can come to you to ask you more questions. There are terminologies that we may be using that they are still growing in the process of learning what those specific terminologies are. And I think that will go a very long way. Secondly, the conference chair has also asked me to make it a public apology. Dr. Alalade is not here and will not be here. So please, if somewhere in your mind seated, you are expecting to see or hear from a Dr. Alalade, that will not be possible. Okay, that being said, I will invite to please come forward Dr. Fasheson. Please, ma, if you are here. There might be more than one Fasheson, but let me be more specific. Dr. Fasheson Oluwatoyin Aditu, if you are here, please make her feel welcome. Please come. I will be yielding the floor to you, but just before that, I will give an abridged version of who you are so that we know who is going to speak to us. Please come here. Aha. You know, when you have microphone, there is so much power in your hand. I can punish her now. Do you think I'm lying? Madam, smile. Okay, stop smiling. You see, I told you. So when you have microphone, you can do many things. But please don't retaliate when I give you. All right. Distinguished guests, ladies and gentlemen, standing before you and me is Dr. Fasheson Oluwatoyin Aditu. She is a lecturer and an honorary consultant in Babcock University Teaching Hospital. She is the pioneer consultant in charge of Babcock University Wellness Center, which is Babcock's University premier drug rehabilitation center. She belongs to a number of certified bodies and well-trained in the aspect of drug addiction, drug recovery, drug wellness, and so much. I may not be able to read one after the other, but I know for sure that she is internationally certified in addiction treatment of women in addiction. So if you are here and you are addicted as a woman, please feel free to see her. She will unaddict you. That one is clear. Has some depth and areas of interest: maternal and child health psychiatry, adolescent psychiatry, addiction psychiatry, and consultation liaison psychiatry. She is universally a trained personality all around in drug advisory programs. Ladies and gentlemen, she is a member of Babcock Research Group, a member of Mental Health and Drug Committee of the Nigerian Medical Association of Ogun State, and also a board member of Time to Heal, a non-governmental organization. Above all things, Dr. Fashason, just in case, remember I said for women, she's a trained one. She is happily married. So gentlemen who are here, take note, happily married. And she has 2 children and 2 beautiful teenage girls. She enjoys teaching and very anxious to learn and is passionate about what she does. Why don't you join me to put your hands together as I will make available the next 30 minutes for you, and then thereafter we'll take questions and answer. May I request that I will be seated at the back around the abstract place. If you have questions, please just wave, somebody will pick them up. The moment I climb up to respond to those questions, we will not entertain any other question. Thank you.
Thank you very much, Dr. Dangana. Good morning, everyone. It's a great privilege to be here today. We appreciate all our teachers here who has taught us, and we are still learning. We appreciate the organizers. We appreciate Babcock. My duty is to talk about the predisposition of substance and drug abuse to brain assaults. And next slide, please. Okay, this is just my outline. Please, let's move to the next slide. I have just 30 minutes. Now, you will all agree with me that recently there's great rise in the prevalence of drug abuse globally. And as a matter of fact, there are several classes of the NPS. NPS is the novel psychoactive substances. It's been introduced into the market, and gradually we have lost count of the number of NPS that we have. People get addicted to several things, even things that you cannot even imagine, and more are still developing. Drug addiction is directly or indirectly responsible for 11.8 million deaths each year in the world. Let me also tell you that according to Global Burden of Diseases study, this number of deaths is actually higher than what you have due to cancer and due to other causes of death all over the world. Please, next slide, please. Okay, I think I need to just move with my own. Please, are you with me? People at the photo. Next slide, please. Okay. Now, due to the increase in substance use, we've also discovered that there's also an increase in the number of infectious diseases like HIV, hepatitis, and TB. Also diseases that are rising due to IUDs, that is using drugs, unsafe sex, lack of concern about health. People do no longer comply with medication. There's also increase in the risk of brain assault, like we see in our practices, medical and mental problems, and also risk of accidents, injuries and disabilities, mood states, perception problem, and all this due to the substances that people are using. There is also loss of productivity. There's increase in healthcare costs. Government is saddled with this responsibility of taking care of people. Families are running into debt due to substance. There's increase in violence, crime, financial instability, emotional trauma. But notably, I want to tell you that mental illness and drug use, there are mysteries. So there's a way you talk about drug use, drug addiction, that you not talk about mental illness. What then is drug? Drug is any substance, with the exception of food and water, that when taken into the body, it alters the body's function. It alters it either physically or psychologically. This drug can either be legal, illegal, illicit. It can be prescription or non-prescription drug. As you will get to know along the line that some people actually are addicted to prescription drugs. And that is what we call safe drugs prescription. What's a psychoactive substance? Any substance that is used to alter the central nervous system in a bid to alter perception, mood, consciousness, cognition, and also behavior. Now, medication that have the potential to prevent or cure diseases. Let me also tell you that these safe drugs, like I said, are also drugs of addiction, which people are using nowadays. People use drugs to alter their perception, to alter their mood, to alter their performance. Next slide. Okay, so what is abuse? Abuse is excessive and maladaptive or addictive use of drugs for non-medical purposes, despite the social, psychological, or physical problems that may arise from such use. It is an uncertain chemicals for the purpose of creating pleasurable effects. Abuse is also when you use drug beyond the prescription, beyond the limits, beyond the duration that is being recommended to you. When you habitually use a drug that is not needed for therapeutic purposes, then I will say that is a psychoactive drug abuse. All right? I wish I'm in control of the slide. So epidemiology. Now, between 2000 and 2019, the number of people using drug between these 9 years increased to 22%. But about a year ago, it was discovered that 275 million people in this last 1 year have used drug. Over 36 million suffer from this drug abuse. Globally, 5.5 people between age 15 to 64 had used at least one type of drug in the 1 year. But in Nigeria, the figure is quite more than that. In Nigeria, in the past 1 year, 14.4 people had used one drug. So in Nigeria, our rate of drug use is about 3 times the world population. 36.3 million people suffer from drug use, and 11 million people in Nigeria use cannabis. That use cannabis needed treatment. Now, in these 24 years, cannabis potency had increased. I jokingly say that the cannabis fella use, because I know that some people had me. They say fella use cannabis and it function. And I told them, I said, the one fella use has gone into extinction. It is no longer available. The cannabis that we have in the world now is 4 times more potent than the ones that we had in the past. Percentage of adolescents using drugs, especially cannabis, is 40% more than it used to be. And it's because they believe that there is no harm. And in fact, if you have a friend that is using cannabis, they will tell you that there is no harm in doing drugs, right? Especially cannabis. And it is due to the lower perception of the drug harm. People believe that there's no harm in using drugs. In fact, some countries actually legalize cannabis. And there was a move in Nigeria that we should legalize cannabis. I don't know if we are ready for that. So based on the demographic changes, I should tell you that by 2030, there will be 11% rise in the number of people that are doing drugs, and 40% of them will be youths. the Gen Z community. Now, by 2030, 20 million Nigerians will be struggling with addiction. All right. So now this year, to commemorate the International World Fight Against Drug and Addiction, we were asked to share facts on drug use. And according to the UNODC Executive Director, he said there is need to close the gap between perception and reality. So we are to educate people and to safeguard the public health. In 2017, UNODC conducted a study in Nigeria, and it was published in 2018. It was bad that basically Gombe State and Oyo State have the highest number of people that use drugs. You will see that it carried about 23%. Why are we laughing? Did I commit any blunder? Next slide. All right, so we also discovered that 14.4 million Nigerians now use drugs, like I said, 10.6 million use cannabis, and 92,000 people use cocaine. Of the 10.6 million, of the 14.4% that I said, 1 in 4 people that use drug is a female, but that was in 2018. This is 2023. I'm sure if we do another study, females will be competing with the guys. If not, that females are doing more. Next slide, please. Oh, Jesus. So 1 in 7 had used drug in the past. Please, let's keep it rolling. I have to be very fast. Okay, 1 in 5 are IUD users. Like I said, 1 in 4 is a woman. All right, 1 in 5 people who use drug is dependent. 1 in 3 people who use cannabis is dependent. 1 in 5 people who use opioid is dependent, and 1 in 7 who use amphetamine is dependent. Now, let me say something about the 1 in 5 people that are dependent on cannabis. Unfortunately, people that do cannabis will tell you that they don't become dependent. It is because cannabis takes a long time in the system. Cannabis is deposited in the fat cells, so you don't really need to use it all the time to get the high that you need. Now, people don't get access to treatment. 40% of people who need treatment couldn't get access because of several reasons. Number 1 is they are afraid of stigma. Number 2, there's no facility. Number 3, there's no information about the treatment. Gradually, we'll get to the meat of the discussion. Now, these are just the classification of psychoactive stuff. We have the stimulants, we have the depressant, the opioid, the hallucinogens. These are based on the part of the brain, the activity they exert on the brain. For the stimulant, they are the ones that, makes us excited. The dopamine level is very high. Then we have another classification that we can say is unclassified. We can't really say this is where it belongs, because these substances act in several ways. They can act as depressant, they can act as stimulant. For example, the ketamine can act as it can act as an opioid, like a stimulant, and also has as an hallucinogen. Cannabis can act as a stimulant, can also act as an hallucinogen. Oh, Jesus. Next slide. Okay, now we have the NPS. These are new drugs that are causing problem for us psychiatrists, for doctors, causing problem for the world. They are drugs that are synthetic, like we have the synthetic catetones, the amphetamines, the tryptamines, and synthetic cannabinoids. Synthetic cannabinoids are, for example, what we call the Colorado, the normal cannabis and the cultivated cannabis. And these are the drugs that are causing problem because we don't even know how they act. They give us the impression that they mimic them, but they exert some effects in different parts of the brain.
Next slide, please. Now, these are the list of the NPS that we have. We have the renal, we have the mushroom, we have the Erujeje, we have the D5, the GOSI, we have several of them. And another new one is the NO2, the laughing gas that people are becoming addicted to. We have Tombo, we have Arizona, we have La Colorado, we have Tutolin, we have Kush, we have Cats. Next slide, please. All right. Now, how the risk factors for drug use, they are quite enormous. The genetic reasons, mental disorders, routes of administration, environmental factors, and early use. These are basically the reason why people do drugs. Next slide, please. Okay, now we will get to see why this happened. How do people progress? Not everybody that use drug eventually become addicted to drugs, but people just start by experimenting, recreational use. The next thing is they increase to occasional circumstantial use. They find themselves in a particular setting and they use, then intensified use, and then it gradually tilts towards the addictive use, which is the adult substance use, unhealthy and problematic one. Nobody actually plans to become addicted. Like I said, several reasons why people become addicted. So let's talk about the effect of substance on the brain. Brain injury can naturally arise from various causes. It can be due to traumatic causes or non-traumatic causes. Substance can lead to brain injury in several ways. Someone after using it can have a fall, get involved in fights, hit his head on the wall, lose consciousness. But we are talking about the direct effects of substance in the brain. When there's a traumatic effect, so many things happen. We can have a primary deficit or secondary injury. The secondary injury can lead to so many things. Brain death due to the imbalances or influxes of the intracellular calcium that can lead to edema, later free radicals, oxidative stress, triggering, and eventually the cell can die due to apoptosis or any other thing. Then the blood-brain barrier can also be compromised. And as a result of that, the brain can atrophy. There can be bleeding into the brain, leading to hematoma, raised intracranial pressure, coning, and eventually the brain dies off finally, or the person dies due to that. But we are talking about the direct impact of the substance on the brain. Next slide, please. And that will be talking about the non-traumatic brain injury. As a result of this, people, when you use substance, it can lead to so many things. Ischemia. Ischemia basically talk about the loss of blood supply to the brain, or poor blood supply to the brain, poor oxygenation, which can cause hypoxia. There can also be aneurysm, or there can be hemorrhagic stroke, which can also lead to destruction of the brain cells. Okay, so a condition that can directly attack the brain tissue, like stroke, heart attack, severe brain infection, drug overdoses. And so today we'll be talking about addiction, addictive effect, logic effect, and the ischemia in our discussion today. So basically, we're talking about clinicals, right? Okay. So if you look at the this slide, you will see where it's just a cascade of what happens to the brain. Where there's hypoxia, that one can lead to the death of some parts of the brain, cerebral ischemia. Due to the hypoxia, there can be calcium influx, which can lead to potassium or sodium influx. There can be release of the glutamate, and eventually, going down, excitotoxicity, there can be vasoconstriction, neuroinflammation, all these things, eventually cell death, edema, and then leading to secondary brain injury. Next slide. Next slide. This is basically what we discussed the other time. Next slide. Now, when someone takes substance, because the substances are usually lipophilic, so they can cross the BBB, the blood-brain barrier. So when they use the substance can involve controlled social use into compulsive relapse. Okay, and that is the people that I see every day. They come in addicted to the drug. So the transition to addiction results from very many things, from genetic. Let me tell you, some people naturally, they will become addicted. And I know that, I don't know if anybody here will fight me for talking about addiction, because I always get this backlash and all that, sometimes irritability, that I'm pathologizing addiction. Maybe I will get it today, or be friendly environment that you people will welcome me and give me a pat on the back. All right, so now some people will naturally do drugs. We'll talk about personality, we'll talk about vulnerabilities, we'll talk about genetic, which has shown that if a father takes drug, the child has a 4 times, especially the boy, risk of taking drugs, of drug in the environment, because it's allowed. There's something we call social learning. The child will generally use drug. No be so? Sorry, not so. Yes. So, okay. So there's a lot of this combination of factors that make people do drugs. But when they do drugs, there's something we call neuroadaptation, which we are all aware of. There's something we call neuroplasticity, where the brain just naturally adjusts to this new environment and start producing what is being challenged to produce. So now there are 3 stages in addiction. We have the binge stage, which basically we talk about the basal ganglia. Under basal ganglia, we talk about the dorsal striatum, which is the abuse circuitry. And we also talk about the nucleus accumbens, which is the reward circuitry. You are with me? All right, thank you. So we also talk about the withdrawal and negative effects, and we talk about the preoccupation and activation. Now, these 3 stages is what we talk about when people with addiction. Why? For the binge and intoxication, is being introduced to a new thing. The brain normally have dopamine, like the last speaker said something about the dopamine. But when you give the brain something else that is more than the brain has, naturally, what does the brain do? It adjusts to the new environment. Now, there's something that happens to the synapse when someone, naturally, we all know how there's communication between one neuron and the other, coming from one axon to the synapse, and then to the other axon, to the dendrite, and then to the other axon. Then we have the synaptic cleft. For drug, what usually happens in the cleft is the dopamine is released. When the dopamine, among other neurotransmitters, when the dopamine is released, it is actually expected to go back to the pre-synapse. But because of drug, when it is released, some certain drug prevents the dopamine from going back to the pre-synapse. So it stays a longer time in the synaptic cleft. Sometimes this neurotransmitter is also hijacked or be replaced by an agonist, which sometimes happens. So in this situation, now the basal ganglia, like I said, is the one that talks about they establish the new substance, the habit forming, the reward system. The extended amygdala talks about the uncomfortable feeling when the person gets when he does the drug. So negative and positive reinforcement. Now, for the reward forming in the basal ganglia, that is where we get the positive reinforcement. For the uncomfortable feeling that we have in the amygdala, that is the negative reinforcement. The body wants to use this substance in order to medicate the uncomfortable feeling that the body is getting, and now moves to the prefrontal cortex. That prefrontal cortex, which we know it is not well developed among the adolescents. So you worry why adolescents use drugs. What adolescents and young people is known for is just 2 things: experimentation and right? Because that part is not developed. So now the prefrontal cortex, that place that talks about the stop and yes, go, don't go, right? Is that place that talks about the executive function. So that is the part that we also want to help, especially when we are managing people who are doing drugs. The changes in brain persist even long after substance is used. Now, in those days, we used to believe that the brain, once it's damaged, thank God we are scientists here and I'm ready to learn, never regenerates. But nowadays, they are talking about neurogenesis for the brain. Am I right? Thank you. Now, this stage, like I said, I think I've mentioned this, the reward circuitry, the dorsal striatum, which is the abuse circuitry. Next slide, please. Okay, I've talked about this one. Now, I just hope you can see the white section here. When someone takes a substance, you know, the dopamine is released from the ventral tegmental area, and then it goes straight to the nuclear accumbens. Remember I said that the nuclear accumbens talk about the reward center. So the brain is open to the new neurotransmitter, especially dopamine. Like I said, there are other neurotransmitters that we play with. We talk about dopamine, norepinephrine, we talk about opioid, we talk about endocannabinoid, we talk about several other neurotransmitters, all right? We also talk about the glutamate and the aspartate, which are basically the excitatory neurotransmitters. So when it gets to the nucleus accumbens, the brain starts adjusting to the new substance, to the overload, to the outpour, to the more than expected dopamine, because naturally we have our own dopamine that is being exposed to. And then when the effect wanes, this area, the amygdala, start feeling it, and then the prefrontal lobe decide, do I want to use the drug or do I want to stay? That is the conscience point. Next slide, please. Please, let's move on. Now, another thing that happens in the amygdala is that after he starts feeling uncomfortable, then the CRF, cortical trophin releasing hormone, releases the stress hormone, which the person starts feeling agitated, the person starts reacting, person starts having some form of palpitation, which is just basically I'm under stress and I needed to relieve the stress. Next one. Next slide. Next slide. Okay, now, this is what we also discover, that if someone had used substance for a long time, but is able to stop, you know, when the person uses the substance for a long time, the dopamine receptors starts reducing, because the negative feedback, what the brain is getting is, you are getting dopamine, so you don't really need to work. So when you check the brain of the person, this is a PET, you'll notice that this is a healthy brain showing that the receptors are still active. But for someone who is dependent on cocaine, you will see that the receptors are down. But after the person stopped for a while, the brain started picking up, all right? Okay, I've talked about this one. Please, let's move on. So I won't waste our time. Let's move on. Let's move on. Let's move on. I've discussed all this aspect. Yeah, let's move on. I've done this one too. Okay, now apart from the brain becoming addicted to the substance, another thing that we find out is the immunological effects of substances in the brain. Now, what studies have shown that though the brain is immune privileged organ, it is protected, it is shielded, especially the BB is the one that is keeping the homeostasis of the brain. But with substance use, we've discovered that they actually compromise this BB. And as a result of that, it triggers the immune cascade, that is the inflammation cascade. And so therefore, the person starts having release of some cytokines, interleukin-2, interleukin-12, interleukin-10. And as a result of that, the brain, there will be some release of some microglia, like those things that start helping the brain to keep the integrity of the brain. There will be release of this microglia, and then the brain is being attacked due to substance use. But many times people are not aware of this. We just believe that the person is is addicted. Next one is to talk about other things so that I won't bore us. It talks about the other forms of trauma that we'll talk about. We'll talk about the addiction. We'll talk about the compromise of the blood-brain barrier. The next one is to talk about the hypoxia that happens. Now, what are the things that can cause ischemic attack or ischemic damage to the brain? We have the cerebral. Don't worry, I'm actually done. Next slide, please. We have so many of these organs destroying the brain directly. They cause ischemia. We have substances that cause stroke. For example, we know that cocaine causes stroke, cannabis causes stroke. Please, we have to be very fast because I've gone beyond this place. Cannabis causes stroke, cocaine causes stroke. We know that alcohol causes stroke. Most of these substances lead to either of the two different types of stroke, which are basically the ischemic stroke and the hemorrhagic stroke. So when people use substance have a tendency to lead to brain damage. That's just the conclusion of what I have to tell us today, either due to traumatic stroke, traumatic brain injury, or due to non-traumatic brain injury. If you look at it now, you see amphetamine, ice that people use now, the umpuru miri that they talk about in the East. You will see how it's causing the hemorrhagic stroke, either due to acute hypertension, aneurysm rupture, cocaine also causing stroke due to atherosclerosis, cardioembolism, reduction of blood supply, hyperaggregation of the platelet. So basically, almost all the substances lead to brain death, lead to brain damage, especially when the person continues to use these substances. Now, where are we going? Please, next slide. Where are we going? Where we are going is that we have substances, especially the NPS, that are very much available now. The NPS are just the novel substances, the synthetic substances that are very much available. Those ones are even more lethal than the conventional one. For example, I was going to give us 3 cases. Here in Babcock, we see some cases of a boy that was brought from town. The boy came in unconscious. When they just told us he used substance. What did he use? Nobody knew. But what we discovered that he was just unconscious, and he did not test positive to the conventional test case, which is the challenge that we have. He never tested positive. And why didn't he test positive? Because the NPS, the plan of the dealer is to make sure that these people do not fail the test. They want to make sure that they escape. So, but unfortunately, these substances are used and people fall into coma. This brought dilemma to the managing doctor, and also it brought frustration to the parents. Next slide. So where are we going to? We also had another case of someone who used substance. After using the substance, he started running up and down. Eventually, he collapsed and he lost consciousness. Why? He used something that we don't know. That is because of the NPS that we are talking about. Then we also talk about what is the African perspective. Drug is with us. It has been with us for a long time. We all know that our parents drank, they used to drink shinap. Even during cold like this, they used to give us shinap. I don't know if some of us remember. You just open your mouth, do and swallow. Am I right? Shinap was with us, 3-3 was with us. Then we had cigar SM. We had bicycles. So this drug has been with us. We had our own alcohol. This drug had been with us for a long time. But what we know now is that we have novel drugs that are with us. It's good that we are gathering here today. I'm not here to prove that I know anything, but I just want to share what we see in practice. It's good that we are here today so that we know the impact of this drug, and it's a neuroscientific conference, so that we'll look into it. Now, please, the previous slide, I've not said anything about that one. The previous one. So why? Drug is part of our worship, part of our celebration. We also need to look at the moralistic view. What do we do in Africa? If I tell you I'm addicted, will you allow me to marry your son? If I tell you I just came out of rehab center. So we look at addiction with a moralistic view, judgmental, then stigmatizing and discriminatory. Then we also criminalize addiction here in Africa. If we want to look at the African perspective, but we also need to know that our brothers and sisters are very ambitious. So we have porous borders where people go in and out with drugs, selling it. We don't have enough data bank. Imagine the last reference, whatever I quoted, was done how many years ago? 2017. So we need to keep working on it. There's depth of expertise treatment center. Then we need to talk to this new generation, the Mafo clan, the Gen Z clan, that addiction is real and it destroys the brain, which we have been able to point out before. What's the way forward? The way forward is let's stop the stigma. The way forward is let us improve research, which is why we are here. Right? Let us go back to the field. Let's go back to our bench to do things, to provide culturally sensitive, affordable care, and also make it a multi-sectoral collaboration. Translational research is actually key, where people in the lab will relate with us that we are in the clinic. This will ultimately help our patients. I hope I've been able to say. Thank you very much. God bless you.
I'm sure that your applause can be louder than what it is. It is not a reflection that they need to come to your center. There is only just one question that I will take. Some people were smiling at me, but like I gave the instruction, I will keep to it. Immediately, I read this. As a neuroscientist, especially in Nigeria, where a lot of our researches are focused on neurodegenerative and neurological disorders. These disorders have prevalence of about just 1% each. According to your presentation, there are several other areas the need to focus on as their prevalence is even more massive. How can researcher get involved in these disorders directly in our locality?
Thank you very much. Incidentally, we have some people that will work together here in Babcock. We've been able to look at the effects of cannabidiol. We've also been able to look at the effects of tetrahydrocannabinol. We are also looking at so many things. So researchers, this is just a call for every one of us to be on our feet. Collaboration here and there, neurophysiologists with neuroanatomists, with public health, with the mental health, with the internal medicine, with so many. We just have to come together. In fact, let me also say that apart from research, treatment of addiction has gone beyond medical. We are getting a lot of people on board. We are getting the religious leaders, we are getting the traditional rulers, we are getting the logistic people. We are just getting everything on board because this is really going out of hand. To be sincere with you, addiction is out of hand. So it's a call for every one of us, especially the scientists, so that we'll see the way out. You know, I mentioned the MPS. Some of them, we don't even know how they act. For example, if I chip, some people are addicted to paint on the wall. Some people are addicted to lizard dung. So how do you do the pathophysiology of that one? Some people, their own urine, some people, their own poop, so many things, paint, so many things. So we need to come on board. Sometimes they use substance we can't even pick in their urine. So we need to come together to see how we'll be able to beat the system to help people that are addicted with drugs. I think I've answered your question.
Thank you. Thank you so much. So just to add to what she said, there are also emerging issues that, and I'm glad that even traditional people are getting involved. I hope that's not Babalao. All right. We are now going to proceed to our breakout session, and I will request that we do that immediately after we take this information. Is there any information? Okay, so it's a breakout session, and I think that should be room 9 upstairs and here. Just a minute. Right, lecture room 2 and 4. Please feel free to meet any member of the LOC to direct you to where those venues will be, 2 and 4, and then this place. Thank you very much and see you by 12 o'clock. Yes, by 12. I've been directed to tell you that those who are sitting to the extreme should proceed to room 2, right? Those who are sitting to the extreme here proceed to room 4. And then those who are sitting from the flower back, you join this group to go, while the remaining will be in this breakout room. Thank you very much and see you in an hour's time.
Yeah, just let me have your attention, please, for 1 minute. The society had the general meeting yesterday, and we came to a conclusion on some issues, which will be communicated to us. But the pressing one that will affect us financially is what I want to say, so that people will not feel so bad about it. At the meeting yesterday, our dues have been jacked up a bit. For the faculty, now we'll be paying 10,000 instead of the 6,000. Then the senior lecturer, and for the lecturer 1 and below, it will be 7,000. And for postgraduate students, we know a lot, there are different categories of postgraduate students. Those who are working, those who are not working, those who are floating, all sorts of, but we don't want to disturb our minds and ourselves. Whether you are working or you are not working, you are paying just 5,000. Only undergraduates will be paying 2,000. I think the category of the undergraduates is the only category that has really enjoyed the increase. It was not almost double because others were. So, and the president has empowered me to tell you that we'll give you till tomorrow afternoon, 12 noon, to pay your arrears. Because as from 12 noon, you know where everybody will be leaving tomorrow. By 12 noon tomorrow, those increases will take effect, proper one. So please, you can visit the desk just on this side of the auditorium at the back where you have the standing banner for the society to pay your dues. And you can also transfer it online. Thank you very much.
Okay, please, let's quickly listen to the presentation that will be taking place in this hall, LRO1, lecture room 1 and lecture room 4. For this particular hall, we'll be taking 4 presentations. Investigative effects of tramadol and coffee co-injection on NICI bodies will be here. Protective effects of Platutus, tuberodium, and Ganoderma lucidium will also be here.
You have posted it already. Just tell them it's in the group and tell them this session will be, because now
It's all stated.
they need to know these people are here.
Okay, please, you can quickly check the NSN platform. We have posted it there. So if you, this is 600 seater. So if you are there, you are presenting here. Then lecture room 1, please, let's quickly move now. Your presentation is already there. And those in lecture room 4, let's also move quickly. You can check the NSN conference platform to know where your presentation will be. Thank you very much.
Hello? Hello, good morning. Okay, let's settle down for, I think we have 4 talks today. Just 4. And I will still stick to yesterday's time because you have to have equal, how would I call it now? Equal opportunity. So whether it's for one person, I will stick to yesterday's time. Technical, could I see the list that is here? So I just, the list not of everybody in here. Oh, we are trying to sort it out. Okay, so I should wait. You only have the slides. Should I wait? Give you like 3 minutes. Okay, thank you. So let's just give us a few minutes for the technical people to talk that. If you know you're among the 4 that was listed the other time, you could come to the front. Prof wants you to sit beside him. If you're among the 4, you better come now before I identify you later. Prof wants you to sit beside him. So Prof is here, you see, and say hello to Prof and sit down. Ah, there are even 2 profs. So sit, and enjoy yourselves. There's 2 more. Okay, just give us a minute. Let's those people. If you know you're supposed to present here oral, I was told we have 4 presentations here. Yeah, we skipped one talk. Today. Yes. Warning, in your minds, be thinking 5 minutes, because that's what we did yesterday when we had many people. So we will still stick to the 5 minutes. So start, in short, you will have opportunities from yesterday. Technical, are you ready for me? Remain small. Jara. Don't worry, we'll keep this time, we'll meet up, we'll keep to time. Maybe we would increase your time with questions. There are supposed to be 4 persons seated here. Okay, while they are on, I think I want to join my president and my treasurer to remind you to commit to your membership. Don't be a floater. If you're benefiting from us, or rather, how would I put it? If you are benefiting from the society should also benefit from. But seriously, I think I would like to encourage everybody as much as possible, try to commit to your membership. We have to grow our local society. Sometimes I always think SFN, the one that belongs to America, it's just America, is the largest, in quote, by individual member, the largest neuroscience society. It belongs to one country. I know America do their things in excess, but that we are the largest in Africa. Agreed, but maybe our pocket is not saying we are the largest, but our number says we are the largest, crazily the largest. So please commit. You make some of all these sacrifices. And for the other ones, you need to join ISN. I know I've been consistently an ISN member since 2012. Consistently, I've not broken 1 year. That's why when they give me grant, I'm happy. When I do their work, I'm also happy. Although there are other ways, I think for like 7 out of those 10 years, I didn't pay fee. I applied for economy waiver. You can do that. You're a student, I didn't pay. I started consistently paying fees 3 years ago. So it's that. So take your membership sometimes seriously. These are where these opportunities could be. Technically, if you're not ready for me by now, I would behave like a proper worry person. Me and you go wear one trouser. Forget all these things. So I come from Wafi. Okay. Who is the first one, please? Why you? Do I know I have time? I should give you 2 minutes when I gave you 3 minutes originally. Okay, I'm counting the 2 minutes this time. Okay, sorry about that. I understand they're probably trying to reshuffle a few things, and we do have a little bit of time. I think we missed one of the talks, so we would leave here on schedule, since I will still stick to my 5 minutes from yesterday. The people from today are trying to bribe me to give them more time. No. Mekavia is my jean trouser. I need to fight. Oh, they are here already. Okay. Okay. Thank you. Abolari Patrick, is he here? Is that you? Is he here? Eh? Your names are not here. Your names are not here. No. This is not our own. The first one was investigating something. Could you just give me your titles or something, please? Your title, I'll call it out. They will look for it, so we don't waste time. Title, I'll call it out. They'll look for it. Just help me identify your abstract. I think you get to be 64. I think who is 65? What's the number? What's the number? What's your 5? Technical, let's just go on. The first one is investigating effects of tramadol and coffee co-ingestion on the Nisi bodies of cerebellum and hippocampus CA3 and behavior in rats. Okay, Dr. Mr. Not sure, Umana, you have the floor. Because I'm a sticker a bit for rules. Everybody have been telling me to give you more time, but I can't. I gave the people yesterday 5 minutes, so you have 5 minutes. Out of that 5 minutes, 1 minute is my own. So 4 minutes plus or minus. Yesterday we had more people, so we had 5 minutes. Today we have less people. We have to stick to 5 minutes.
Watching. Okay. Good morning, panel. Good morning, neuroscientists. My name is Dr. Samuel Umana. I'm going to be presenting on the investigative effect of tramadol and coffee co-ingestion on initial bodies of cerebellum and hippocampus CA3. behavior in rats. Next slide. Okay, I'm going to be using this model to explain the abstract. Tramadol and coffee is being given to Wister rats, and then we'll now look at the neurobehavior, and then look at the tissues, the hippocampus and the cerebellum. And there, we want to look at in the cerebellum and the hippocampus, we want to look at the initial bodies. If they are or staining intensity to bodies. All right, then, by introduction, a lot of people, especially young people, take not just tramadol these days, but in combination with other drugs, take tramadol with alcohol, take tramadol with other things. So we are trying to investigate the combination of tramadol with coffee. So this has to do with substance abuse. So substance abuse is attributed to some of these neurodegenerative disorders. So tramadol is an analgesic drug that is used for the treatment of pain. It could be renal colic, it could be toothache, could be post-operative pain, cancer pain, any of that. And then coffee is the most regularly consumed caffeine-containing beverage. And caffeine has been known for used as an analgesic adjuvant and also as stimulants. Next slide. All right, so we'll be using the following. We used 45 animals with an average weight of 190 grams. The animals were divided into 9 groups of 5 rats per group. Got ethical approval from Faculty of Pharmacy, University of Uyo, consent from NDLEA, Akwa Ibom Command. Tramadol and coffee were gotten from these manufacturers. Continue. Next slide. 10 grams of each of this powder was dissolved in 100 ml of phosphate bufosal served as the vehicle of administration. And then the median lethal dose at the LD50 was done using the LOX method. And then the dose calculation was also done. Next slide. So this is the design. We have the control, normal control. And from the LOX method, we could get the low, middle, high doses of coffee. We got the tramadol. We used 200 and 400 milligrams of tramadol. We had to do the calculation and get the actual doses there. And then the combinations. We just did a random combination of the tramadol and coffee. And we gave the administration for 28 days. Continue. After the 28th day, the 29th day, neurobehavior was done. The novel object recognition test was done. After that, the animals were sacrificed using ketamine, 50 milligram per kilogram intraperitoneally were given. And from then, the animals were perfused with phosphate bufosal and phosphate buffered formalin. The brains were sized and then fixed in phosphate buffered formalin for tissue processing, and then cruciferous violet, 10% buffered formalin for tissue processing and for staining, the cruciferous violet staining. Statistical analysis was done using the ANOVA. The next slide. All right, for the body weight on the organosomatic index, looking at the administration, okay, we in the organosomatic index, the combination group showed significant increase compared to the control. Next slide. Then the novel object recognition test, we could see the total exploration time. An animal shouldn't have a minimum less than 20 seconds. So, but in the total exploration time, we had a significant decrease in the combination groups. And then the discrimination index showed decrease in the combination group compared to the control. Next slide. Then looking at the initial bodies of the cerebellum, we could see that the control, we had a high staining intensity, also in the low coffee, middle coffee, high coffee a bit. But looking at these combination groups, we see a reduced staining intensity of the initial bodies. The next slide. Also for the hippocampus, we had a reduced staining intensity in the high coffee and also in the combination group, the MC plus T2 combination group. You had a reduced staining intensity compared to other administered groups. Next slide. So in conclusion, we could say that the combined administration of these two drugs had more deleterious effects than the single doses, as they caused reduced novelty recognition, and also they damaged the cerebellar and hippocampal tissues as a result of the reduced intensity of the initial bodies, respectively. So the implication of this study is that the co-ingestion of tramadol and coffee is detrimental to the brain, which was manifested in the tissue and the behavior. Therefore, advocacy should be done such that prescription or abuse would be abolished. Next slide. Our future findings is to look for medicinal plants that can actually ameliorate tramadol intoxication and also discover long-term links between all this substance abuse with neurodegenerative disorders. These are my references. Continue. I want to acknowledge the next slide. Professor Anoze Ngiri, my supervisor, Dr. Idorin Moore, Prof. Akmane Kanem, and Dr. Muzi Sekong. Thank you very much.
Thank you. Maybe I should explain why I have to stick to 5 minutes a little bit further. Per Nsn tradition, these are scored, and I think there is a prize or something at the end of the conference, I'm not sure. So since there was 5 minutes yesterday, I have to give 5 minutes today. And warning, if you see that 5 minutes, the people scoring, they have been instructed by me to score it down. That's just a warning. So, but good talk. Any questions? Questions? Okay. And my rule in my heart, if you guys don't ask questions, I will ask you. So, please.
Okay, thank you, Dr. Mana, for that presentation. So I would have thought that, you would have, or you should have used many or more behavioral paradigms than you just checking using NOR. There are several behavioral paradigms that you could work with, you understand? And even your histological assessments, you just went for perceived violence staining alone. You could, it's very difficult for you to say your results is just because of, as in your findings, it's very difficult for you to just pin the outcome of your findings to this, just doing one behavioral analysis, and now you are now generalizing your findings based on that. So you could do a number of behavioral analysis, then compare what you see, and that would help you to drive home your points and respond.
Actually, this is just a part of the work. We did TMA spontaneous alternation test with this, and also we also looked at immunohistochemical distance, GFAP, NSE. Also did the histology. We did biochemical analysis, oxidative stress markers. Also, this is just a part.
You could have added a little more. That's what she's saying, a little more. Then I have a question. You use coffee, as in the regular coffee that we know.
Yes.
The one we buy in the market, the one in the sachet, yes, or in the cup.
The one in sachet.
The one in the
caffeine containing.
I know it's caffeine containing. Okay, this is just a suggestion. I think you should have be very specific to state the source and maybe tell us how much caffeine or caffeic acid or whatever it is that normally stays in that coffee. That you understand what I'm trying to say? Okay. No, it's there. It's listed on the coffee cup. I'm very sure it would have its content, right? You understand what I mean? So because they just said coffee right now, we're not too sure how much of the dose. You understand? Okay, thank you. You can, any other question? Or we should let him, okay, so we should let you go. Thank you. Good talk. So my next talk would be, hold on, please, guys, pardon me. Pleurotus, right? Protective effect of Pleurotus tuberosum and Ganoderma lucidium polysaccharides on lead-induced neurotoxicity in Wister rats. And that will be presented by Sobowale Michael Tosin. We should clap now. Thank you. You have 5 minutes officially.
Thank you very much. Good morning, ladies and gentlemen. Standing before you is Sobowale Michael Tosin. I'm from Pharmacology and Therapeutic Department, University of Ilorin. Next slide. I'll be presenting on the topic protective effect of Pleurotus tuberosum and Ganoderma lucidium polysaccharide-rich extract on lead-induced toxicity in Wister rat. I'll be guided by this outline. Next line, please. By way of introduction, lead is of public health concern due to their toxic effect on bone, blood, and brain and in kidney. Chelating agent that can be used in management of lead acetate are DMNC, penicillamine, and calcium disodium EDT. Qualitative and quantitative analysis show that mushroom contains some biological active compound, which include sugar and amino acid. The presence of polysaccharide in mushroom suggests that they can be used as an natural health promoter against parasites, bacteria, and viruses. Next slide, please. This is the diagram showing the picture of Pleurotus mushroom, Pleurotus tuberosum mushroom. Next slide, please. And also, this is the diagram showing the picture of Ganoderma lucidium in its natural habitat. Next slide, please. Justification of the study. The current chelating agent have toxic secondary effect and unavoidable in a resource-poor country. So therefore, Ganoderma lucidium and Pleurotus tuberosum are available as an antioxidant nutraceutical, and it is possible that their consumption will prevent lead-induced toxicity. Next slide. The aim of the study is to evaluate the preventive effect of Ganoderma lucidium and Pleurotus tuberosum mushroom polysaccharide-rich extract on lead-induced toxicity, while the objective are to evaluate the preventive effect of Ganoderma lucidium and Pleurotus tuberosum mushroom polysaccharide-rich extract on lead acetate-induced neurobehavioral deficit and hippocampal neurodegeneration in rat, and also to evaluate the preventive effect of Ganoderma lucidium and Pleurotus tuberosum mushroom polysaccharide-rich extract on lead acetate-induced brain dysfunction in rat. Statistical analysis were done using graph path presimplified that is under the specific objective, materials, and method. This is the treatment protocols that were employed during the study. 6 groups were used. Number 1 is the group 1, which is the control. Distilled water were given to the animal. The test period was 28 days, and the number of rats per group were 8 animals. The group 2 was lead acetate, and the lead acetate was given 25 milligrams per kilogram body weight. The group 3 were given lead acetate in combination with Pleurotus tuberosum, 25 milligrams and 100 milligrams of Pleurotus tuberosum and Ganoderma lucidium, respectively. Also, the standard drug, DMSA and penicillamine. Next slide, please. Then, these are the analyses that were done during the result: antioxidant, inflammatory neurotransmitter, apoptotic marker, and histology. The statistical analysis was subjected to graph path using graph path presim 5. Next slide, please. Result and discussion. Here is the table showing the result of the neurobehavioral result. 3 neurobehavioral results were carried out. Number 1 is the YMT, number 2 is the TST, and the number 3 is the sucrose splash test. In the YMT, we can see there that the animals that were given the Pleurotus tuberosum and the Ganoderma lucidium extract, they have more correct alternation when compared with the lead-induced toxicity, which is group 2 lead only. Also, in the sucrose splash test, the animals that were given the extract have more grooming time compared with the animals that were given lead acetate. Next slide, please. Here is the result showing the antioxidant parameter. Here we have about 5 antioxidant parameters: MDA, nitrite, GSH, catalase, and SOD. In nitrite, the animal that were given the extract, the mushroom, Gandama lucidum, and Pleurotus region, have a low level when compared with the one that were given lead acetate only. The level of MDA in this result show higher the one that were given lead acetate. Please, next slide. 3 brain regions of the animals were employed: the striatum, prefrontal cortex, as well as hippocampus. In the nitrite, the animal that were given the lead acetate only, they have higher level when compared with one that were given the extract of the mushroom, which is Gandama lucidum and Pleurotus region. This is another antioxidant parameter, the GST. Here, the extract, the mushroom, was able to increase the level of the GST in the group that were given the mushroom extract, that is the Pleurotus region and the Gandama lucidum in striatum, prefrontal cortex, as well as the hippocampus. Next slide, please. Here is the next slide. This is the result showing the catalase in the striatum, prefrontal cortex, as well as in hippocampus. The result was similar to what we obtained in the GHs. The animal that were given the extract had higher level of catalase in striatum, hippocampus, and as well as prefrontal cortex. Next slide, please. Then this is the result for the SOD parameter. You can as well see that the animal that were given the extract, the group 3 and group 4, have the higher level of SOD in striatum, prefrontal cortex, and in hippocampus. Next slide, please. Here is the neurotransmitter enzyme. In this neurotransmitter, we analyze for the acetylcholine esterase. The animal that were given the acetylcholine esterase, the level that were given lead only, have higher level of acetylcholine esterase when compared with the one that were given the extra. Please, next slide, please. Here is the result showing the neurotransmitter for the glutamic acid decarboxylate. The animal that were given the extra, the Pleurotus region mushroom and Gandama lucidum, have higher level of glutamic acid decarboxylate when compared with the one that were given the lead acetate only in striatum, prefrontal cortex, as well as in hippocampus. Can you go to the next slide, please? Here is the result for the neurotransmitter for the monoamine oxidase. The animal that were given the extract have higher level also. The extract was able to increase the level of monoamine oxidase in the Pleurotus region and Gandama lucidum when compared with the lead acetate only. Next slide, please. This is the inflammatory enzyme to know the effect of the extract on the brain region of the animal, the striatum, prefrontal cortex, and hippocampus. The animal that were given the lead acetate only have higher level of inflammation when compared with the animal that were given the extract, that is the Pleurotus region and the Pleurotus region and Gandama lucidum. The group that were treated with the extract has low level of inflammation, that is the group 3 and group 4, most especially in the striatum and in the prefrontal cortex. Next slide, please. Here is the result showing the neurosignaling molecule, the dopamine and the serotonin were analyzed. The extract, the group that were given the extract, the mushroom extract, have higher level of dopamine in striatum when compared with the toxicant, which is the lead acetate only in striatum, prefrontal cortex, as well as in hippocampus. Next slide, please. Also, this is the result showing the neurosignaling molecule in the serotonin. The group that were treated with lead acetate only had a lower group, but the extract was able to increase the level of serotonin in striatum, prefrontal cortex, as well as in hippocampus. Next slide, please. The apoptotic marker we analyzed for caspase 3 and caspase 9. The group that were treated with lead acetate only had higher level of the caspase 9, but when the mushroom extract was administered to the animal, the extract was able to reduce the effect of caspase 9 in the group treated with the Gandama lucidum and Pleurotus region, especially in hippocampus region of the brain. Next slide, please. Here is the result showing the caspase 3 in striatum, prefrontal cortex, as well as in hippocampus. The group that were also treated with the extract, we can see that they have low level of caspase 3 when compared with the one that were treated with the toxicant only. Next, please. Now, this is the histology section of the brain, the striatum section of the brain. Here, the blue arrow shows that there is normal neuronal cell, including medium, while the black arrow shows that there are some blotted and degenerated neuronal cell. Next slide, please. This is the result of the hippocampus section of the brain. The white arrow here shows that there was normal structural organization of the cornus harmonic, including CA1, CA2, and CA3, while the blue arrow shows that the hippocampus with mild distortion of structural organization. Next slide, please. This is the frontal cortex section of the brain. The cortex shows normal lamina in the blue arrow, while the black arrow shows some blotted and degenerated neuronal cells, especially in the group treated with the lead only. Our discussion. In this study, our result demonstrated that the Gandama lucidium and place of the baridium elicited the recovery of anxiogenic and depressive profile, as well as short-term memory damage induced by lead toxicity. MDA and nitrate value in lead-treated group significantly higher than the control group, but the extract was able to increase the level of GSH, SOD, and catalase. In compared to lead-treated animal, all pro-inflammatory were decreased markedly after administration of GFPS and PTP in both groups. This shows that Gandama lucidium extract and plutonium have anti-inflammatory impact against lead-induced toxicity in brain. Next slide, please. Our results show that Gandama lucidium and plutonium significantly suppress all signal transduction process induced by oxidative stress. In conclusion, our study characterized protection by Gandama lucidium and plutonium, and it also gives us a new insight to the neuroprotective mechanism of Gandama lucidium and plutonium polysaccharide-rich extract. Acknowledgement to God Almighty and to those who contributed in one way or the other to this project. Here are my references. Thank you for listening.
Thank you. Very oratory, if my language is correct. Can you hear me? It feels like, is it working? Oh, you understand the difference, right? It felt like I was not hearing myself. Okay, nice to hear you. Before that, I think I'll just list a few corrections so you ask questions, because for most of us who are experienced, we noticed some errors, some very correct, which we should correct. And that's one of the things about coming to conferences, so that you don't bother. I know some of you, so we don't bother trashing that. So let me start with the slide on MDA. Be fast, quickly. They are not there. I know these technical people. They keep going. And okay, let's start from here. Oh, you were saying there was difference, but there's nothing indicating there was significant difference. You could either say we see a likely increase, but not, you get what I'm trying to say, because this look blank. So this one is simple. Next, start coming down quickly. Oh, I will hit them. Continue. Okay. This is neurotransmitter, and okay, this is what? Acetylcholinesterase. So I will correct myself. I was confusing with what you had when you call them neurotransmitter enzyme and neurotransmitter signal. Okay, this is the enzyme. So the slides on your photomicrograph, keep going. Go. I will eat them. Is somebody doing the H&E slides? Okay. Leave it at the hippocampus. We can do that. Hippocampus, please. Okay, here you say including, but your section does not tell us whether it is CA1, CA2, or CA3. I understand that you may have looked at all of them, and then your representation is one, but you should specify which one we are currently seeing. And it is obvious to everybody in this room that your last group is not a brain section. I don't know if you noticed that.
No, it is pre-special, it is manifestation that differ.
No. Even in our dreams, we know that that is testis. You understand? It's a major error. Please go back and check this one. You are seeing this one. If you wake most of us from our dream, we'll know that that is testis. Okay, so I don't know how that got into there, but for accuracy's sake, that is not correct. Okay, questions. So I've corrected all these small things that all of you are doing your eye like this for. Now come and ask questions. Okay, thank you.
If I may get you right, you are trying to say I should compare the result in monoamine oxidase and dopamine. Yes, let me start with dopamine. We all know that dopamine, when there's a presence of, when there's an absence of dopamine in the body, when the level of dopamine is reduced in the body, it can cause a cognitive, reduce the level of cognitive memory in personality. So we are trying to see if the extract can increase the level of cognitive memory in the animal that were administered with the mushroom. So when we administer the Pletostobaridium, which is the conventional edible mushroom, to this animal, we find out that they were able to increase the level of dopamine, which will help their cognitive memory in normal human being. Similar thing is what we got in monoamine oxidase, because there are neurotransmitter that can help us to know whether they're able to transmit the signal or synapses to coordinate it within the sense. So that is what we did, sir. Thank you very much.
Advise and look at your work again. Okay. No other questions. Thank you. You have a question. Okay. I don't know why I don't have a spare mic this time. Okay, it's here. The spare mic is here. Before your question, I want to say something very funny. These people sharing me tickets, they should remember we are here. That's how they forgot us yesterday. and today they've given warning that once they finish sharing, they will not share again. So we are here, bros. Don't mind me. You know, sometimes the worry has to show. Okay, your question.
Okay, so in your slide, you mentioned the other you forgot to mention, you did not mention G lucidium, like where can we get it from? The other one is from mushroom, but the G lucidium, you did not mention it in the slide. Oh, they are both mushroom species. It was just stated that the other one was a mushroom species. Okay.
Did you co-administer the two species, or you try to look at them differently in conjunction with lead? You separated them. There was no group that was given maybe after you given. Yes, I'm saying this because of your topic. You said something pleurotus, tuber, whatever, and G lucidium, as though the two were matched together. So you gave them separately as an independent.
So when we now get to the treatment group, they were different.
They were different and they were given separately, I guess. Lucidum and lead, or rather, and the other one and lead, not co-administered. Okay, it's fine. Oh, now they cooperated today. Yes, nice. Thank you. I don't know my next talk, so you would have to put it up. Technical. Are you the one? Just come and tell me your abstract. Let me know. That would be Alex Gim. That would be assessing the prospects, and strengths of computational neuroscience among African students and researchers from Alex Gideon. Solo. I will learn it.
Good afternoon, everyone. So I'm Alex Gideon. I'll be looking at to assess the prospect and strengths of. So I'll be looking at how to this topic is about to assess the prospect and strengths of computational neuroscience among African students and researchers. So computational neuroscience is the bridge between neurobiology, psychology, statistics, and artificial intelligence. Computational neuroscience is also the interdisciplinary field of the development, emulation, analysis of multi-scale model, theories of neural functions from level of molecule through cells network up to level of cognition. So it's about what brain computer interface, how the brain, how computers or the brain actually interlinks together. Next slide, please. So this slide now is about Artego device developed by an MIT student. So on this device now, the user don't need to actually press a phone. You just have to think about something you are trying to buy or you want to search something online. So you just think about it, and then it will be connected to your phone device or to your system, and then it will be searched. You can even order items online, either on Jumia, on any platform. It's just by thinking about it. Next slide. Okay, so our aim was to understand the prospects and strengths of computational neuroscience among African students and researchers. So we also want to assess the current scenarios towards unraveling the factors that limiting the development and advancement of the field in Africa. So we know that the field also has many challenges. It's actually rising in Africa, and it's actually very new to some of us here. So our statement of problem was to assess the paucity of computational neuroscience research and facility in Africa. We're actually very poor in this field of Africa. We actually nowhere there. So please, so methodology now was that we actually form a structured survey designed to assess in total, we had 86 persons across Africa that we actually got their consent in our survey between April to July this year. We actually started April this year up to April. It's still ongoing. We hope to use more time to get our data, so we're still working on it. So between the age of 20 to 50, we're actually looking at young people, early career people up to 50 years of age. So we obtain our data and then we analyze our data. Next slide, please. Okay, please. Next slide, please. Okay, so here is it now. We got data from Cameroon, from Egypt, from Ghana, from Kenya, from Nigeria, from Rwanda, from Senegal, from South Africa, and Tanzania. So in this research, most of the stuff we actually had are from Nigerians. We are struggling to get more data from other African countries because I remember I actually sent our data, our survey form to some African countries, and they're like, this from Nigeria, we are actually scammers, so they can't fill our questionnaire. So these are some of the challenges we are having. All right, so between age 21 to 30, we had 82% of persons actually in our form. Then between 31 to 40, these were the data. Then the highest qualification we have from BSc, current undergraduate students, MSc, PG, and PhD students. Next slide, please. All right, so here's our data we go. So we actually, on our data now, we actually asked our participants to rate their current level or knowledge of computational neuroscience between the scale of 1 to 5. So 5 was actually the highest. So we saw that between 1 to 3, that was the level of the people had rate the current level of neuroscience. So 1 was the lowest level. So 1 there was it meant that on the slide there, so many persons actually don't have knowledge of computer neuroscience. Then the next one there was rate your current interest in the field of computational neuroscience. So 50% of persons had more interest in computational neuroscience, and then 19.8 and 18.6, that is from 3 to 5. They were more interested in computational neuroscience. They were more interested, but then they don't have any background in it. So over 8.1 and 3.5 persons were saying that, okay, I'm not interested in computational neuroscience. So more persons that were actually interested in neuroscience. Over given in Africa is very over from 1 to the attention given to computational neuroscience were very, very poor. And then we just have 7, 4, and 5. We're saying that, okay, we see that because we have some other schools that are coming up now. Trent just did one in Accra, Ghana, then in Bizo, and then Brain Project. They also, most of the funding opportunities in Africa were coming from European nations funding projects, workshops, and training schools in Africa. So within us in Africa, the attention is very, very low, and then we've not done anything yet. Please, next slide. Next slide. So look at, okay, if there's a school, if there's a workshop that is being organized, they should rate their current level. Okay, would you like to attend? So we saw that between 3 to 5, we saw that most persons were actually interested. Up to 70% of the persons were actually interested. Okay, if there's a school in computational neuroscience, I would like to attend, but there's no school. It means that we still have a long way to go. They will not say that, okay, read the possibility that you will choose a career path in computational neuroscience. More African persons, more young people say that, okay, I would like to take a field in computational neuroscience. That means that if the field is in Africa, many people would like to venture into the field. Next slide, please. All right, so from our discussion, we saw that the result of shows that the African students and researchers, they are very enthusiastic about computational neuroscience, although the knowledge of the field is still at the infancy. We've not gone anywhere, we've not started. And then it's also important to know that the rising interest in computational neuroscience is very positive because more people are actually interested in it. It means that we still have a future. There's a future ahead if we venture into it. Many people want to go and then bring more innovations into it. So computational neuroscience is very positive, and if properly developed, will result into scientific intervention and then artificial intelligence in the future. So these are next. This the world is what we are going to. So it's also encouraging that we all look into it. But then our recommendations now, we also recommend that we should incorporate a computational neuroscience research group into African academic institution, not like our normal conventional research we keep doing in Nigeria, normal histology and all of those things. We actually have to take up to the next stage. So we also do that we support and organize local-based training in computational neuroscience in Nigeria, and not just in Nigeria and other African countries. So instead of Trent selected 200 persons around Africa, and then we are more than 500 million persons in Africa. So within us, Nigeria, and within each state, we can also organize those things within us, within the graduate system. Most people can have access it, because the one trend that organized was very, very competitive. So just 200% were able to access it all through Africa. So we can also organize local-based stuff in Africa. So set up institutional labs to equip students. Most of us, or our senior professors and our mentors, we have labs where we bring students, but then there's no in computational neuroscience. Like, as of when I was doing this research, there was no publications, like nothing I could access from Africa in computational neuroscience. So we make funding available for African students to attend international computational neuroscience workshop, conferences, and then summer school. Most of our undergraduate students, we can network with them and then organize trainings and then from that team. Thank you very much. Next slide. Next slide. Okay, I just want to acknowledge Mr. Amadi, Dr. Shina Ulrich, Dr. Berwigo, and then I'm a member of Youth in Science Association, and then Nesula for sponsorship coming to the conference. Thank you very much, sir. Thank you very much.
Thank you. You're picking up interest, but don't call the other ones conventional. They are not conventional. They are just different lines. Okay. There are questions, but before questions, just a few things for you to note. I'll take questions. Technical, go up, 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 up. Okay, the pie charts, just small things you should correct. Like your age, you started 21 to 30, 31, 40, 41 to 50, and you now go below 20. You should be, you understand now, it should be a BSc, first degree, currently undergrad. You understand, right? It should a little bit systematic. Now, questions, okay? Where is my... And hence, the color separation are a little bit too close with your red and orange, but that's good, huh?
My concern is actually from the age range. Why 50? 20 to 50.
Let me even say, and you said you said young.
And you said young.
Okay. It's 50, 45. Okay, maybe they are still in Nigeria, everybody's a youth.
Okay, please. The topic is about African students and researchers. So even at 50 people, so between I should noted African students and researchers, but our age range, because most people above this 50 do not be actually interested in us. If I send a survey to them, they might not really have that time, and they might not be really interested because they are very, very busy and all of those. Okay, let's 20 and to 50, they might actually look into what we are doing. Yes, exactly. Thank you.
There's another question. Oh, that means we have to go back and be learning maths. You know, for some medical schools, people always knock off maths after year 1 or year 2, as the case may be. Okay, and even statistics. Yeah. Okay. And maybe you should ask another question. How these people like maths? Somebody wants to do computational neuroscience. You should poke a question about their interest in maths and statistics. You understand? Subject, I'm just my contribution.
Next slide.
Next slide.
The slide showing the branch. The next one. Like they have gone. Yeah, I just wanted to point out that on those bar charts now, the color, you use the same color for all those. You know, I think you should have represented them. At the bottom. Did I see? Okay. Yes, we can't even see the slide. The next one, please. Oh, okay. I was going to say for those maybe the bar charts, you use the same color for all of them. You know, we couldn't contrast which one is going up, which one is coming down. Okay. See, though you put the percentage, okay, but still okay. The percentage has done that. Yes.
Any other question? No question. Thank you very much. There's a question. I have time. All of you that are, I have time. The other people, I'm sure they have not gone half, so I still have time. Or LOC, don't I still have time? There's supposed to be 4 presentations. Oh, sorry. I cancel your question by the virtue of me. You can take the question. Okay, ask.
Okay, so computational neuroscience, in some parts I'm interested in it because a few years back, I spoke with some of my friends about something that Ategio device talked about.
Yeah.
But the challenge you have, or that we have, is translating it into, like, bringing it into use, like translating your research. So I wanted to ask, how easy is that in Africa currently?
One would not happen.
Okay, let's not even say Africa, like neighboring continents. Like, do you have any idea on how they go from doing the research and translating it?
Yes. You know, there's a link between the science aspect and the technological aspect. We are doing the science aspect, computational neuroscience, and then there's BCI technology, brain-computer interface, just like cochlear implants, prosthetic technology, and a lot of those things. So the technology is in use, and it has gone further than we would expect. So it's in use.
We are behind because you people end up jackpotting all the time. And when you jackpot, you don't remember us. All of you here, if you do jackpot, you don't know. You will see me in your dreams. Yes, you will see me. I will be calling you, come back or send something back. I'm not saying don't jackpot, but remember that we have to grow. Okay, thank you. There is supposed to be 4 presentations in this place. We've only had 3. Is there one more person? It's you. I've been looking for you. And it's not fair. You have 5 minutes, you will keep to it. I will not shift to the left or shift to the right. What's your top? Have you given it to me? This one? Okay. So please, he will present on effect of Nigeria bitter honey on brain biochemical parameters, histology, and neurobehavioral parameters following aluminum chloride induced memory loss.
Yes, good morning, everyone. I am Hadi Ayodej David from the Department of Physiology, Babcock University here. Please, next slide. Just move on. One of the most important functions that is peculiar to humans is the ability to acquire new informations to alter behaviors. And that is what learning is all about. Now, memory, on the other hand, is the ability to store acquired informations. However, memory loss is becoming a major concern, and that is one of the reasons why this study was designed. I don't want to go into this. I believe we all know it. I will just go straight to the statement of research. But before I go there, I would like to say that in Nigeria, the attention has been focused, much more attention has been focused on traumatic brain injuries, neurodegenerative diseases, among others, side effects of medications as part of the majority of the factors contributing to memory loss. But recently, researchers have focused on the toxic which are in abundance in our environment, examples of which we have aluminum, cadmium, lead, among others. Please move to the statement of the research problem. All right. The World Health Organization estimates that 50 million people worldwide have memory loss, with over 60% of these individuals residing in low and middle income nations. And according to a research, to an output by the WHO, the total number of people who currently have memory loss in this country will be doubled is expected to double by 2050. All right. Now, bitter honey, on the other hand, I'm sure everybody must have come across one or the other. But the fact is this: bitter honey is a little bit different, but the difference is not really conspicuous. Bitter honey is, let me tell us what happens to honey. Honey is produced based on the availability of the plant around. Yes, the plant, the nectar around is what honeys use to produce, honeybees use to produce honey. Now, bitter honey is a species of honey that is produced by some species of plants. All right. Now, it has been shown to possess excellent anti-inflammatory and neuroprotective capacities, and that is what informed this study. Please, can you go to the, yes. The aim is to evaluate the role of bitter honey on aluminum chloride induced memory loss in Vista rat, and the specific objectives are to assess the effect of aluminum chloride on histology of brain tissues, on neurobehavioral parameters, and on biochemical parameters. Please, yes, just go. This is the study design. We have 5 groups of 7 animals each, and group 3 and 4 showed graded doses administered to rats in this group, while group 5 has a reference drug, donepezil, which is a drug that is used to treat neuroinflammation. And all this happened within the space of 14 days, please. Yes, let's quickly go to the results. Now, this is the result for the number of entries into Y-MACE neurobehavioral apparatus following the administration of aluminum chloride. And we could see a significant reduction in the number of entries in group 2 animals, which is somehow reversed in group 3 and significantly reversed in group 4. Please, next slide. This is the one for the spatial alternations in Y-MACE. We could also see it is significant in group 2 relative, and it was reversed in group 3 and group 4 significantly. Please go, move on. We have numbers of line cross. We wanted to know locomotion as well, because locomotion also can be traced to neuroinflammation and memory loss. And then you could see it's also significant, especially in group 4 at 400 milligram per kilogram body weight. Please move on. And then we have the T-bars, that is for malonaldehyde. We could see this is conspicuous, you know. The malonaldehyde, because of lipid peroxidation, we all know that it is high compared to the others, and it is low in others. The next one is for GSH, that is glutathione. Please move on. And this glutathione shows no significant difference. Please move up a little bit. All right. It doesn't show significant difference, but we could see a slight rise in group 3 and group 4 animals compared to group 2. Please, let's move on. Yes, this is for catalyst. Now, without wasting time, I would like to go to the histology. Please, let's go to the histology. Histology, the photomicrograph. This is the photomicrograph of group 1 animals. We could see the hippocampus. The cycloarchitecture of the hippocampus is intact. It appears normal and intact. This second one is for the prefrontal cortex. And we could see that the nasal integrity is also normal. The pyramidal neurons population are also normal. Please move. Yes, this is for group 2. We could see distortions. And this is for the prefrontal cortex of group 2 animals as well. Let's go to group 3. All right. It is evident here to let us go to group 4 and group 5. Thank you. Please, can we go to discussion? Yeah. Previous the neuroprotective, ameliorative, anti-inflammatory, membrane stabilizing potentials of bitter honey. And this is one of the factors that informed the choice of this natural product. Now, aluminum chloride is a known agent which has been modeled by many neuroscientists for memory loss for Alzheimer's. Now, aluminum chloride has been known to distort the permeability of the blood-brain barrier. That is number one. Aluminum chloride has been known to disrupt the hippocampal prefrontal striatal loop. We all know that loop. It is very important for the formation of new memories. That is a connection between the hippocampus, the prefrontal cortex, and the substantia nigra pars compacta. I'm sure we all know that this particular pathway or axis has a lot of dopaminergic neurons. So we can relate dopamine with memory in a way, all right? Now, findings from this study reveal the distorted morphology in prefrontal cortex. Now, let's move on. Let's move on. We don't have time, all right? Now, let's move on. Okay, bitter honey has been reported to possess anti-inflammatory properties, which may contribute to its neuroprotective effects. The bioactive compound in bitter honey, particularly flavonoids, have been shown to inhibit pro-inflammatory enzymes like cytokines in order to do what? To reduce inflammation. Please move on. Additionally, bitter honey has also been reported to have immune modulatory effect in such a way that it suppresses the inflammatory response, thereby stopping the progression of what? Of neuroinflammation. Researchers have also shown that brain-derived neurotrophic factors levels increases as a result of bitter honey. And so we could see that bitter honey is all-encompassing. I don't want to talk much. It's all-encompassing in function. This study is an ongoing process, and conclusively, we have been able to discover that bitter honey at graded doses showed some promising ameliorative and anti-inflammatory activities in brain tissues, and we can subject it to further studies. Thank you.
Questions, comments? Questions, comments. While he's going for the comment, you don't want to talk much. Anyways, that's just by the way. But your discussions could be a little bit more concise. Just learn that here, more concise, more brief, so you don't. Questions?
Yeah, so thank you so much for the presentation. It's an interesting presentation. My question does, because you keep mentioning that bitter honey has anti-inflammatory capacity and the rest of it, but your measures are antioxidants, right? So you may want to, okay, let me make that as a comment. You may want to focus on the antioxidant capacity of bitter, right, rather than talking about, and again, you mentioned something about donepezil. I beg to disagree because of the young population that want to plan their research. The donepezil is an acetylcholine esterase inhibitor. It's not an anti-inflammatory drug, right? So, and that is why it's used in. So next time you have to plan this kind of a thing, you may want to check for acetylcholine esterase level, right? Cholinergic activity, if you're using donepezil. But I can still say that it's going to be a good design if you remove donepezil from the story, and then you put an anti-inflammatory if your interest is on the anti-inflammatory capacity of bitter honey.
Okay.
Thank you.
Thank you. If there's one more, I will only take one question. And I was the one that kept the time yesterday. Hey, they finished before me. Okay, quickly.
I want you to identify the difference between the more significant, important, the reason why I use bitter honey compared to normal honey. Knowing fully that in our environment here, people use normal honey to stimulate their memory, and it is an established work in some other literature. So what makes your bitter honey more significant, or what are the things that you can tell us that make bitter honey more significant to the normal honey, and even its availability?
Before you answer, technical, please, could you put the title again on the board, the title of the talk, please, and do it quickly. Thank you. Yeah.
Yes, thank you very much, sir, for the question. Yes, for the choice of bitter honey, you know, I said it when I was about to commence this presentation that honey is honey, but the difference is the nectar around that honeybees use to produce. Bitter honey came into limelight in few years ago in some areas in western part of this country and Africa as a whole. There are some specific trees. I think I made mention of those trees. Please, can we go up a little bit? Please, can we just go up a little bit? Now, let me not waste our time. What is peculiar about this is that these plants, they have been shown to possess a little bit phytochemical components compared to normal honey. We have done a phytochemical analysis. We have done an analysis of this honey, and we have been able to see that although it is still a subject of further studies, but we have been able to see that, take for example, agave species, all right, datura species, all of these, in an area where we have them clustered together, you will see that honeys that are produced there, they are a little bit bitter. And when you taste them, they are actually bitter. But then the phytochemical component of bitter honeys happens to be the subject of our research. And that was why we chose it, because it has some other like additional components, which we have not confirmed anyways, but which we have been able to see over time that has some potentials more than the normal honey.
I think you go and talk to him later because he wants to know how do you differentiate between sweet and bitter honey? Is it that you would have to taste it first, or we could tell from, you are not answering now, my time is up, but these are some of the questions. We could tell from when it's being produced in the honey bee that this is bitter, and you understand what it is. Thank you. And thank you very much for being here with us.
All right, good afternoon, everyone, and welcome back from the other session, and welcome back to the next set of talks that we're going to have. Next on the line is Professor Paul Menga, and he's from the School of Anatomical Sciences, University of the Witwatersrand, Johannesburg. And he's talking that out of Africa, there's always something new. I learned that yesterday his citation was already read, so he doesn't want me to do that anymore. So I think we'll just go right to it without wasting time. Paul?
All right, thank you. And when our technical is ready, they can, there we go, perfect. All right, so what I want to talk about is the work that I've done, or highlights of the work I've done over the past 20 years in South Africa, looking at brains of different African animals. And Pliny the Elder, in ancient Greek, said, out of Africa, there's always something new. And originally from Australia, I've lived in the US, I've lived in Europe. The first 6 months I lived in Johannesburg, every day I saw something new. And every day I've been in Nigeria, and this is my 5th trip here, I've seen something new. There's always something new to see and something new to experience, and that extends to the brains. So could I have the next slide, please? So Africa has a huge biodiversity. About 25% of the mammals across the world are endemic to Africa, around 25% of birds, 20% of reptiles, 15% of amphibians, and the majority of these animals are endemic to Africa, meaning they're only found on this continent. The next slide, please. I've been collecting brains over this time. We have a bunch of brains from the Afrotheria, elephants, hyraxes, golden moles, and other species, primates, rodents, bats, insectivores, the tree pangolins, carnivores, the zebras and rhinos, and many different types of artiodactyls, with things like giraffe, hippos, and some whales, and plus a range of reptile and birds. So we have this extensive collection of brains. And I want to show you some pictures of how we collect, or how we prepare these brains. I want to talk about how we prepare them. We do this. All the brains are perfusion fixed with 4% paraformaldehyde in phosphate buffer. That's absolutely essential, because preparing your tissue is the most important part in any of the studies that we do. They're stored at minus 20 degrees Celsius in an antifreeze solution, and they all have very good immunohistochemical staining, and we've actually sectioned and stained a lot of brains. Now, the next slide. So look at an animal like an elephant. How do we get a brain out of an elephant in good condition, like you would do to an animal in the lab? Those of you that are a little bit queasy or don't like the sight of blood, you should probably look away for a couple of minutes. Okay, can I have the next slide, please? So after we've killed the elephant, we then have to take the head off. This is not an easy job. It requires a lot of effort and a lot of chainsaw, and you get covered in a lot of blood. And the next slide, please. We then take the head to a place where, through gravity perfusion, we can actually pass the fluids through the venous system of the head. Next slide, please. Once the animal is perfused, we then use a chainsaw to take away the bone from around the outside of the elephant's skull. And you can see that the brain sits very low inside the elephant's skull there. All right, most of the skull is actually this big frontal sinus. And you see the brain at the bottom there, and around the spinal cord, I put a little rubber glove there to protect it as we keep going. Next slide, please. We then use specific tools to take the skull away from the brain. The next slide. And this is when the bone is moved away, and you can see the dura mater lying over the brain. Next slide, please. Remove the dura mater, and you can see the brain very nicely there. You can see it's been perfusion fixed. A lot of it's nice and white. The next slide, please. Then you get your hands underneath, pull out the cranial, cut the cranial nerves, and lift the brain out. Next slide. And after a few hours of work in the hot sun, this is the size of an elephant brain. I'm holding it there, okay? It's about 5 kilograms, so the size of a baby. All right, cover the next slide, please. So I want to start talking now about some of the different species. And I started this talk with tree pangolins because I was lucky enough to come here and work with Imam, and Imam did his PhD on tree pangolins, and we caught these in the areas around Olorin. So you guys all know about the tree pangolin, right? So this is work that came out of Nigeria. Now the next slide, please. One of the first things that we found was there was a really unusual decussation of the pyramidal tract. On the right-hand side is a view of the brainstem of a lion. And you see the pyramidal tract coming all the way down, and then it decussates just at the level of the spinal cord, at the spinomedullary junction. However, in the pangolin, you see the pyramidal tract decussates far more rostrally, right about the level of the facial nerve nucleus. There's only two animals that this has been seen in. It was in the pangolin and the Australian animal called the echidna, the spiny anteater. Both of them are insectivorous and they use their tongues a lot to find food. So you can see that much of the pyramidal tract, instead of actually really going down into the spinal cord, is actually going to the hypoglossal nucleus for controlling movement of the tongue. So the pangolins are amazing. Their tongue starts at their hip bones, moves all the way up and comes out about 40 centimeters from their mouth. So they can use it to grab ants with. It's very cool. The next slide, please. The other interesting thing is the pangolin's about 1 meter or so long. It's quite a long animal, but the spinal cord itself is only about 13 centimeters long. So it's a very, very short spinal cord and a very long cauda equina. Even though the spinal cord is short, it's organized in a very similar way to other mammals. It's just incredibly short. And this seems, and the other animal that shows this is the echidna from Australia. And both of these animals roll into balls for protection. So it seems to have something to do with rolling into a ball. The next slide, please. The other thing that was really interesting, and this is immunostaining for tyrosine hydroxylase, was that the locus coeruleus was absent in the pangolin. It's the only species, we've looked at about 50 different species, and it was the only one that had an absent locus coeruleus. It's like all the neurons seem to have moved out of the gray matter into the white matter of the brainstem. It's a rather unusual feature. I'm not sure what it means, but it's definitely something different. All right, come to the next slide, please. Zebras. I talked a lot about zebras yesterday. I'm just going to show you one other really interesting thing about zebras and other horses. Can I have the next slide? The thalamic reticular nucleus. You guys are all familiar with this. It's normally an inhibitory nucleus that provides inhibition back to the dorsal thalamus. In the equids, this is in the mountain zebra, plain zebra, also in the horse and in the donkey that we studied, was that the neurons of the thalamic reticular nucleus appear to be dopaminergic. And this is a very unusual position for dopamine to appear in the brain of any mammal. So there's something unusual going on there. Not sure what it means, but it probably plays a role in the thalamocortical loop and neural processing and altering that. The next slide, please. Here we have an animal called the spring hare. I don't know if you guys are familiar with the spring hare. I think it's more in southern Africa. It's quite a big rodent, about 3 or 4 kilograms, and hops around like a kangaroo. The next slide. What we found in the spring hare was it has a really unusual architecture of the cingulate cortex. The cingulate cortex, being that part of the brain, evolved in modulating emotional behaviors. And can I have the next slide, please? And we see this when we're looking at different stainings. Look at Nissl staining, immunostaining for V-GLUT2 and parvalbumin. You can see that there's an extreme number of layers there, which is very unusual. In a lot of animals, there's only the typical 6 layers, but sometimes layer 4 is not very clear. In the spring hare, we can see layer 4. It's a very dark band going across, a little bit of staining in layer 3. Then in the parvalbumin staining, you see layer 4 is quite clean from that. This is also a very unusual observation in the spring hare, and we really don't know what it means, but I'm just sort of showing you some of the unusual variation that can be found. The next slide, please. The lion, okay? Yes, we have perfused lions, and we do the same thing as we did with the elephants. They're not so scary as you might think. The next slide, please. What we found in the lion and in the leopard, as well as also in tigers, is they have the largest bet cells of any mammal that we've seen. So bet cells are the cells in the primary motor and premotor cortex that give rise to the corticospinal tract. These were absolutely huge in the lion. And I've got a comparison there to a gibbon, which is a large, similar primate with a similar brain size. And those two photos are taken at the same scale. So you can see that the African lion is much larger. In the graph here, the hollow circles on the bottom are from primates. The dark circle, full dark circles are from other carnivores. And then you've got the leopard, lion, and tiger on the top in the stars. So these large, or these big cats appear to have really large bet cells. And this might serve to be able to recruit them, to recruit a whole bunch of muscles during hunting. So they can make major rapid movements of the whole body, like when they're pouncing and that kind of thing, which is quite interesting. Come to the next slide. We also looked at the eye of the lion, and it seems that the lion is the only animal that we've found that has a fovea-like specialization that isn't a primate. And in fact, when we look at it more closely, if we focus on this little pit, you can see there's a kind of pit in the retina, and you focus up and down, you can see the pit in different focal planes. And you can see when we look more generally around that there's a clear pit there. When you calculate the visual acuity of a lion, it sees better than what humans do. A lion can see us from about a distance of 3 kilometers. We can only see a lion at a distance of about 2 kilometers. All right? But the really interesting thing is this fovea-like specialization only occurs in the adult male lions. Juvenile males and female lions don't have it. So it's a really unusual feature that's very specific to the adult males. So it must be something to do with protecting their territory. They need better visual acuity to protect their territory. But the fact is they can see us well before we can see them. Of the next slide, please. Right, here's an animal you guys might not ever have heard of, a golden mole. Has anyone heard of a golden mole? Apart from a mom who was in my lab. He's heard of all these. These are little subterranean Afrotherians. They live under the ground and they dig through the ground, and they grab worms to eat them. You can see they really don't have any eyes, right? They're subterranean. They do have an eye, but it's less than 1 millimeter in diameter, and you have to look really hard to find it. And they've got these big claws at the front that they use to dig under the ground. So they're not moles like the moles you hear from Europe or America or anything like that. They're an unusual species that are actually related to elephants, believe it or not. Go to the next slide. They have a really unusual flexure in their brain. You see what I call a mesencephalic flexure there. Their brain does this unusual curve at the bottom. There's nothing like what you see in many other species. There are a few animals that show it, but not many. And there are only animals that are related to this. So we see the olfactory bulb on that side, cerebellum on that side, and you see that beautiful bend going around. And it seems that their brains have sort of become foreshortened and pushed back together and become a little wider, which is probably something to do with their burrowing through the ground so they can actually push their way through. Over the next slide, please. The other thing we found was that they have what I've called a hypercholinergic brain. They have cholinergic neurons in a lot of places of the brain that you don't find cholinergic neurons in other animals. And cholinergic neurons are related to levels of arousal and also to blood flow. So they have this unusual feature. We're really not sure what it does, but it seems that even though they're subterranean animals digging through the ground, they have a very, very active brain. Something's really going on in their heads. And it'd be really interesting to figure it out. The next slide, please. The hippopotamus. Everyone knows about the hippopotamus, right? Africa's most dangerous mammal, yeah? And I think it's second to mosquitoes in the amount of people that it kills in Africa. All right. But we got brains from these guys as well. The next slide. What we found is when we were staining through the brain of the crocodile, we were looking at the sleep systems, but we saw something really unusual. In the dorsal thalamus, there's a region called the intramedullary nuclei. And these intramedullary nuclei are involved in levels of cortical arousal. And we found that they had an excessive number of cholinergic neurons in there. And we haven't seen that in any other species that we've studied, these cholinergic neurons in the intramedullary nuclei of the thalamus. And this might relate to when hippos suddenly change their brains and they charge and they run you down or they bite you. They're very short-tempered animals and they're very, very quick to react. So this might be a neural underpinning for that. The next slide, please. The rock hyrax. Everyone knows what a rock hyrax is? No, yes, no, maybe. Okay, the next slide. In their brains, they also have an unusual cholinergic system. This is the lateral-dorsal tegmental and peduncular-pontine tegmental nuclei that are typically found in the rostral brainstem, and they're related to the production of waking and to the production of REM sleep. When we look at it, you can see there's two different types of neurons there. There's larger ones and there's smaller ones. So we've got magnocellular and parvocellular groups, right? Nor in most other mammals that we've studied, there are one or two exceptions, but in the vast majority of other mammals, only the large cells are found there. So we thought, well, there's going to be something unusual happening in the sleep of these animals. Now, the next slide, please. So we recorded sleep and we found that there was this novel sleep state that's kind of a mixture of REM sleep and non-REM sleep. We called it somnus innominatus, sleep without a name, because we couldn't think of anything else better to call it. But it was a very clear and unusual sleep state that's occurring in the brain of the rock hyrax that appears to be related to this unusual cholinergic system in the pontine region. Have the next slide, please. Everyone knows a giraffe, right? Can I have the next slide? We looked at the visual system in the giraffe, and you can see it has a beautiful tapetum lucidum, and it's all located on the upper side of the retina. So that's the tapetum lucidum acts like a mirror to reflect photons back over the retina, so the retina has two opportunities to detect light. Now go to the next slide. Then when we looked at the ganglion cells, those that are taking the visual information back to the brain, we again found that all the information that the giraffe was looking at was coming from below. So the giraffe's really looking down on everything, which kind of makes sense because they're very tall, right? You don't need to be looking out to the side. When you're that high, you need to be looking down to see what's around you. The next slide, please. The other thing we found in the giraffe brain was with the erexinergic system. They have this novel parvocellular cluster of erexinergic neurons. So you see the main cluster, optic tract cluster, and zona incerta cluster. Those three are typical across mammals. But this small cluster of small cells in the medial hypothalamus is very unusual. We first had it in the giraffe. We've now seen it in all the artiodactyls we've studied, in cetaceans as well. We've seen it in all the equids and all the perissodactyls that are related to them. And also we've seen it in lions and cheetahs as well. It's an unusual cluster of neurons, and it seems to be related to keeping the animal awake so that it can get enough food to eat. The elephant also has this cluster as well. We first described it in the giraffe. So all those animals that eat a massive amount of low quality diet need to stay awake longer to get enough food into their system to survive. And this seems to be the mechanism that's allowing these animals to stay awake longer. Now, the next slide, please. So let me get back to the elephant. I want to show you a few things that we've done on the elephant because I think they're kind of interesting and they're fun to look at. So this is a hemisectioned elephant brain. You can see that it's got a very typical appearance of a mammal brain. All the normal structures are there. Can I have the next slide, please? But there were unusual aspects of the arousal systems in the brain of the elephant. We have the parvocellular excinergic neurons, and in the locus coeruleus, where the normal locus coeruleus is, that's at A6D, there was another cluster of neurons called A6N there, and that doesn't occur in any other mammals. So we thought there's two really unusual things about the arousal systems in the elephants. How do the elephants sleep, or how much do elephants sleep? Now go on to the next slide, please. So we used a thing called actigraphy. We darted an elephant, it was immobilized. In the field, we did surgery and implanted a wristwatch-sized device in there. You guys know of Fitbits, right? You know what Fitbits are. It's kind of like the scientific version of a Fitbit. We implanted that in the trunk of the elephants, then let them go for 6 weeks. And these were wild elephants living in Botswana in a national park. Have the next slide, please. What we found was that elephants were only sleeping for 2 hours a day. That's all they need to sleep. The rest of the time, their trunks were active, which means that they were likely pushing food down their throats. The time of day that they slept was around 3 or between 2 and 5 a.m. They would go to sleep well after dark, and they would wake up generally well before sunrise. There were some nights when they didn't sleep at all. If you look at the two graphs on top, if you look at approximately the 17th of April and the 8th of May, and then the 17th of April and the 5th of May on the bottom one, and sorry, and on the top one also the 14th of May, there were nights when these elephants didn't sleep at all. And what was really interesting on these nights is the animals would move about 30 kilometers. So these were matriarchs, they were leaders of their herds, and those nights they didn't sleep, they'd move about 30 kilometers. One of the interesting things we found is on the first night where both the matriarchs didn't sleep on the 17th of April, they were located close together, only about 500 meters apart. Suddenly at the same time, they both took off in different directions, one for 40 kilometers and one for 30 kilometers. It was really quite a spectacular finding because we had GPS collars on the animals. And it seems that in this area of Botswana, the elephants are hunted by lions. So it's possible that a predation attempt scared the elephants and they took off and they ran off going in all different directions. So it was really quite interesting. Go to the next slide, please. The other thing that we found is they only lay down to sleep every third or fourth day. So when we go into REM sleep or dreaming sleep, we lose all body muscle tone. So most of the time when the elephants are sleeping, they're standing up. Okay, not like that guy over there. Not you, the one next to you by the flowers. Yeah, he's sleeping there. That's okay. Yeah. It's all right. You can lie down and sleep. So when we're in REM sleep, we lose muscle tone and we can't stand. And the elephants only lay down to sleep every third or fourth day, meaning they probably only had dreams every third or fourth day, which is an unusual thing. The sleep onset and offset was related to the environment. So when elephants went to sleep and when they woke up was related to the combination of temperature and humidity, had nothing to do with light. And that was really interesting. It was very, very strong relationships between this combination of temperature and humidity, which we call the real feel of the environment or a measure of apparent temperature, and when elephants went to sleep and when they woke up. And they can also go up to 48 hours without sleep, and they don't need to catch up on that sleep. They can just miss that sleep and they're fine. The next slide, please. We looked at the olfactory bulb of the elephant. Elephants are known to have the broadest sense of smell of all mammals. There's about 2,000 active olfactory receptor genes. Their olfactory bulb has a honeycomb glomerular layer. So normally this glomerular layer in the olfactory bulb is a monolayer, but it was honeycombed. It had several different layers there, which is quite unusual. You can see that in the one labeled D there. You can see the glomeruli stacked up on top of each other. The other thing we found is that they don't have an accessory olfactory bulb. The accessory olfactory bulb processes information about pheromones. Now, elephants are known to really behaviorally respond to pheromones, and they do that very nicely. What we've figured out is that instead of actually smelling pheromones, the elephants experience pheromones as a sense of touch. They're very unusual. They actually feel them rather than smell the pheromone. And generally for smell, we do actually have a tactile aspect to smell, right? When we're describing smells, we talk about a sharp smell, a dull smell, those kind of things. So there is a tactile aspect to the sense of smell. And in the elephant, for the pheromones, they've gone to the point where all they're doing is actually feeling or sensing that tactile aspect of the odors that they're perceiving for the pheromones. So they don't have an accessory olfactory bulb, but they do have a vomeronasal organ, and it's a trigeminal nerve that's taking that information back to the brain. Have the next slide, please. We looked at the elephant brain in numbers. We turned the elephant brain into a soup, and we counted the number of nuclei in this. They have 420 billion neurons in their brain. It's quite a lot. Humans only have about 80 billion neurons. So the elephants have more neurons in their brains than what we have. The cerebellum has about 205 billion neurons. That's where we're supposed humans that have 69 billion neurons. The cerebral cortex had 5.1 billion neurons. But in humans, we have 16 billion neurons in our cerebral cortex. So there's an interesting difference there. So elephants have about the same number of cortical neurons as a gorilla does. If you were to sort of very superficially think the numbers of neurons equals intelligence, then you could say an elephant has about the same smarts as a gorilla. All right? Definitely significantly less than humans. Have the next slide, please. When we look at the cerebral cortex, elephants in all parts of the cerebral cortex lack a layer 4. One of the unusual features of their brain, there's no layer 4 there. And we've looked at that closely. And even though the cortex is 2.5 to 3 millimeters thick, which is similar to humans, you can see the neuronal density is lower, but there is a normal range of neuronal morphologies. Next slide, please. We did Golgi staining, and you can see some of the different types of neurons that we revealed from, we call them crab-like neurons, the one in the upper left, a typical pyramidal cell in the upper right. In the lower left is a very large pyramidal neuron, and in the lower right here is a typical glial cell. These are taken at different scales just to show the features. Now, the next slide, please. When we did three-dimensional reconstructions of them, we found that these larger neurons in the frontal cortex of the elephant were similar in size and complexity with neurons in the frontal cortex of humans, which is really kind of interesting. They've got some parts, even though they have lower neuronal numbers, they have some neurons that are as complex as what we see in humans. May I have the next slide? Now, we all know an elephant never forgets, right? That's one of the stories. So what about the size of the hippocampus? They must have a huge hippocampus, yeah? In fact, they have a normal sized hippocampus, one that you would expect for the size of their brain. This is all the data showing it with the statistical confidence and prediction intervals, and the African elephant falls right into that part. So can I have the next slide, please? And when we look at the anatomy of the elephant brain, so this is MRI imaging in a horizontal plane, you can see the hippocampus located in the typical position in the temporal horn of the lateral ventricle. We have typical adult hippocampal neurogenesis. Can I have the next slide, please? But when we look at the dentate gyrus, it's really unusual compared to other animals. Instead of having a 3-layered organization, they have almost a 5-layered organization in the dentate gyrus. And you can see the staining with calretin at the bottom. You can see that extra layering of the dentate gyrus. So there's like a splitting or a doubling of associative and supramammillary pathways in elephants. So does this mean that elephants have a special memory? I don't think they have a memory that's any more than what's needed for a long-lived terrestrial mammal. Okay, probably pretty much like ours. They have a memory capacity and clarity that is probably similar to humans. We do forget a lot of things. Can I have the next slide, please? It's most pragmatic to conclude that the clarity and utilization and recall and all those things of memories in elephants is very similar to what we have in humans. The hippocampal circuitry might be a little more complex, but we don't know what effect that would have on memory. We haven't been able to look into that. But like all other animals, we need to forget a lot of things, okay? We don't need to know what someone had for breakfast 3 weeks ago, right? You can look that up on Instagram. But we don't need to remember those things. There's certain facts we need to remember, and there's certain things we need to forget. Have the next slide, please. The other thing we found was elephants have a very large cerebellum. The next slide. In fact, when you compare it to all the mammals, the elephants have the largest relative cerebellum size of all species. So they have the largest absolute cerebellum in terms of size and the largest relative size. And we see you compared this to lots of other species. Now, the next slide, please. Oh, sorry, go back one. I forgot something. The cerebellum is related to motor movement. The elephants have an incredibly complex trunk that they can move in all sorts of directions. So the large cerebellum is likely related to motor control of the trunk. Also, the neurons of the cerebellum, I don't have a picture of it. The neurons within the cerebellar cortex of the elephant are far more complex than what you see in other species. So they have an incredibly large cerebellum. There's a lot of neurons, and these neurons are very complex in their organization. And this allows them to control a trunk that has about 100,000 muscle fascicles in it and no bones to move things around. All right, next slide. And getting towards the end. So the other thing that elephants do is they communicate through infrasound. They're these low frequency calls that are below our level of hearing, and they use these to communicate. The elephant produces the infrasound. It travels through the air, and it's loud enough to travel through the ground like a seismic wave. The seismic wave, which they sense through their feet or through their trunk if it's touching the ground, travels at about 5,000 meters per second, very fast. Whereas the aerial wave travels at about 350 meters per second, much slower than what the seismic wave does. I go on to the next slide. So if we look at the seismic reception in the brain of the elephant, it's known that there are a high density of Pacinian corpuscles in the feet of the elephant. They're like, they detect the incoming seismic waves. You have large dorsal column nuclei, a significant spinotectal tract, the ventral posterior inferior nucleus of the thalamus, which passes on these Pacinian inputs. We also have the pulvinar nucleus that probably interacts with the somatosensory cortex and association cortex associated with it. In the next slide, please. Auditory reception comes through the cochlear nerve into the cochlear nucleus that passes on to an enlarged superior olivary nucleus. The superior olivary nucleus is related to interaural intensity differences of sound. Is the sound louder on this side or is it louder on that side? The inferior colliculus creates a map of auditory space, and in the superior colliculus, the auditory signal and the seismic signal can be integrated together. This then is passed on to an unusual nucleus in the thalamus called the transverse infrageniculate nucleus, and it seems to be involved in processing low frequency sound waves because this nucleus is not found in other animals. Then we have the pulvinar and associative cortex. We have the auditory cortex and then the associative cortex. So we can see the somatosensory and the auditory signals coming together. The next slide, please. This is a picture of the transverse intergeniculate nucleus. You can see it stained up nicely here on the bottom right. It has a bilaminar appearance and it's only found in elephants. The next slide. The other thing for producing the vocalizations, the elephants need a specialized nucleus. And it's not so easy to see here because the cell density in the brainstem of the elephant is quite low. But either side of the midline, can you see a big round nucleus almost going from top to bottom there? It's called nucleus ellipticus. And this appears to be a motor pattern generator for generating vocalizations. I have the next slide. So producing the infrasound, we likely have anterior cingulate cortex initiating the sound. So humans are a little different to other animals. We have a neocortical aspect to creating sound. In all other animals, it's the cingulate cortex that starts the sound. So you know when you get out of bed in the middle of the night and you stub your toe, right? You don't start saying words, you first yell out, like, ah, and that kind of thing. So this is what cingulate cortex is doing. It projects to a range of different structures in the forebrain, then down to the nucleus ellipticus and onto nucleus ambiguous, which is the phonatory premotor neurons. So that's then creating it's giving the neural impulses to the parts of the body that create the sound. Over the next slide, please. So how do they produce this sound? The idea we have, and we're trying to show this, is that it works as a Helmholtz resonance chamber. Does anyone know what a Helmholtz resonance chamber is? You know when you have a bottle and you blow over the top and go, right? You know that. That's a Helmholtz resonance chamber. So what we're thinking is the elephant can use this huge frontal sinus in order to use it as a Helmholtz resonance chamber to create the infrasound that it uses for communication. It can do this through a connection from the frontonasal duct. The frontonasal duct comes down from the front of the sinus and empties into the larynx just in front of the vocal cords. When the elephant exhales, if they open this frontonasal duct, the front of the resonance chamber and produce the infrasound. Now the next slide, please. So for me, even though I've been in Africa for 20 years, it's been a never-ending source of adventure. I've driven from Johannesburg to the Congo in my old car. That's kind of what it looked like after we arrived in the Congo. I've been in caves in Kenya catching bats, off on the beaches catching bats in different places. Been lucky enough to be in sand dunes in the Sahara and had a huge number of adventures, and I've really appreciated it. And then the last slide, please. And I want to thank the mentors that I've had during the time. We've talked a lot about mentors. None of these guys were formal mentors. They were just people that I worked with. Ralph Leon Hughes was the first to take me on as an honors student in Australia. when not many other people wanted to have anything to do with me. My PhD was done with Jack Pettigrew, who was a fellow of the Royal Society, but the way he behaved, you would never have thought he was such a highly regarded scientist. Ted Jones, or E.G. Jones, who's famous for the book, The Thalamus. I worked with him as a postdoc in the US, then with Jerry Siegel on sleep at UCLA, and Shel Fuchser from the Karolinska Institute. Shel Fuchser is famous for describing the dopaminergic, well, the catecholaminergic system of the brain. He was the first person to do that. And I also thank all my current and past postdoc students and collaborators. The one from Nigeria that I've worked the most with is Imam here. So he can tell you lots of stories about how wonderful a guy I am. And my funding has primarily come from the National Research Foundation of South Africa. Thank you.
Okay, so thank you. We'll take a few questions, just a couple, I guess, so that we can, so let's see the hands so that we can number them. Do you mind? Okay, do we have another microphone for him to take? Okay, so we'll take you first and the other person.
Okay, thank you, Professor Manga, for your presentation. My first question is about the actigraph. You said it's used for measuring sleep. So do you have a specific actigraph for the elephants, or are they the ones normal humans wear? And how accurate are they in measuring sleep? Then I can see that you have worked with many animals. So do you ever come across, do you ever encounter challenges when it comes to animal testing regulations before working with these animals?
All right, so the ActiWatches we used in the elephants are Philips ActiWatches. They're a scientific version of the Fitbit that was made for sleep studies in humans. And we've just adapted them and used them in the elephant. They're very accurate. They have, they basically have a piezo accelerometer device in them, and you get a sum of the number of acceleration events that the instrument has undergone per minute. So that's how they read out from it. But you can make it down to, the sum of the acceleration events per second if you wanted to. But in terms of biology, per minute makes more sense, because you can't have an animal going from wake to sleep over a few seconds, Regulations, yeah, I do a lot of paperwork, a hell of a lot of paperwork with ethics and with other things. A lot of the animals that we collect are animals that are being killed for population management reasons. So it's been decided by others to put them down. And I formed a network in South Africa that when this is happening, they call me and allow me to come and get the specimens to harvest the specimens from the animals that are being put down. So the elephants, for example, we got them in Zimbabwe. There were 8 elephants being put down on a farm because they had too many male elephants. So we managed to get brains from 3 of those that were being put down. So we don't normally go out to kill the animals. We harvest the tissue from when animals are being killed. Okay.
Thank you, sir, for the wonderful presentation. I have a question from your presentation yesterday and one from today. So, presentation yesterday very interesting because in the field of neuroscience, where we are used to neurological disorders and neurodegenerative disorders, so seeing a different approach to studying neuroscience was quite interesting. So, my question is, as a body researcher, what areas of neuroscience can you suggest for body neurosciences that will address some of the pressing issues in our locality, like climate change, as you mentioned, and let's say malaria, the major issues that we are facing in Africa and Nigeria generally? Then my second question is, I'm just curious about how the elephants were sacrificed. If they were anesthetized, how many liters of anesthetic was used to do that? Thank you, sir.
Okay, so to be honest, I don't really care about human diseases and problems. Climate change is one thing that not only humans are going to suffer from, it's going to be all life forms on the planet that suffer from climate change. Right? I've always taken the approach that there's so many people working, trying to solve human diseases and human problems that I don't need to do that. There's a lot of amazing researchers doing a lot of work on that. So I never put a lot of thought into those kind of things. However, some of the stuff we've done is actually leading to very interesting changes in the way we look at human sleep. And we have some inventions that we've patented that we think in a few years will be changing the way humans sleep as well for the better. So from the comparative work can come major advances for humans. We don't need to work solely on the rat and mouse model and molecular biology to find out something about humans. And I think I said yesterday that paradigm actually hasn't led to any cures of diseases or anything in humans either. So I could turn around and say, well, why are you working on the rat in order to understand humans? You understand what I mean there? Okay, then the second part of your question is how do we put the elephants down? So the time we got the elephants was 2008. And in Zimbabwe was at a huge low there. There was a hyperinflation, about 100,000% inflation was happening in Zimbabwe. The stores were empty. Everyone was very broke. The people on the farm were getting paid. Their paycheck was in the trillions of dollars. That was how low the value of the Zimbabwe dollar had fallen. So the villagers in the area were also quite hungry. So the elephants that we put down, we used a drug called scoline, which is an inhibitor, acetylcholine inhibitor. So it actually stops the animals from moving, and then we shot them through the heart. The reason we use scoline to do this is that the meat could then be consumed by people. So the 3 elephants that were killed and we did I got the brains from, they provided about 100,000 meals to the villagers in the area in Zimbabwe. So it's kind of it's a weird mixture. You understand what I mean? Is it ethical to put elephants down, or is it ethical to not feed humans and that side of things? So we tried to make a balance where when we immobilize the animal, the elephants with scoline, as soon as they stopped moving, we put 4 bullets through their hearts, so they died very, very quickly. Okay, so we didn't use pentobarbital or anything like that, and using scoline allowed for the meat to be consumed by people.
Yeah, thank you, Prof, for this lecture. I'm just a bit curious because from your presentation, you've had quite a lot of years of experience. So I'm just curious, like being a researcher that is widely traveled and lots of experience, how have we been able to cope as from the family front, as a father, as a husband? How are you able to, get all this together and still, keep your sanity and still be okay?
Well, you see the size of the bags under my eyes? No, it's not that bad. You can make the balance. You just have to work. When you're at work, when I'm at work, I work. When I'm at home, I spend time with my family. And so I don't try to mix the two. Normally in South Africa, when I finish work, that's it. I don't look at emails till the following morning. And I only open emails when I arrive at work. I don't own a cell phone, so I'm not on a WhatsApp group where people are WhatsApping me at midnight or anything like that. They just have to wait. Nothing is that important that it can't wait till the next day when it comes to work. So I make that very strong disconnect between work and home. That's how I manage. And when I travel, I'm lucky that my wife and the nanny will look after the kids nicely. All right.
All right. So, so thank you, Professor Paul Minja. Please let's appreciate him once more for the excellent talk and presentation. Prof, I'm going to say also that I've worked in your lab before, but you're only recognizing Imam. I'm not jealous. All right. Thank you. Well, I think we'll go straight ahead to our next talk, and we'll be bringing up another eminent scholar. I'm going to be bringing up Professor James Olopade. And I have a few things here to say. Professor James Olopade graduated with the DVM veterinary degree in 1992 and obtained his PhD in 2006 from the University of Ibadan. He carried out his postdoctoral training in brain research at Marine Brain Biology Lab, MBL, in Woods Hole, United States of America, and then went to Penn State University at the Ion Lab of Professor James Clark, and also in the United States of America. And then also went to the University of Würzburg in Germany. He's also a Grass Fellow at MBL, and he has served at various capacities as Sub-Dean Undergraduate, Head of Department, the Dean of the Faculty of Veterinary Medicine, University of Ibadan, President of the Society of Neuroscientists of Africa, SONA, Counselor, International Society of Neurochemistry, ISN, is a member of African Regional Committee of IBRO, and currently the Director of the Veterinary Teaching Hospital, University of Ibadan. Let's applaud that. Professor Olopade is the most cited veterinarian in Africa in the field of brain research, and he has hosted over 5 IBRO ISN schools. He's passionate about African youths being trained to become leaders, and has mentored and still mentoring many young brain scientists across the continent. And I must say that I'm one of those that he has mentored. Sir, you have the floor.
Thank you very much. Professor Olude is the first professor produced in my lab. So it's a privilege. When you teach somebody undergraduate, then when he finishes, he comes to you for masters. And after masters, you want to drive him away. He comes back for PhD. And I appreciate Paul Menjas, who was part of his research stay. And then that student suddenly becomes professor, and you are still young. So it just tells you that you still have more time to put in. I think Mamus, I don't know whether he was complimenting me or teasing me or abusing me. He said he worked on nickel and vanadium, but he wants to talk on nickel, that there are some people who work on vanadium. Next year is interesting for me. It will be 25 years as a lecturer. And I have worked on vanadium neurotoxicity. I have worked on basic anatomy of the skull or head region. I started actually working on anatomy of the head region. I do comparative anatomy in wild rodents. Some of those work that Paul Menja is showing. And currently I also work with viruses. So it's 3-4 areas of research with postgraduate students in all parts. But the encouragement is that research takes doggedness and consistency, particularly in the Nigerian environment. I was talking to someone, I said, a professor's salary now cannot buy a standard antibody. Nice antibody you want to buy, Carbindine, it's 400 euros. Okay? Yesterday it went to 900 naira to $1. So a professor can't even buy an antibody. But yet, we must do research. We must be productive. We must have active laboratories, unless there's no hope for the future generation. They may not, many of them want to run away, or let me say, go away. And I don't blame them. But even before you go away or run away, there must be a place where you'll be trained first and have the basics. And when you want to come back, there must be a place you can come back to and continue to work. So that's why we have to keep on the research. So today I've picked 3 publications that I want to tie together on vanadium research. The first time I started working on metals in the brain was 2003, 2004, early 2000s. And I was using the West African dwarf goat as a sentinel for environmental pollution. So spent a lot of money in those days, level of vanadium, level of copper, level of iron, level of zinc, level of lead, and different parts. No, first and foremost, I took the whole brain and digested it. And I sent it to a journal and I said, this is the level. And the reviewer wrote back, nice effort, but I'm sorry we can't take this paper. You said level of iron in the brain. Is that olfactory bulb or cortex or globus pallidus or nucleus accumbens? Which part are you talking about? Some of us suffered lack of mentorship. So, but the reviewer was a good mentor. So I went back and then olfactory bulb, cerebellum, cerebrum, thalamus, and then got that first paper published on metal concentration, looking about 7 metals. And then went to Johannesburg for the Ibro school in, I think 2005. 2005 was in Joburg. I said Ibro school, SONA. And then I met James Connor. And James Connor said, through email after I went back home, he said, young man, you can't be studying all the metals. You're not going to go anywhere. He was studying iron and he had been studying iron. He's still studying iron. He will be 70 next year. And said, pick one metal and dig deep on that metal. And then give us information on how that metal works. So I took his advice, but he said something. Pick a metal of epidemiological importance to your environment. So I got thinking, okay, what is in our environment? We survive in Nigeria by crude oil. So which metals contaminate crude oil? So there wasn't too much internet in those days. Things have changed now. You want to do a Google search, 30 minutes. I need to leave the office and drive for about 20 minutes to the only cafe in UI area. It was in Bodija that has internet, and then you pay for an hour. So I said, well, instead of doing that, I drove to petroleum engineering department and requested to meet somebody who was into environmental pollution and asked him the metals in Nigerian crude oil. Thank God I met a brilliant young man. So he gave me about 5-6 metals, but he mentioned vanadium. And somehow vanadium just rang a bell. So I now drove to the cyber cafe and booked for 1 hour and looked on vanadium and saw that every publication on vanadium and the brain were in areas where they had crude oil pollution. Mexico, Libya, Saudi Arabia. That's where the articles were from. And that's how the story of vanadium started. And like I said, I'm still into many other things, but you don't leave your first love. So I'm still into vanadium. So today I'll talk about vanadium neurotoxicity, long-term exposure, and generational issues. Slide. I think once it's just see this, it just means so I don't. Okay. While we are told that the brain is protected by the blood-brain barrier, in reality, the brain is under siege. By the time I did that, my first analysis of metals in the brain of West African dwarf goats, it was obvious that some metals that the literature reference point is nil, insignificant, they were in high quantities in the goat. So the brain in polluted environment is under siege. From heavy metals, the environment itself, the atmosphere, you've seen forest fires nowadays across the world, volcanic ash, wildfires, all that particulate matters find their way to the brain. Next. Oh, okay. So let's move from a general story to heavy metals. Generally, when heavy metals get into the brain, they bioaccumulate. That's dangerous because a metal can react with available water, cellular water, generate reactive oxygen species, hydrogen peroxide, superoxides, but the metal remains and combines again with another water molecule. So you have constant production of reactive oxygen species with constant, even though there's a constant level of metals. So bioaccumulation occurs, oxidative stress, and then you have neurotoxicity. Next slide. Now that's a big problem because it's not that we don't have metals in the brain. Some metals are essential for the brain to function. Okay, you get to the synapse, you will see copper, you will see zinc, performing physiological functions. So on the left is synapse, in physiological conditions. But when you have metal toxicity, mercury, lead, vanadium, metals that the brain, who are foreigners to the brain, you see what you have on the right. You have a shrinkage of the synapse. You have a bigger synaptic cleft. You have destruction of physiological metal receptors. You have a degradation of postsynaptic density. You have destruction of the cytoskeletons of the synapse. So those are the problems you have when you have a metal toxicity, and then the nervous system cannot function properly. So be it anxiety, be it depression, you have problems like that occurring because you do not have a properly functional synapse. Next slide. Okay, so like I said, the brain is under siege, and one of the most popular ways or physiological ways is that particulate matter is taken by inhalation. Okay, through the nasal path, cribriform plate ethmoid, and then to the olfactory bulb. And then there is like an organic distribution sometimes from the olfactory bulb to different aspects of the brain. Okay, when you have metal polluted soil, metal polluted water, and people drink such water, it gets to the GIT, from the GIT to the capillary system, and then it gets to the brain. Particularly metals, I'll talk about vanadium, that can actually disrupt the blood-brain barrier. So, or from when you are breathing, while some can go to the olfactory bulb, some go straight into the lungs. And from the lungs, at the alveolar system, they get into blood. And then from blood, they find their way to the brain. So the brain is under siege once there's excess metals in the environment. Okay, so these are the three papers I want to, or these are pictorial depictions of three papers I want to talk about today. That when there is exposure to vanadium, like you have constantly in the Niger Delta, okay, crude oil burning is taking place, gas flaring, forest fires. These are avenues of vanadium release. Three things are apparent. One, it's a lifestyle, it's occupational, and it's chronic. So some people can be doing jobs like this for 20 years. So they've been exposed. So there is chronicity as an issue. And like Professor Manga was saying, The reality in the field is that people are exposed for 20 years. But we do experiment exposing the rat for 1 week and write our paper and make inferences on the consequences of vanadium exposure. So you do not have a model for what is happening in real life with people. Now, apart from being chronic, is that, like you know, half of the population are females. And females do get pregnant. Inhalation, bloodstream, it gets to the next generation. So you have a generation exposed before they are born. So there are generational issues involved. Remember, that is the early formative days of the brain. Remember that there's vulnerability, genes are being developed, they're expressing themselves. And then tied to generational issues are mothers who go into the environment to work. And the vanadium enters through inhalation, bloodstream. You now have vanadium in the blood. And vanadium from the blood is well filtrated into milk, being taken by the protein lactoferrin, binds to vanadium. So vanadium is in the milk. And then the infant is exposed through lactation. So if you have a caring mother that says the environment is polluted, the child will not follow me, the child should stay at home and be safe. But she goes out into the environment and inhales vanadium, is going to the child via lactation. And lactoferrin binding is very strong and very efficient way of transmitting vanadium. Next slide. Okay, so let me now major on vanadium. To the chemists, that's all the chemistry involved in vanadium, atomic number 23. What is not here is that it's a transition metal, so it shifts in oxidative states from +2 to +5, and then that becomes a basis why it generates a lot of oxidative stress, reactive oxygen species rather. And this is my first pure vanadium paper, 2011. Now not metals but vanadium. And vanadium will always lead to hydrogen peroxide. Dependent or independent of ion, leads to reactive oxygen species, oxidative stress that can lead to DNA damage. And for the infant that is developing, that has no myelin yet, vanadium will destroy the oligodendrocytes. And then that infant will not have proper myelination. Next. Okay, so vanadium is everywhere. The statement below is that's 2017, it's probably more true today. The human emission of vanadium to the atmosphere is almost 2 to 1 to what is from the atmosphere itself. So humans are contributing more to vanadium in the atmosphere than what is coming from natural sources. And natural sources exist. Volcano releases a lot of vanadium. Forest fires, be it natural or human made, releases a lot of vanadium. Okay, now human steel factories, because vanadium is very important for iron ores, coal mining, petroleum ash, crude oil extraction like in Nigeria, all this, and there's actually also atmospheric vanadium. So, and that rains down, and these are the sources of vanadium in the atmosphere. Finally, also to groundwater, so it draws home. Next. So, like I said, vanadium leads to hypomyelination. So now I'm coming home now to strictly what happens in the brain to vanadium. I think I need a 10 seconds break so that those sleeping can be awake. Hello? If you are listening to me, please, can I see your left hand up? If you are here, left hand, if you are listening to me. Okay, so it's obvious to know the people not in semi-sleep, but in deep sleep. Okay? I don't think I can do anything about deep sleep, but I think I've helped semi-sleep out. Okay, so this is the first, like I said, pure vanadium paper that we had. And this, there are two of them, but this one, vanadium leads to hypomyelination. Next. So, and vanadium leads to demyelination. Okay, so I have this paper from Aziz and Igado that these are beautiful papers because this was the first time we used black gold to stain for myelin. So hitherto we had used MBP, but black gold is beautiful. Black gold histochemistry actually shows myelin better, and then you can appreciate demyelination. Next slide. Okay, sorry, it's a bit crowded, but I didn't want to separate them. I want to use this slide to make a statement because these are three, I still consider them standard papers that have come out from my lab, but came from one student from the PhD work. And it was easy to produce 3 papers from that work because this work was based on epidemiological realities. For the first time, we systematically, not daily, but systematically expose rats to vanadium for virtually their lifetime. From postnatal day 1, they were exposed through their mom, the dams, that we made sure the dams had vanadium. So the first exposure for 14 days was through milk. So after 14 days, we now started exposing the animal, and we exposed them for 18 months. So there's a lot to study. We are still going to repeat that experiment because it's brought out a lot of gold from such a work. So we studied the memory pattern of the animal over time from 3 months, 6 months, 9 months, 12 months, every 3 months. So produced a Morris water maze pattern of memory in vanadium exposure. And you could compare it with the control. Then we had a group that was exposed only for 3 months. And then we took the vanadium away. And then they lived till 18 months. Withdrawn. So we could compare, if you actually withdraw from vanadium, how long does it take to recover? So, and the animals didn't come back to control levels until about 9 to 12 months. Well, actually, until they were old, that they could now compare to control. So that was the first paper. The second paper looked at, as vanadium was entering the animal, we were able to map its location in the brain. So at 3 months, where was vanadium in the brain? At 6 months, and the demographic was changing. And just like when you have some diseases that affect the brain, toxoplasma, you see them go to a particular section of the brain. You wonder that what's the basis for a predilection. Same thing, there were some areas of the brain after 12 months, 15 months exposure that hardly had vanadium. Whereas some areas accumulated. It's an organic, inorganic matter, but some areas of the brain had predilection. So let's be careful. You just open up an animal, open up the brain, and just cut one part and say brain. No. There are predilections sometimes to where they are present. And that's that paper in Frontiers. And then the last paper looked at what we worked with biochemists and looked at the antioxidant profiles over time in chronicity, and then what happened, how did they recover with withdrawal. Next slide. Okay, so I'm about to move into the 3 papers I want to discuss, 3 specific papers, but this is our latest review on vanadium. This was a book, it's in a book, it's a chapter 10. This was a book edited by Ashna and Costa. I think it's 2011. Yeah, 2021. It's 2021. So this This is our most recent review on vanadium. We'll probably not try another review again until maybe 2026. After 5 years, at least there are new developments and go. So this is probably our third review on vanadium in the brain, vanadium neurotoxicity. Next slide. Oh, I've talked about this. Next. Okay, so I brought this again just to talk about the 3 papers. The first one is the first experiment we did to see whether vanadium could pass through milk to affect the infant. So the infant is not exposed. It's only the mother we gave vanadium to. And then we looked at the neurotoxic profile of the dams, of the pups. And then the second paper I will talk about is on generational effects. So the mother is exposed to vanadium. The baby is born and remains in that environment. That baby grows up and produces another baby, and that baby will now took it out from the environment and took it to somewhere where there's no vanadium. And we now started monitoring the neurotoxic profile of vanadium after it had left the environment, even though 2 generations were exposed. And then the third paper, which is chronic level. What that changes in chronic level. Okay, look to one. Next. Okay. Vertical administration of vanadium through lactation. So this was a Fatola's master's work. So how key on vanadium expose the dam. So the dam was taking vanadium, and then the pups were born. We continue to give the dams vanadium. Let me give a little background. 2008 to 2009, I went to do my postdoc in Penn State. And Penn State taught me immunohistochemistry. That's the first time I was ever doing it. I only heard about it till then. So they taught me, and I was excited. I was excited and disturbed for 2. It looks contradictory. I was excited, but I was disturbed. I was excited because when we did the immuno, it worked. The pictures were beautiful. I was disturbed because I didn't see what I did to be a big deal. It wasn't rocket science. So I queried why we don't do this in my country. We spend billions of naira. It doesn't take much to get this done. No, our fathers have shortchanged us. I said that to myself. I can't forget the day I saw the first picture. So our fathers have shortchanged us. So I came home in 2009, wanting to set up an immuno apparatus. But without saying too much story, the environment was not ready for the setup. Let me put it as little as that. So I went back to Germany, or I went back abroad and continued doing immunohistochemistry, this time Western blot and PCR, and came back home with some money from ISN. We really appreciate ISN. And then I bought everything needed for the immunohistochemistry. And this time around, even though the environment was still resistant, I wasn't a small boy anymore. So I fought back and we set it up. So we now decide let us do immuno for the first time in this place. So I had Fatola master student. Fatola is a big man now. He's finished his PhD. So we set up the immuno. Next slide. Okay, I'll just rush through this. So we had known in the lab that for the pup's brain development, age 7 to 14 were crucial periods. Okay, day 1 to day 6, the brain doesn't really increase that much. But 7 to 14 were crucial periods. And so both body weight, control to vanadium, control is in black, vanadium is in red. A group that received vitamin E was in the middle, the rescued. And then this was the brain weight that actually vanadium led to reduced brain weight. And it's logical because vanadium demyelinates. So once we were giving it from the milk, we did not allow oligodendrocytes to really bring out myelin. So the brains couldn't weigh much. Next slide. And then we now did the immuno. When I look at this, I laugh now. First, definitely, I don't know whether I have other pictures. We do immuno far better, but I cherish the first one I did. It had its imperfections and co. But we're able to get it done, and then we now continue to build on it. You must start from somewhere. If you are not ready to start, you won't do. So I got a picture, at least you saw the big soma, big dendrites of the astrocyte, control, vanadium and vitamin E. Next slide. Okay, so vanadium in milk is transported in a bowel complex with lactoferrin. Lactoferrin is very efficient. It easily passes through blood-brain barrier in an active manner. It binds to vanadium. It's the bond between lactoferrin and iron and lactoferrin and vanadium are very tight, even though they can be reversed. And Ebel and Sagioni actually found out that when an animal took milk once from the mother, days after, we still saw vanadium all across the tissue. So showing that it was a very soluble complex that had easy access. And that's just nature. The milk from the mother has easy access to all parts of the body for growth. And that's why once the mother is exposed, the baby is in trouble. There's no protection. Next slide. Okay, so that's vanadium through milk. This in vivo vanadium induced neurotoxicity and withdrawal in 3 consecutive generations. So you have the dam exposed to vanadium. They gave birth to a filial generation. That generation was allowed to grow, gave birth to another generation. But the 3rd generation, we took away from vanadium and tried to look at the brain of that 3rd generation when they became 21 days without any exposure to vanadium. This Lee Joka, he was the master student in the lab. Myself and Dr. Egado supervised him. So next slide. So this describes the pattern of exposure of vanadium. Next slide. Okay, I want to talk more about this, but what is obvious is that the generation 2 that still had exposure had the worst outcome in body weight development. Generation 3 got better, and that's what you see at the graph below on the right. But there were disparities in the earlier generations. Let's move. It's the brain issue. Okay, so behavioral tests. So we tested generation 1, control versus vanadium. Generation 2, control versus vanadium. Generation 3 that was not exposed. So control versus vanadium not exposed. Let's put it like that. And so vanadium is in red, while control is in black. Then generation 1 is on the left, generation 2 is in the middle, generation 3 is at the third side. But what you will appreciate most of the time is that generation 2 had the worst differentials between control and vanadium in the barrage of behavioral tests. Now, the summary that you see in generation was not exposed at all to vanadium. They compared with control in many locomotor tests, but they still did badly in cognitive tests. Cognitive, memory, intelligence, they were still worse off, even though they were not exposed to vanadium. So when people grow up in an environment, generation to generation, even when you take them out of there are still consequences to their cognitive behaviors, even though they can recover as per locomotion, okay, over time. And that was the major thing we saw. And which the immunohistochemistry also gave some credence to. Next slide. Okay, so one major consequence we've seen from vanadium exposure in the lab is that, and it's documented in some other metals, is that vanadium leads to multiple layering of the Purkinje cell layer of the cerebellum. It's a toxic reaction. It's the body trying to react. So if top left, that's our control. I think those are our controls with generation 1, filial generation 1, and you could see already multiple layering in B compared to A of the Purkinje. Here, 2, multi-layering was evident, but the cells were not even alive again. Most of the cells became hypnotic. Generation 3 is recovering from the multi-layer, but you can still see almost a bilayer of Purkinje cells. Okay, so we see recovery. I mean, and we hardly see apoptotic Purkinje cells in generation 3 compared to control. Next slide. Okay, so this is GFAP stain in the CA1 region. So this was done in the lab. So compare this to the initial immuno we did in the lab. We got better in our pictures. We also got better in microscopy. So sometimes not just if you don't have good microscopy at your disposal, you will also not. So generation 1, that's control to vanadium. And you can see in generation 2, things really got pretty worse. And generation 3, even though it was not exposed to vanadium, this is just the offspring, but you could still see moderate astrocytosis in the filial generation that had no exposure to vanadium at all. Next slide. Dentate gyrus, the same thing. Generation 1, by generation 2, you had more numbers, bigger stomas, more dendrites. So really reactive. And then generation 3, at dentate gyrus, you had far more recovery compared to control, even though it's still alarming to have this because it wasn't exposed to vanadium at all. Next slide. Okay, this is MBP, myelin, and then you see generation 1, that's the corpus callosum, but you could see the dots in the area of the hippocampus. That's why memory is always a problem with vanadium. You could hardly pick myelin stains in the myelinated axons of the hippocampus. The myelin there is gone. So even though corpus callosum was affected, by the time you get to generation 3, the corpus callosum is relatively good. It's not as good as control, but you did not get that recovery in the, you see the red arrow at the bottom right. You don't get that recovery at all in the hippocampus. So why corpus callosum is seen, and that's why we talk sometimes about predeletion. Why the demyelination of corpus callosum is seen to be recovering, you hardly have any recovery in the myelinated axons of the hippocampus. And that's why we could see in the behavioral test that they kept doing bad in cognitive tests, but they could do better in locomotor tests. And that's what we got from the 3rd generation. Okay, next slide. I think I should be rounding up. Yes. By generation 3, you could see generation 2, clear difference. This is now the cerebellum. This is now the cerebellum, the abovitile. But you can see at generation 3, they're almost the same. The recovery was well marked there. You couldn't get this recovery in the hippocampus. And this also contributes to locomotor becoming better, while we're still having problems with cognitive apparatus. Next slide. I think I should. Okay, so I'm zooming in again on this paper on brain metal distribution. So I have talked about lactation. I have talked about generational. Now I want to talk about chronic. This was the paper I was talking about, where we were exposed for 18 months. Control, exposure, and withdrawal. Next slide. So that was probably in frontiers in neuroanatomy. This is GFAP. I will talk, but I want you to look very well. That's 6 months control, 12 months, 18 months. Now, in the middle is the vanadium exposed group. you could see the astrogliosis at 6 months marked. It is far more marked at 12 months. You can see the quantification below, 12 months. But by the time we got to 18 months, the difference between control and vanadium was not as marked as when it was in 12 months. The astrocyte seems to be reorganizing itself and recovering with time. If we did not do a time lapse issue and we just did 18 months straight, we would have been surprised. The astrocyte seems to be recovering, and the last group is withdrawal. So, and like I said, below is the quantification. So astrocytosis seems to be recovering, reducing, attenuating as time. It definitely is shot up 3 months, 6 months, 12. But after 12 months, it just started going down. The astrocytes were recovering. The astrocytes needs to perform a lot of physiological functions. And once they are under assault for a while, they actually have intrinsic mechanism to repair themselves back to function. Next slide. But you won't see that when your study is 2 weeks. We never even saw it because we had done 3-month study previously before. We had done, 3 months. First 1 month, then went to 3 months until we said, let's take it the lifespan of the animal and mimic a lifetime exposure. However, this is also astrocyte, but this time I think this is at the corpus callosum. Likewise, just what we saw, still at 6 months compared to control. At 12 months is worse, but the brush borders were reducing. By 18 months, astrocytosis has started to attenuate relative to control. Next slide. So it's the same, it's just from 2. However, this is microglia. Microglia was not attenuating at all. At 6 months compared to control, at 12 months, at 18 months, the microglia became angry with brush borders. The amoebic forms that we were getting before, all the processes were out. And that's the quantification you have at this last slide. Just this microglia alone in vanadium group, 6 months, 12 months, 18 months. Microglia kept getting worse. Next, I think I have one more micro. Yeah, so this is at cerebellum. You see microglia, it gets worse at 12 months. It gets worse at 18 months. So in chronicity, the major phenotype we are seeing was more of microgliosis than astrogliosis. Next slide. Okay, so I think I have 2 or 3 more slides. Talk about future directions. Let me say this with a great sense of responsibility. I'm grateful to God that Paul Menja spoke first. Paul Menja is a senior, a big senior. The future of any academic system lies with professors who keep thinking. It can't lie with the zeal of the younger generation. We thank God for their zeal. But if we do not have people at the upper echelon who, they may not be hands-on like they were 25, 30 years ago, but they keep innovating, asking questions, having more objectives of what can be done. And that only comes through one way. One way, basically, they keep reading. They keep reading. The most important thing in a research environment, in the research industry, is not money. I know you all want money. I've seen where we know departments that money has entered. They didn't buy fluorescent microscope. They changed the rug in the HOD's office and changed AC from manual to split and changed the freezers. Money came in. So when we say we don't have money for research, is money really the problem? It's a problem. But it's not the most important thing in a research industry. The most important thing in a research industry is ideas. And it's when there are lack of ideas, money is misused. So it's a sense of responsibility. And that's why I said, future directions. Yes. 25 years next year, working in the department. I've been a professor for, next year makes 14 years. But I'm still at the job. So I have to keep thinking. I have to keep innovating. What is the next? I'm into anatomy research. I'm into vanadium research. I'm into virus research. Each one has the next level. So that when the postgraduate students come, I don't say, okay, sit down. So what are you interested in? What do you want to do? What are you thinking about? No, we have plans on ground that they are coming. The paths are clear. They are just coming to sail. Younger people, create an environment better than the one they are creating for you. Okay? You are young now. You are complaining. But have a dream that when I get up, I will make it better for those who are coming. So we have future directions. That's animal experiment of 18 months. We still have the blocks. Now, how prone are those animals after 18 months to expressing amyloid? Alpha-synuclein, my MDS, Oga, and tau has constant vanadium exposure over a long time to advanced age predisposes animals to express amyloid. So if we think that metals are risk factors to amyloid, okay, we want to see about vanadium. So Folani is off in 2 weeks to a tauopathy, proteinopathy and tauopathy lab in Göttingen to go and look at those blocks for amyloid expression, tauopathies, alpha-synuclein, whether they are expressed. Next slide. Yeah. For those of us that went to Porto for the ISN, you will know this face. That's the thing about keep pushing the bar. So a restaurant also showing conference and go, you have to keep going to conferences. You see new things happening. You now know possibilities of things you can do. I was at Porto. I met this Nigerian job by Providence. I was conducting oral exam, Viva, from Porto to Funab. You know, this is my Oga. So I said I will send somebody. Say you can't send somebody. Do it yourself by Zoom. So as I was doing the oral exam, this young man was walking at the end of the bench, and he just probably had some Nigerian slangs for my mouth. So I passed by him. I mean, there were probably like 500 people, 600 people at the conference. The black people, either Nigerian or all the blacks were like 15. So when you see a black person, you know you've seen somebody. So when I passed by, I just say hello. He said, "You are Nigerian?" I said, I could hear from. So I said, "Oh, is he Nigerian?" Because his slangs were American. So I saw his badge. Wow. Ukbo Eyo. He said, "Yeah, he's a Nigerian." I said, "Wow. You are born in the US?" He said, "No. I left Nigeria 20 years ago for university education to the US. And now he's an associate professor." And we got talking, just sharing research experience. And I told him about this work. And he has a microglia laboratory and heard about this constant microgliosis over time. When we got that data, the next plan in my mind was to go to a lab where they have pure microglia culture and expose the pure microglia culture to subacute dose of vanadium over time, then harvest them periodically, and then look at the expression of the inflammatory genes, how they change over time. So I now told him about this. He said, no. You don't need cell culture. Since you are having microgliosis, real life, in vivo, we can repeat this experiment. And then this your 3 months, 6 months, we can extract the microglia genes from the brain, and then we can look at their expression. So this is normal microglia, like we're having our controls, 3 months, 6 months, 12 months. These are genes that maintain microglia homeostasis. However, when there is an insult like vanadium, they become disease-associated microglia and express some of these genes, not all, some of these genes. So at 3 months of exposure, how does this picture look like? Then how does it look at 6 months? How does it look at 9 months? Then how does it look at 18 months? That's one. Number two, how do this picture differ from different regions of the brain? Is it the same picture in olfactory bulb? Is it the same picture in the cerebral cortex? Is it the same picture in the visual cortex? Is it the same picture in the prefrontal lobe? So we look at the different regions. And then Ayo has established clearly some genes that do not change in females in microglia, while some change in male when insults come. And that he had shown that the female has more ability for the microglia to be self-sustaining and resilient because there are some genes that remain constant while they are disrupted in the male. So sex difference, regional differences, and time difference cut out for us so that we can increase the knowledge in the field. Next slide. Advice and closing from the youth. I came in today, but I saw this yesterday on the platform. You are the one sending them. I've caught you. Please give her a round of applause. I saw it and I said, we're in trouble. There's a young army interested in what we are doing. Professor Remo Susa, at their stage, when Oga says shut up or sit down, Oga will go home. He will come back. He will say, why are you sitting? He says, sir, you told me to sit down. We will never stand up. But you don't do that to this one, so. They are restless. They want action. And they will tell us to our face, what are you people doing in this place? That's what they're going to tell us. So when we have an army of young people, we can have our job cut out for us. But to the young, I need to advise you, keep up hope. Research is not a quick fix. Research is stamina. Marathon obstacle steeplechase. It's not Yahoo boys. The results don't come quickly. You don't get your respect, reputation, and fame overnight. Ask Paul Menja, ask you stay with it. So if you know it's your passion, have the mentality that you're going to stay with it. Your mate who went into IT will have bought the land. You are still looking for Chrissy Violet. They will have had the third story. You are just getting the antibody. That's the reality. But is it your passion? That's why we stayed. It was our passion. It was not the money. And later the money is coming to meet it. The money will meet the passion. So keep up hope. Nigeria is tough now. Very tough. Very, very tough. But you must keep up hope. Okay? Like my Ethiopian friend told me, he said, in Africa, you can't just be preaching science. You must preach social science. Because if you don't mention the social science, the science will not work. Because there are social problems and things with that. So keep up hope. Be consistent. Be consistent. Come to lab on time. Read the literature. They correct the manuscript. Sometimes you're going to correct it 5 times before we submit. Stay with it. Focus. Yeah. I mean, I teach 200 level. I've been teaching 200 level for 20 something years. The 200 level we taught 15 years ago, and not the 200 level I've been teaching in the last 3 years. They are different human beings. They are different people. There has been a mutation in the genes. Got to be on your toes. As you are teaching, they are doing the goggle, whether you are saying the truth or not. If you are saying rubbish, they will use nose for you that, oh, Baba, old school. So with them, you have to keep on, but please stay focused. Don't allow yourself to be too distracted if you want to do research. And we have to upgrade. We have to upgrade. You can't do a PhD and be staining H&E. We have to upgrade. Particularly to we the seniors. As we become HODs and co, things have to upgrade. Because while we will write reference letters for many to go abroad that will not come back, yet we have to prepare the ground for the few that will stay. So it's a big job being a leader. You'll be the same person to sign for people to go. When they are now there and they need a reference to stay permanently, it is still you that they recall. But for those who will stay, let's make it worthwhile for them. And please, when you go, remember where you came from. Send us antibodies. Write grants together with us. Collaborate. That's how China abides. Remember where you came from. So that we too can, let me tell you this. So this is not, I didn't see it by research. I'm a man of the spirit. Africa will still be great. That's the truth. It will still be great. Many that went, they are coming. They are coming. But we must make sure when they come, there is something to come and work with. Let's put things on ground. And the way to that now is collaboration. I don't know any lab. I don't know any lab that is functioning optimally, doing work publishable internationally, that does not have overseas support or overseas collaboration. Where is the money here? How do we want to buy an antibody now without overseas collaboration? How will your students be exposed without overseas collaboration? So while we are preparing for the promised land, today we need collaboration. So once you are an elder, you are sitting, once you go to conferences, exchange emails, collaborate. Let them see what you are doing. Tell them your limitations. How can they help? PhD student can come for 3 months, can come for 6 months. That's how we are going to build a hybrid vigor. There's a lot in the experiments you do here. There are gold mines in the plants that we are carrying about. But those plants will not have any international bearing if you just squeezed the water out and drug the rat. Nobody is going to accept that. There has to be thin layer chromatography. There has to be NMR. We have to identify. We have to now take it with a binding ligand. But you can't do that here. You have the plants, but you don't have the facilities. Collaborate and bring something better out of that plant. Next slide. I think it's time to say thank you.
Wow, thank you very much, sir. And I'm sure we have a lot of questions, but we'll take only questions. We'll keep the comments for after, or except for the seniors, only comments. But we'll just, can I see hands so that we'll limit it to 4 because of our time. Okay, I saw you first, so I'll take you. Okay, we have 5 hands up just from this row. So all the 5, we'll take 5. So your hands, please, can you please pass? Let's start from the rear. Let's start from the rear. Start from the back. Yeah. Okay, thank you. From the back, please.
Thank you very much, sir, for the wonderful presentation. My first question is, I viewed on, or what are the possible solutions to the vanadium toxicity, like as an environmental toxin? So, and I also wanted to ask, because I saw one of the results that we compared time, about 6 months, 12 months, and 18 months, and also the generations. So I wanted to understand, vis-a-vis the environmental toxin that we have faced now, what generation are we? Like we that were sitting here, what generation are we? And is there a possibility that over time, there's a possibility that the vanadium can clear itself, just as the results indicated? Thank you, sir.
That's a very good question. Well, you are Generation Z. Isn't that what you call yourself? You are the last. Vanadium can actually clear itself. We've done that with some of the withdrawal work. Not 100%, but we are even still trying to study that mechanism. Even vanadium in the brain, over 6 months, can start to wear out. So vanadium can clear itself. The only problem is that some of the cognitive results don't really return back to normal. So that means there are some damages that have been done and that does not go back. The problem with vanadium is the constant exposure and how acute it is. So if we can withdraw ourselves from exposure, which is becoming a big issue nowadays. Coming to your first question, the world is getting more polluted. Volcanoes, forest fires. I know maybe now they are trying to use electric cars to reduce vehicular load. But I think the practical functional thing is good nutrition saves us from a lot of metal toxicity. Okay? Regular vitamin C, take a lot of water, exercise. Those things save you from metal toxicity. So I think for us, one of the things that could be our saving grace is good nutrition, particularly vegetables, fruits. Although nowadays we are now going away from those things, the natural vegetable, the fruits, and all of us take only Coke and sausage. So the more we stick with good nutrition, I don't see us really combating environmental pollution, particularly in Nigeria with vanadium. It's going to take a while. We will still explore crude oil. The people in Niger Delta will continually be exposed. I think I said this when I spoke in Port Harcourt. When he told me, I think it was 2011. I don't know the data now, but 2011, majority of the patients in UCH psychiatric ward were from Niger Delta. Majority of the patients in Ibadan. For them to come to Ibadan, that means the Port Harcourt couldn't handle them. And they now transferred them to psychiatric ward in UCH. Majority of the patients were from Niger Delta. So there's a problem. But I feel good nutrition, since oxidative stress is a problem, constant vitamin C with a lot of water can also help you mitigate the negative effects.
Good afternoon, sir, and thank you very much for the lecture. In the research you were showing to us, you made us understand that the last generation, they were not exposed to the vanadium, they were taken out of the vanadium environment to a new environment to grow. And despite the fact that there was an improvement in locomotion, there was no improvement in cognition. Now to ask that, where is the place of neuroplasticity in this, such that, is it possible that with training, their cognition could be improved? And also, if they give birth, is it possible that as they continue to give birth, without them mixing with those with good cognition, is it possible that the cognition gets improved over time? Those are my questions.
Excellent question. Let me qualify a statement. Cognition, not that it didn't improve, but it didn't get better enough compared to control. Okay? So that's the issue there. Yeah, training, plasticity, those are some of the ways, like I said, when I was talking exercise, and can help to improve conditions. And Dr. Igado is somewhere there. We are looking at some plants that promote remyelination. Okay. Now we have stopped at generation 3. Now that generation 3 that was not exposed, if they now produce their own offspring, generation 4, they're also not exposed. So you have another research topic. You can continue that. Are you getting? That's the beauty of research. So you can also look at that. And that's why we are happy with collaboration now with AYO. That is there 1 or 2 genes that were affected at the early days that never recovered? Okay. Why some genes can come back? Okay, there can be DNA repair. There can be some genes that they are so conserved that once they are destroyed, you have problems. Those are things we can still find out when we have better tools like molecular work. Thank you.
Sorry. Thanks so much, sir, for the question. Thanks so much, sir, for the presentation today. We really appreciate. The question you just answered now answered my first question, so I'll just go to the next question. So have there been researches or findings on how to trace, okay, for example, the 3rd generation of the specimen used? Okay, let's say, for example, in real life scenario, generation being exposed to vanadium, okay, gave birth to a new generation which transfers to an environment where there is no vanadium exposure. So over time, and if there is loss of cognitive memory, could it be traced to vanadium? Like, are there techniques or researches towards that?
Very good question. Not that I know. Not that I know. I mean, when you have gene mutations, multiracial societies like South Africa, you have a condition like maybe multiple sclerosis in somebody. You want to trace that family line because you know that there are some races that are predilections for such conditions. But to trace something and say it was induced by vanadium, no, it's not something that I think has been done. But mind you, like I said, over time, if you come out of a vanadium environment, the bioaccumulation of the metal can leave. But the damage that has been done may still be there. Now, whether you can now trace with epigenetics that those have a signature, that if you see this kind of expression pattern of transcription factors is vanadium, it's not something that I think anybody has done.
Thanks, sir.
Thank you, sir, for the eye-opening lecture. While you were speaking, you mentioned something about vanadium being easily attracted to lactoferrin. So you also mentioned that earlier you did a study where you quantified the amount of various metals in different parts of the brain. So my question is there like a way we can know the parts of the brain that are specific to different metals, like these sections of the brain accumulate these metals more? Now, why I'm asking this is because for my undergrad study, I worked with 2 metals, nickel and vanadium. And while we did the study, generally, we did not really consider the fact that maybe the thalamus accumulates more of nickel or vanadium. So I wanted you to just give me.
Thank you. Yes, like it was that job Larry did in Penn State using the laser ablation mass spectrophotometer. That's how we're able to see the different levels of vanadium in different aspects of the brain. And many metals are like that. Iron is also like that. There are some areas of the brain that iron concentrating. For iron, maybe they've also tried to characterize receptors, ferropotin, where you have high concentration of ferritin, which is the iron storing metal. But we don't know anything about that for vanadium. Because vanadium, like nickel that you mentioned, they are not natural metals of the body system. So iron we can understand because iron is important for physiology. So we are not surprised that there are receptors, there are storage proteins But for vanadium, only in lower forms, there's a particular mollusk that actually has vanadocils. They are vacuoles that store vanadium, okay, in a mollusk. So you can Google that with high concentration that is not in the body tissue, but it's inside those vacuoles. So that tells me one way or the other, there's a physiological way of releasing. But human brain, mammal brain, we do not have such a record of having a storage. But because body is plastic, if we are exposed to vanadium for 200 years, I won't be surprised the body will develop storage areas for vanadium storage proteins. The body is plastic. It will always find a way and go. But as at now, we do not. And that's just a speculation that with time, when something is constantly in the body will be trying to see how it's going to take care of it. I think that's the last.
Thank you very much for your lecture. So I want to know.
Excuse me. Are you a lecturer?
No, sir.
No, you should become one. I had you giving your oral presentation. You are speaking from your belly. Well done.
Thank you very much. Sir, please, in your lecture, you told us that you expose some animal over the lifetime, 3 months, 6 months, and all the like, that you measure vanadium from there. Now, I want to ask, how are you able to measure the level of vanadium in the brain of the animal periodically? Since you say you expose them for the lifetime, is it hematology or what did you do to be able to get the level, especially in the different brain region, that you know that the vanadium is there and it's affected? Then my second question, sir, I'm only curious, I want to learn from your wealth of knowledge. Permit me to say this. In your lecture, you keep mentioning that you got grant from ISN, from this. I follow your lecture very well. Right from my MSc base, I've been writing grant and all the like, and nothing comfort up to PhD. And our colleague that travels abroad, they work with grant, they work with what is happening in Nigeria, and how do we go about it? Thank you very much.
Thank you. Let me start from the second question. First and foremost, let me tell you that we got grants. We wrote many and we didn't get. Many were not successful, but we didn't give up. Okay? We didn't give up. So that's the first part. So once you are writing and you remain, you are going to get one day. Grants are becoming very competitive. Very competitive. But they are there. What is just natural, maybe to help you, is that 2 things for you. As tough as things are, try to publish your work. Because the person that wants to help you wants to see evidence that you have been helping yourself. Okay? So that's first. The person who wants to help you wants to see some evidence. The goalpost is changing. When we were looking for grants in the early days, the important, they would just say, you must have masters. Later, they say you must have a publication. Later, they are saying it must be in a journal with an impact factor of 1 and above. There will be another rule tomorrow. So the rules keeps changing. So it's tougher, but grants are easier through a multi-sectoral, multidiscipline. That means you join yourself to a lab. The professor writes for grant. They will tell him in that grant must be 3 PhD students that must be sponsored on the grant, 2 postdocs, and code. You start from there. Okay? And then with time, you finish your PhD, you have opportunity by God's grace to go and do a postdoc in a good lab. And so things build gradually. 2009, I know I got a UI Senate research grant. It's bad. I won't color things for you. I don't hear of Senate research grant in any federal. Maybe you still give them Unilag. But we don't have anymore. Good. When I was a student, lecturers used to get every year a Senate research grant. So those are things we need to bring back. The problem in any society is that don't allow things to spoil really bad, because when they do, Even when things are supposed to get better, people don't have the inertia to go back to how things are properly done. So, but when the Oremosus, Oyeles, they become provosts, VCs, and co, I want to believe, because someone asked me, why are these VCs behaving like this? Was he not doing research when he was a professor? Now he has become VC, he's doing nothing research. I said, when he becomes VC, he has become another human being. He's no longer a researcher, it's something else. So you have that problem. So let me go to your Vanellis. At 3 months, we sacrifice some and bring out the brain. Okay? So in the US, they had this equipment that from the brain slides, they had this laser ablation that could pick the quantity of vanadium in the different regions by doing an excitation to the wavelength of the metal. Definitely, you only do that with collaboration. There's no way you do that in Nigeria. So thank you very much. Oh, difference between hypomyelination and demyelination. When oligodendrocyte is destroyed before myelination, oligodendrocyte produce myelin. But in the first 6, 7, 10 days of life in the rodent, when oligodendrocytes are maturing and they are destroyed, definitely it's not 100% destruction. The amount of myelin produced will be lower than what you expect. That's hypomyelination. Your compound didn't destroy myelin, it destroyed the myelin producing cells. Okay? So vanadium can destroy the cells, but vanadium itself by oxidative stress can cause lipid peroxidation and destroy myelin. So that's demyelination. So the myelin was there, but the vanadium came and destroyed it. That's demyelination. But when you destroy the cells producing the myelin, reducing their capacity to produce, that's hypomyelination. Apart from the brain, what part of the body system is affected by vanadium? Virtually all. Virtually all. I think when I did some of my first experiments with rodents and I sacrificed the animal, I looked at different parts, different organs. The testis was gone. I remember that very well. And I had a Cameroonian who came to work in my lab. The ovaries, there were atresia of the ovarian follicles. So vanadium is really a bad metal. It has no use in the brain than to destroy. Thank you.
Thank you very much, sir. I know if we're given the time, we'll have more questions, but there's just, please let's appreciate him very well. Thank you very much, sir. Before we call it a wrap in this session, there's an announcement. If you forgot your AirPods in one of the sessions earlier in the different locations, please see the organizers with proof to collect your personal belongings. Thank you.
Thank you very much, sir. It's been a long day and we've had speakers, 5 speakers giving inspiring talk, and this last one just burst my bubble. I mean, I've always known him as a scientist. All of a sudden he became a motivational speaker, and so there's nothing as amazing as having people who inspire you to aspire for greatness. And I just love to fraternize with people who have provided leadership. Maybe I should say this, that Professor Lokpade was instrumental to bringing Paul Menja to this conference. So please put your hands together once again for him. So now I want to sincerely apologize to the crowd, to the entire participant here. We have overshot our time. We ought to go for break 1 p.m. and this is about 3. So you can imagine we ought to be having someone who is waiting to give his talk online. We have his pre-recorded session here. So I will just ask him to say hi to us and also apologize to him. So why I'm doing this is that I want to persuade us. We have our meal tickets. We'll just step out to just pick our brunch. Please make do with that. We will have a proper dinner this evening. So please just go. Please, that will be after the person online waiting. He will just address us for just 2 minutes. I just want to also apologize to him and hear his response. All right. He is supposed to speak 3:30 and he's waiting to take his talk. So Professor Kahir, okay. Can we hear you? I know you're hearing me.
Okay, I can hear you, yeah.
So we just want you to say hi, and we will have you come. We'll have your talk. We'll give it just a short break. So it's almost 3 now. So I think
please do take a short break
for us to go pick our meal and come back to take.
Yeah.
So have we agreed to come back by 3:30? We can be eating while we are listening to Professor Kahir. So Professor Okin Kahir. So please, if you don't mind. All right. Yes. So we want to first appreciate you accepting our invitation. And I know I got to respond to you a little late just last weekend after the last mail I got from you, and you've accepted to take our invitation. And I also want to tender apology for the time change that we're having for your presentation. So we just want to hear your voice. I'll be coming back by 3:30. Do we all agree on that? So please let everybody be seated by 3:30 to have Professor Kahir Okin from Department of Genetics, University of Cambridge, to speak to us. All right. So let everybody hear you say hi.
It's a pleasure to be here. I'm sorry too that I couldn't make it in person. I would love to have come in person, but this is all I could manage, unfortunately. So it's like everybody's been working hard, and you need a break. And I'm happy to wait and come back at 3:30 as well. So I think the way the way John has set it up is that he'll set up a recording of my talk, but I'll also be present online as well. So I'm be around afterwards, answer questions as well, for as long as people want to keep asking.
All right. Thank you so much. So
thank you very much, and thank you very much for the invitation.
All right. So by 3:30, we'll take the pre-recorded session. So you just wait to take the questions. Thank you very much.
Okay. Yeah. Okay.
Yeah. Thank you. Bon appetit.
Hi guys.
Hi guys, do you think you know about human brain? I meant to tell you that I cover symptoms. My name is Luca and I am advising you to Nsn Babcock 2023.
This Nsn Conference will be taking place at the Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but yet so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I'm here to tell you that I cover symptoms. My name is Boka for cancer and I am advising you to Nsn Babcock 2023.
This Nsn conference will taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys. Do you know that complex organ is so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I'm here to tell you that I cover symptoms. My name is Boka for cancer and I am inviting you to Nsn Babcock 2023.
This Nsn conference will taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but yet so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I'm here to tell you that I cover symptoms. My name is Boka for cancer and I am advising you to Nsn Babcock 2023.
This Nsn conference will taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but yet so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I'm here to tell you that I cover symptoms. My name is Boka for cancer and I am advising you to Nsn Babcock 2023.
This Nsn conference will taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but yet so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I'm here to tell you that I cover symptoms. My name is Boka for cancer and I am advising you to Nsn Babcock 2023.
This Nsn conference will taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but it gets so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I meant to tell you that I forgot to tell this. My name is Boka for Cancer Surgery, and I am inviting you to Nsn Babcock 2023.
This Nsn conference will be taking place at the Babcock University from the 23rd to 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but it gets so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I meant to tell you that I forgot to tell this. My name is Boka for Cancer Surgery, and I am inviting you to Nsn Babcock 2023.
This Nsn conference will be taking place at Babcock University from the 23rd to 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but it gets so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I meant to tell you that I forgot to tell this. My name is Boka for Cancer Surgery, and I am inviting you to Nsn Babcock 2023.
This Nsn conference will be taking place at the Babcock University from the 23rd to 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Hi guys, do you know that the brain is the most complex organ in the body, but it gets so fascinating? I think it's one organ you really need to know about.
Hi guys, do you think you know about human brain? I meant to tell you that I forgot to tell this. My name is Boka for Cancer Surgery, and I am inviting you to Nsn Babcock 2023.
This Nsn conference will be taking place at Babcock University from the 23rd to the 25th of August 2023.
And this conference cuts across different neuroscience fields like neuropharmacology, neurochemistry, neurotoxicology, neuroepidemiology, and I'd like to see you guys there.
Thank you very much for the information. It's better. and show how human genetics can help us to understand how it might be.
You are all welcome back to this session. Before we take the lecture by Professor Kahil, there is an announcement here, please. Listen to this announcement. If you have paid your dues to be a member of the Neuroscience Society of Nigeria, please listen to this number so that you can write it down. You can send the evidence of your payment to this number, which is the president's number, so that you can be included on the WhatsApp platform and other things that will need to be done. The number is 080-350-65190. Let me take it again. 080-350-65190. Thank you very much. So, like we are all aware, we went for the break just before Professor Kyle will give his lecture. So we are back. Before we allow the video to run, I would like to read the citation. After the video has finished running, we will have opportunities to ask. He's actually on standby, just like you know. We'll have the opportunity to ask questions, which he will respond. Okay, let me take the citation. Professor Kyle O'Kane is a professor of genetics and the head of postgraduate studies, Department of Genetics, University of Cambridge. He grew up in rural Northern Ireland and studied genetics as an undergraduate in Cambridge. A big picture shocked at the time. He did his PhD in bacterial genetics in Trinity College, Dublin, and as a postdoc in the University of Basel, he developed, he developed the widely used tool of enhancer trapping in Drosophila. His current research interests are in using Drosophila to understand how neurons function at subcellular level and how they can go wrong in ways that are also informative about human neurodegenerative diseases. He enjoys teaching and mentoring, contributing to undergraduate teaching in cell biology and genetics, and acting as director of studies in Churchill College. Research interests in his lab. The lab is interested in the basis of axonal endoplasmic reticulum and its relevance to neurodegenerative disease, using Drosophila as a model. Axonal ER, which is endoplasmic reticulum, forms a continuous tubular network throughout the neuron, comparable to a neuron within neuron, potentially able to integrate physiological responses throughout the neuron. It is important. Is it important? Almost certainly the motor axon degeneration disease, hereditary spastic paraplegia, result from mutation affecting several ER-shaping neurons or proteins. So that is the bio of Professor Kahir. So please sit down, listen to the lecture. I believe we are going to be educated for us to appreciate why they titled this idea neuron within neuron. Thank you very much. So technical, please, can you play the video for us? Even though it's a video, can we put our hands together for Professor Kahir? Thank you very much. So technical.
Understanding how is organized ER is in the half of the other half is how the effects in that organization lead to action degeneration. So in other words, other actions are question to you in asking what's the current that's what one reason why I was and here in the bottom row is an example of the speed of here on my share. And the bottom row shows very American of the software and not states related to cell bodies, which are circles with the black nucleus in the middle. But if you look at these in the middle, or not that one, these columns here are subscription marks, so not they illustrate that the option not needs black brush. It's really clear when we realize that there are markers. Most of the markers that were commonly used in ER that were proteins involved in the rational function of ER, which is protein export and proteins like the chaperones, proteins, or components of the spatial pathway. And then we realized that we should look at markers which newly are or tubular which is much less active in proteins. There's one of the sites of lipid biosubstance. And here's an example of why I feel that particular one is tubular. So the little bit of plastic the against the like rising started losing was found in the sea kinds of New York the distribution of the concept ER marker in the whereas the different white meat is one you can even see that my kid goes all the way to the very now, I want you to use more to practice use for a lot of work. This is a effective example of our but it's very easy to work with. Was actually used an embryo of 5 years later. The embryo was hatched and growing to the length of 4 meters. So they can then the content the back end on the right to the second dish and use scientific view on the back and the right on the left body wall onto the dish. And then remove the always and then this back we can see a very little script that America just If you use the given the maximum, you have to go further use additional markers that we can express in one or two accents at a time. Let's use a very partial system called down four expression where we use two components. We use constantly try one component these constructed background four, which is not present in the emergency system. And then we express they are memory marker in this case, ACL, which is a if it's used to the US to applications that are in mind or prescription factor, it's remarkable. Should I use this one? Where we see that these spastic parties, proteins that spastic parties, membrane shaping membrane proteins are essential for axon ER, contributing both the number of tubules and contributing both the diameter and also contributing both diameter and also how do they
Professor, thank you. Professor, thank you. Professor. Before I open up, I would like to say a number of things, then we'll open the floor for discussion. You know, if you have followed this lecture very carefully, you will have appreciated a number of things. Number one, Drosophila studies have been in the making for over 100 years. And quite a number, if you go and take a journal that is like 30 years old, you will not see these things, or you will not see all this fine visualization and stuff like that. You will not see. In fact, that is why today, when you go to some places, in fact, journals are doing competition for images. They will say, if you think you have a very fantastic biological image from your lab, you can send it to this competition. You know, so it has now become a competition for art in life sciences. Some of the images that they are generating, as you just saw from the labs, they look like artwork, not really science. And this is because each of these things and each of the antibodies that have been used to serve as markers, you know, they are giving information that may look just like an artwork to somebody who is not really familiar with what is happening. But another thing I would like you to also notice is the fact that Professor Kahir, PhD was bacterial genetics. Did you see any bacteria in the presentation? Did anybody see anything bacteria in the presentation? Okay, so this is another thing that we also need to learn. You can actually start an interest in a particular thing and eventually change your interest from bacterial genetics to Drosophila axonal ER research. That is one thing I would like you to also learn. Then the other thing is for us to appreciate what he just said, neuron within neuron. Why do we have endoplasmic reticulum in the, by the way, what we know as cell biologists is that endoplasmic reticulum actually is just opposing the nucleus. You get the logic? And then can get continued to the cell membrane. But you can see this one is not only within the cell body, it has gone down to the terminal. So, and that is what they are actually trying to look at. And if you follow the lecture carefully, you will have appreciated the fact that they are trying to establish the role of this endoplasmic reticulum in hereditary spastic paraplegia. I had the privilege to be in a lab in 2012 where we were doing something a little closer to this, even though in a completely different model. And the model is squid. You know, squid is a marine organism, and squid actually has what we call a giant axon. This giant axon, you don't need a microscope to actually see it. You can see it with your naked eyes. You know, so what we were doing there was that we'll get our squid, we'll come and dissect the squid, we'll remove the giant axon, we'll cut part of it, we'll do fine dissection and extrude the axonal content, what is called the axoplasm, and then we'll apply whatever chemical of interest or ligand of interest or whatever thing that you think can actually help. Because if you follow this lecture carefully, you will appreciate the fact that is the communication between the cell body down to the next neuron. That is where the problem is. And if this is actually hampered, that's what actually leads to neurodegeneration. Like in this instance, hereditary spastic paraplegia. That's the same thing that actually happens in all other neurodegenerative diseases. Alzheimer, Parkinson, Huntington's, and stuff like that. So, we would like to open up the floor for questions to Professor Kahir on neuron within neuron is structure and function links to hereditary spastic paraplegia. Questions? Yes, sir. Professor, will you? Okay, I thought you are raising your hand to. Okay. Questions? Dr. Imam? Technical, please, can you get the mic?
It's very complex for many of us, right, to kind of go with the lecture. We really enjoyed everything. What I will appreciate is if he has the time to help simplify the role of mitochondria, right, in keeping the integrity of ER, right, within the exons. So even if that's going to be the take home, we'll start from there and then read further and then follow your research output to also update our own knowledge too. So thank you so much for your time.
Thank you, Dr. Imam. So, Professor Kahir, can you just take 1-2 minutes to simplify the whole concept so that the audience can have a take home?
Yeah, so we haven't worked much with mitochondria. Well, we looked at the imaging, and mitochondria and ER they contact each other and talk to each other a lot, and in case one is that is signaling and which helps to stimulate. In most cells, the main source of calcium for mitochondria comes from the ER. So the ER is a really important calcium store for the cell and act as a source of calcium, not only releasing it into the cytosol, but also releasing it to other organelles, including mitochondria. So I would expect, although we have, I would expect that our mitochondria, as a result of the ER defect that we're seeing, would also lead to reduced ATP generation and increased energy stress for the neurons as well. But that's just a prediction. We haven't had a chance to test that yet. Now, the other way in which ER mitochondria talk to each other is by exchange. So most lipids in a normal cell under normal circumstances are made in the ER membrane, and they're then transferred to other organelles by close contact between the ER and other organelles. So you get lipids going from ER to mitochondria. You also get lipids going back from mitochondria to ER as well, depending on where they're made and where they're needed. And so the two organelles do affect each other quite a lot. And communicated back to the ER as well by those exchange of molecules in between them. And so ER stress can be communicated to mitochondria. Mitochondrial problems can also be communicated back to the ER as well. I don't know if that answers your question, mitochondria. I'm not sure if I've simplified the ER much, but I've tried to explain a little bit why we're interested in mitochondria and why ER and mitochondria. It's really important to consider them together. If I haven't explained, just ask me or ask me again. Sorry about that.
Thank you very much. Are there other questions? Understand the fact that the function.
The simpler the question, the better, probably. The most important questions are simple ones.
Any other question? Oh, okay. There are two hands. Oh, okay. There's one hand at the back there. Let's remember the function of the ER, the function of the mitochondria. So there is always an exchange between them.
Thank you very much for the presentation. My question is a little bit deviated from the ER and but I'm trying to understand that most of your research, you use the Drosophila larva for most of your research. And I was thinking, is there a possibility and I'm particularly interested in the ventral cord of the adult Drosophila. And I was thinking if there's a way of imaging or possible imaging of the Drosophila adult ventral cord and all the axons and neurons in the flies.
Yes, it's possible to image adults as well. One of the reasons why we do a lot of our work in larvae is to generate a lot of the genotypes that we need to use. We need to set up a cross between a paternal genotype and a maternal genotype, and we then get progeny of the genotype that we want. And we get larvae with and can start imaging them. Whereas we wait for adults, we would probably have to wait at least another week to get them. Also, some people do work with adult ventral nerve cord as well. I find them just a little bit harder to dissect, but they're not that difficult. And of course, one thing that although we're working with genes that are implicated in axon degeneration, you might have noticed that we're not actually looking at any degenerative phenotypes in larvae. And I think if we wanted to look at degeneration itself, rather than just looking at the subcellular organelles, so the larvae are great for subcellular organelles, but if we want to look at degeneration, I think we would have to start doing more work with adults. We've tried a little bit of a work with adults and haven't really seen degeneration, but the mutants that we look at, they die after about 10 days of adult life, and we might need ones that live a little bit longer to be able to see neurodegeneration. So adults, you can certainly work with adults. Some things larvae are better for, other things adults are better for. You choose the system that is best really for the question that you want to ask.
Yeah, thank you very much for that answer. My friend, the story is this. you locate the most appropriate thing that can answer the question you are pursuing. And that is why, for instance, in developmental biology, eggs are actually used to study whatever system, for instance. Like I just told you, the experience I had, we are not actually, we are not even using the nerve like they are actually using. We are even extruding the content, out of the nerve, and then studying how axonal transportation is actually taking place, which is what I actually saw they are doing in the lava. So in this particular instance, the lava will have been the best thing to actually use for this study. So if you are even using whatever model, you can use whatever part of that model or whatever developmental stage of that model to actually answer the particular question. And in this instance, this particular approach, it will have seemed the best because you want to see what is actually happening. And as you can see, they have reported through their studies, they are able to establish, or they are able to actually see that there seems to be a link, between this extensive anatomical feature and hereditary spastic paraplegia. So do we have another question? If we don't have.
Yeah, that was a really good question about the adult, by the way.
Okay, can we put our hands together for Professor Kahil? Professor Kahil, thank you very much.
Yeah, you're welcome. It's been a pleasure. And thank you very much for the invitation.
Our hands together. John?
All right. Good afternoon, everyone. Some people are dozing, and please, if somebody is dozing beside you, just give him a nod. Thank you. Some people are sitting on the style and the position of almost to sleep. Thank you for sitting up. All right, ladies and gentlemen, I'm sure that we are having a swell time, and it has been a rewarding moment that we have exchanged thoughts and are still learning from each other. And I sincerely want to thank the last speaker. We will be taking another speaker, and I will tell you authoritatively that that will be the last for the day. Please, if anybody comes forward and call the name of another speaker, carry your bag and walk away and tell them that I said so. If you quote me, I will deny you. All right. So we had brunch. Can you please, where you are seated, just stretch. I don't know how you stretch, but I don't know how you do it, but just stretch. Just stretch. Maybe you should just stand small. Just stand, stretch for 30 seconds or 60 and sit back. That's all. I'm not asking you to do anything more rigorous. Please, please, please. Hell, see the way you're even looking at me. Please now. Thank you. Yes, ma, you are so balanced. Thank you, thank you. Now, those people that didn't stand, watch them. They may likely doze or sleep off. I'm telling you. Aha. Please, you may be seated the moment you feel very comfortable. Thank you very much. May I invite the console, please bring forward the next speaker. We will be having Dr. Joshua Owolabi. He's going to be the next speaker. Until I see his face, I am not going to say anything. Dr. Olabi, can you confirm hearing me?
I can hear you, Dr. Dagana, and I can see your face.
Wow. Wow. Let me publicly give this disclaimer. Dr. Olabi is my personal friend beyond this podium. It's good to see you. I will not do justice because I am intimidated by what I will read. So I will just be very brief. Dr. Olabi, if I allow him, he may combine all the plenary session time that we have spoken today for him to speak. I can tell you that for free. I'm going to take liberty, sir, that because I know you in person, I will make some unprofessional statements like the one I just made, that you will talk for long. So, but I know that you will be able to manage our time and your own time also. He will be, after I tell you who he is. Distinguished guests, ladies and gentlemen, before us, virtually, is Dr. Joshua Olabi, and he will be speaking on neuroepidemiology and drug epidemiology, making sense of experimental, clinical, and epidemiological neuroscience research towards effectively addressing Africa's burden of neurological disorders. I hope you wrote that down. Okay. Ladies and gentlemen, Dr. Joshua Olabi currently serves as an associate professor at the Philadelphia College of Osteopathic Medicine in Georgia, United States of America. He teaches anatomy, neuroscience, and related basic medical science subjects to medical and allied health students. Olabi is never tired of learning and reading. He has a PhD in anatomy and then another PhD in public health with emphasis in neuroepidemiology. Olabi also has an MBA, postgraduate diploma, a master's in medical education, and others. He even has WAEC result. Very clearly for us to know, Dr. Olabi has had the opportunity to have a research fellowship at Madrid and a postdoctoral study in Yale University. He won the Ibro Return Home Grant upon the completion of his postdoctoral training in neuroscience. He has served as the vice president of the Society of Clinical Anatomists in Rwanda, and he has also served at IFA World Council. He is and has served as the pioneer head of anatomy department at the University of Global Health Equity in Kigali, Rwanda. He loves reading, he loves publishing extensively. You can go online and find out more. He has also served as an external examiner to a number of universities within Africa. But I will tell you this, which is not written here. Dr. Joshua Olabi is happily married. Just for you to know, he's happily married and he has 3 beautiful children. Because he's an anatomist, I'm telling you the 3 children was when he left here. I don't know after he left if more blessings came his way. Thank you very much, sir. You have the floor, and we will give you half an hour. And I hope that that should be sufficient, and then we can get reactions. Please, I will request that as he speaks, you please put down your questions. Thank you very much. Half an hour.
Thank you very much, Dr. Dagana, for the introduction. I hope I'm audible enough. Just wanted to be sure. And for adding what was not written. So I'm trying to share my screen now. I hope you can see my screen, and I hope I'm audible enough. All right. So I'm going to speak on the topic as introduced.
If you can hear
addressing-
me, if you can hear me, you are audible and we can see your screen. Just to
Thank
give you
you.
feedback.
All right, sir. Thank you very much. So addressing the burden of neurological disorders in Africa, that makes it short. By integrating experimental, clinical, epidemiological neuroscience in the context of neuroepidemiology and drug epidemiology. And I want to acknowledge the leadership of Neuroscience Society of Nigeria, SONA, Babcock University being the host, all neuroscience educators in Nigeria, the neuroscience community in Nigeria, mentors and leaders, some of which are also my mentors and my leaders, every one of us. Thank you very much for being a part of this. And for me, it's a privilege again to be part of this. Having said this, it's important, therefore, that I speak about three things as I also try to do justice to this topic from a scientific perspective. Why neuroepidemiologists? I'm going to talk about my inspiration, my experiences, and my recommendations. And that is what we should do at the end of science. We should be able to inspire further studies. We should be able to talk about solutions. So, how did I become interested in neuroepidemiology? Because from the introduction, I happen to be an anatomist primarily. I had my PhD in anatomy. I remember the first encounter I had with neuroepidemiology was as a Yale postdoc, and I was talking about how much research I wanted to do. And my mentor called me and said, "What problem would you like to address here?" And I said, "Epilepsy." And she said, "Great. Can you talk to me about why you're interested?" And I gave very beautiful scientific presentations of what epilepsy is and all of that. And he said, "Okay, I expect that you probably go back to Africa when you're done with this. So what exactly is epilepsy all about right now in Africa? Prevalence, people that are affected, causative agents and all of that." And I struggled to answer that question. And it dawned on me that no matter how well I performed in the laboratory, if I didn't understand carefully and very well the problem I was solving, I'll probably just be doing the routine. So by the time I was concluding my second postdoc, I decided to enroll. First, I wanted to do neuroepidemiology straight away, but I couldn't get that. So I did public health, but then with neuroepidemiology specialization. I was done with postdoc and I thought I should just return and do what I've been doing before. And I continued my experimental neuroscience research. And I had another encounter with Professor Davies. Again, I was representing my institution, as at that point in time, I was a visiting assistant professor to Nebraska. He was a vice president, senior vice president at Nebraska. And I started talking about research again. And he asked a similar question that I had a couple of years before then. And he said, "Great, you will be dealing with epilepsy. What is epilepsy? Where you come from? What are the risk factors? How many people are affected? What justifies," those were not his words, but I was trying to summarize. "What justifies the investment that you want to put in this research and the return on this investment to your population and whoever is funding this?" Again, I was stunned. And it became clear that I also needed to do more to understand the problem that I was trying to solve. So those two encounters, among several others, actually steered me into neuroepidemiology. Having said this, so that accounts for my inspiration. I will talk about my experiences shortly afterwards. My time has just been reduced by half, so I've got to make the best use of it. So I will start with this question: why do we have to do research? And there could be 1,001 answers to that. But again, I will start by way of reflection. And I like how Elon Musk will answer a question like this. He will talk about first principle approach. Don't forget that Elon Musk is a physicist. And what is the first principle approach? Is to break things down. So why do we do research? Primarily, we could have a lot of answers, but very importantly, we want to improve the current situation and performance, life performances, whether it is mental health, brain health, or what have you. We want to create solutions through products, processes, etc. That's the basic reason why we do research. Of course, secondarily, there are other reasons. For professional advancement, if you're in academia, you have to publish papers. If you're in the industry, you have to create products through research. There are people who do research for humanitarian purposes and others because it's just their inspiration. So it's therefore very important that when we talk about neuroepidemiology and neuroscience research in general, we should ask ourselves the question, why do we even do research in the first instance? And we should be able to answer that at our own level. Why am I doing research? So let's take a quick look at what we call the global burden of disease. Presently, globally, we know that mental or brain health problems that we call neurological disorders actually constitute the leading cause of death and disability globally. That is documented. And we can look at the array of problems that are contributing to mental health disorders or challenges or problems. But this is a global picture. So it's important, therefore, to localize this and think about Nigeria and Africa in this context. So a very quick snapshot at what we call area of concentration of research in Africa. And special thanks to Maina et al. And Ekerian and Okechukwu for actually doing our work on this. And they realized that experimental modeling actually topped the list in terms of how we do research. But the technical input is low to medium. So the question is this: does the research or do the research that we do, does it match Africa's research needs? So, and then you can see that there's this distribution. If you look at the North African, you see the bubble, it's relatively large. Then you come to West Africa, and then you go to Southern Africa. These are, so to say, the powerhouses when it comes to neuroscience research in Africa. But don't forget that most of what comes from here, if you look at the blue circle, is experimental research. Now let's go to neuroepidemiology. What exactly is neuroepidemiology? The term is not new. It was coined over 50 years ago by a man called Hotsky. And he actually talked about doing epidemiology in the context of neuroscience, more like building epidemiology into the context of neuroscience. So neuroepidemiology, it's not new. And he further posited that it's not just about looking for prevalence and data, that if we're able to do well with neuroepidemiology or epidemiology in general, we will be able to prevent most of the problems that we are actually trying to solve. And it's always better to prevent as such. So what are perspectives, therefore, to neuroepidemiology? One would be classical neuroepidemiology. Another one is from clinical perspective. And another one is from experimental perspective. And when all these come together, we can have an integration, a point at which we bring all these three domains together. So what would be classical perspective to neuroepidemiology? That is what a typical public health person would do, looking at mental health problems or brain health problems. What would be the clinical perspective to neuroepidemiology? That is what a clinician would do, such as from the perspective of neurology and other mental health disciplines. What would be experimental perspective to neuroepidemiology? That is what experimental neuroscientists would do. All the effects of one substance on a part of the brain and so on and so forth. And so the primary domain here will be biomedical sciences and all its subspecialties like anatomy, biochemistry, pharmacology, physiology, etc. And that is why we use model to model whatever we see in real life. But it's very important to know that these perspectives have to come together. So we should be able to create overlapping perspectives. Very importantly, if we advance the field of neuroepidemiology, clearly it's going to open new doors of career paths and opportunities for African neuroscientists, especially those that are aspiring to do much more than effect case reports and proposal of interventions of different types. Because everybody can find their niche, whether they want to study an agent, the environment, or the host. This is what constitutes what we call the epidemiologic triad. Now, we should know that neuroepidemiology or neurological disorder is not just about the disorders themselves, but the burden that they create. And they make the highest contribution to what we call DALYs, that is disease-adjusted life years. Some of them don't kill, but they cause disabilities that are significant. And if you look at it, stroke tops the list if you look at the other diagram here. So what would therefore be African needs? We need to do more research in neuroepidemiology. Data is sparse. We need specialized education and skill in the aspect of neuroepidemiology. We need funding, and we need to take care of what we call implicit bias. Oftentimes, we have more funding from outside Africa for neuroepidemiology than within Africa, and the reason is clear. When you put all technocrats into the room and all of us have perspectives from only experimental neuroscience or clinical neuroscience, we would definitely think the way we have been trained. And that is a sort of implicit bias. So we need to widen the scope. We need multiple perspectives. And implicit bias is real. Whenever everybody has the same background, has had similar experiences, it's always hard to think out of the box. We need to network effectively. We need to begin to start new programs, and we need adequate and implementable policies. Policies become very important. Again, this explains why scientists need to study and better understand policies as well. Science is now much more beyond the laboratory. So we can now take a look at what we call leading neurological disorders. And we know that even when we know that in the world, the top is probably stroke. In African context, it's in the order of epilepsy, neuropathy, peripheral neuropathy, stroke, and of course, HIV/AIDS is now contributing significantly, as well as trauma and alcohol abuse. In fact, it is known that dementia and HIV-related neurocognitive disorders are top in the list, and there's evidence for that. So when we take a case of Konzo, for example, Konzo was an outbreak in Congo. And what was the problem? Poorly processed cassava. So imagine what the scenario would have been if there were no adequate epidemiological studies, if all of us were busy trying to look at whatever was happening, whether in the body systems or in the cellular domain. So what we are trying to say is, why some of us will be busy looking at the cells and tissues, some other people are also looking at the whole body versus the environment and other factors. And that simply means that all perspectives to neuroscience research should be considered very important. Now, I like this, and I want to talk about each of these areas that I've highlighted: experimental neuroscience or neuroepidemiology, clinical, and of course, neuroepidemiology, about African experience. We need to now begin to think about system. How do we begin to enhance our system for research? Program is another thing, people skill, funding, facilities, and the culture of our research. But before I come back to this, let's look at what we call the pyramid of evidence. I'm a member of the Joanna Biggs Institute of Evidence Synthesis, and we talk about the quality of evidence. At the very base of this pyramid, we talk about background information and expert opinion, then animal or laboratory study. But the most quality evidence that people really deploy at the level of policy and products, such as drugs and vaccines and what have you, is the one that come at this level: systematic review, meta-analysis. Now, for most of the experimental neuroscience study we do, this is where it falls: animal study and laboratory studies. So that speaks to the applicability of some of these. They will contribute to knowledge, and there is no prejudice against that. But let's begin to say what is going to take to actually bring to bear on the real life solutions, the research that we do. How do we address this? Let's begin to now think about how we run our programs. Research agendas, do we have them clearly set? Our master's program, do we have clear focus for them? Our PhD, our postdocs. As a policy advocate, one thing I've always advocated for is that we should have more postdocs being trained on the continent than those that are trained outside and come back. And I must say, those who train outside and come back don't have great experiences. We will always name that. I had the privilege of being trained when I was at Madrid by Professor Paula Bugolenta. If you know her, she's going to be the IBRO host for this year, one of the greatest, mentors and researchers of our time. I learned about not just, doing research in the lab, but organizing system. So it's therefore very important that we begin to replicate that, but that we train people within the context where they would work. It matters a whole lot. Now, when it comes to clinical neuroscience, then we look at the body of evidence. Most of the research will best qualify as case series and case report. Again, on the pyramid of evidence quality, we see where that will take us or where that belongs. So what do we need to emphasize in this case? Among these key considerations: system. We need to begin to train more of physician scientists or physician researchers. We have those group of people in developed part of the world, such as the Americas, for example, thinking that reporting cases translates to cutting-edge research needs to be reviewed again. I don't want to say it's problematic. We need to look at people's skills. To be able to interpret research findings goes much more beyond classical, pattern of interpretation. So we now begin to enhance people's skills. When it comes to neuroepidemiology, we need to think about system. How do we make sure that we promote neuroepidemiology and that we create system for it to thrive? So that we have a group of neuroscientists, for example, they are not just like the anatomists, physiologists and pharmacologists, for example, that we have psychiatrists, that we have public health people, that we have nurses who are interested in mental health, that we have educators who are interested in education and neuroscience, that we have policy people who are interested in system management, for example. And this should now influence our culture of research. It should be pragmatic, should be multidisciplinary, and so on and so forth. So someone might ask, what are your experiences? What have you done? For example, I've been promoting neuroepidemiology and drug epidemiology, and I'd consider drug epidemiology as a critical subject of global health, mental health, and health equity. Because drug epidemiology, now I've moved from neuroepidemiology to drug epidemiology, because it constitutes an integral aspect of epidemiology globally. Because epidemiology simply means the distribution of diseases upon populations, epi, upon people, diseases upon people. And so if a significant population globally suffers from mental health problems that are associated with drugs or substance use, then it should be of interest to us as neuroscientists and brain health researchers. So drug epidemiology therefore requires adequate attention, and very importantly, intervention should be evidence-based and of course, health equity principle guided. For example, this is what global data tells us about the burden of drug abuse globally. It tells us that when you look at the top of the list, that cannabis, followed by opioid, top the list. But this is our finding, which I co-published with Olarewaju et al. Our finding shows that in certain part of Africa, Nigeria to be specific, tramadol, cough syrup, shisha, and cannabis actually comes before even some of those ones that are consumed or abused in other parts of the world. What this tells us is that there's nothing wrong with the global data, but we now have to begin to understand our local context through research. According to the WHO, any substance that affects mental processes is considered a psychoactive agent. And when it is abused, it becomes problematic. It could even be illicit. And so people have now started using what we call new psychoactive substances. And this includes repurposing pharmaceutical products such as codeine, we know that as Nigerians, plant-based psychoactive agents such as datura, newly modified drugs such as synthetic cannabinoids, mixtures such as nyaope in South Africa, which is becoming endemic. And of course, these are often difficult to track because what we have on paper is that we have NDLEA and other bodies chasing people who are carrying cocaine, heroin, which is great. We should keep doing that. But very importantly, there are others that are not necessarily considered hard drug or illicit, but they are creating mental health problems. So, codeine, newly modified drugs, nyaope, datura, even fauna, these are dropping. It's now considered a psychoactive agent. So what are the key points here to researchers? We need quality data so that we can have good return on investment in terms of the research that we do. The practitioners, they have to be pragmatic, looking at what is really relevant to the environment. So policymakers, we need to align our policies with what research, data, evidence tells us. So public health people, we really need to address problems and not just do routine. To the society, we need to support research. We need to take responsibility. So having said this, let's come back to our own approach or the predefined approach to neuroepidemiology. So let's talk about basic epidemiological research. We need to have data on prevalence, causation versus association, for example, survey, and much more about the impact on this on the quality of life. So we need to stop doing just prevalence study to start looking at quality life adjusted years. disease life adjusted years, the burden of, disorders that result from mental health problems on, the population, the well-being of the population, even the economy of the population. When it comes to clinical and research reports, we need to begin to be able to track outbreaks and trends, for example, using data. Experimentally, we need to be able to model more effectively model diseases. So we need to be able to think through what we do and be able to model real life situations. So if I'm using a particular dose of an agent to induce a condition, is that the quantity that people are typically exposed to? So an example was when we were doing the research on radio frequency radiation, and we needed to look at, okay, what is the typical human exposure to radio frequency radiation through their phones or their mask or whatever it is per day? And we had to look at the tissue. This is an animal tissue, this is a human tissue, volume versus, the size of the cell, the intensity of radiation. Otherwise, we'll not be getting results that could be applied. And not every experimental result actually makes sense in real life if we don't properly model it. They can contribute to knowledge, but are they really solving problems? Of course, we need to also think about policies, and we should be able to engage the powers that be. Having said all of this, research in this postmodern era is typically cutting edge and collaborative. So collaboration. When we talk about collaboration, it has to be multidisciplinary. It also should cut across borders and boundaries. And that is why the new generation of researchers should be able to network and be able to think in such a way that our thinking transcend our locales. So neuroscience is growing in Africa. Laboratory experiments have dominated what we do, but we need to now begin to focus more on neuroepidemiology and drug epidemiology without stopping what we are doing before. In fact, we need to do more of what we're doing. So there's no prejudice against experimental and clinical, aspect of neuroscience. By the way, I still do my experimental neuroscience to the best of my ability. We need to understand our strengths, our weaknesses, our opportunities, and threats. Very importantly, we need to strengthen the culture of our research system through what we call academic and research leadership. And what should effective leadership do? And when I say the leadership, I'm just not talking about president of association. I'm a student of John Maxwell School of Leadership, and I know that there are levels of leadership: leadership by position, leadership by influence, leadership by productivity, leadership by identity. So every one of us that belong to a sphere, that find ourselves leading within a sphere of influence, should be able to contribute to the advancement of science by acting as leaders in those, spheres. Having said that, effective leadership should be able to lead. In other words, for someone to be able to lead in research, there should be a model. So how do I become a director of research, for example, when I don't show that I'm doing quality research, cutting-edge research, but I'm the one to set agenda for others and give them policies? And so there's no leadership when that leadership cannot be a reference, cannot be a model. We should be able to equip people. That is why we need to train them. We need to give them certification program. There's always something new to learn. I always enroll in programs to learn more. To envision, we should have research programs. All institutions cannot be specialists in every aspect of research. So we should be able to bring our strengths together. We should be able to inspire the younger generation and the world around us. And there will be a few other things that we could do as well. So again, for example, how have I been able to do this? For example, I belong to groups with 4 active grants. We have a grant from the Center for Chronic Diseases and Aging, for example, and we are characterizing ultrastructural changes in neurodegeneration disorders leading to the cause of death using human tissues. We are also doing what we call a community research. Research. We also have a community research fellowship grant, and we are looking at neuroepidemiology in the Georgian community. In other words, we are studying how people come down with neurological disorders, and we have people from different fields, including anatomists, pathologists, and so on and so forth. I also belong to the Harvard Culture Lab grant group, and we are looking at something as simple as muscles of facial expression. That will interest you. Why is that neuroscience? But of course, behavior also has some behavior is also like the neuroscience. And of course, we are also looking at how we can use educational technology. So in other words, we should be able to model what we preach, so to say. And of course, in a community of practice, for example, we have African Network for Drug Epidemiology and Neuroepidemiology. So like they say, you put your money where your mouth is. So if we say neuroepidemiology is important, then we have to start advocating for it. And so there's an Pan-African movement that is working in that regard. I also consult with Academic Without Borders Canada. I've been supporting a number of federal, at least currently 2 federal universities in Nigeria. I've been a guest editor to neuroepidemiology, for neuroepidemiology on SAGE. And I've used my position as a vice president of a professional body to advocate for diversity in research. And that's just among a few other things that one does. I've been asked to speak for less than 30 minutes. And it's good for Dr. Dagana to know that people change. So I won't speak more than I've been asked to speak for. I've included a few references here. And of course, I will await your questions. Thank you very much.
Can you confirm hearing me? Oh, Josh.
I can hear you. Yes.
Dr. Olabi, can you confirm hearing me?
I can hear you. I do confirm, Dr. Tangana.
Okay. Why that is being sorted? Excellent. Excellent. I will give room for questions, and I will start with this role. Does anybody have a question to please signify? Okay. The chance for grace is over. I will come to this role. Any question? Your silence can mean 2 things. 1, we fully understand, we don't have a problem. 2, be fast, let's close and leave this place. So is it A or B? Okay. Okay. All right. So the middle role now. Question. Okay, that's 1. Can he? Is there a movable mic? Okay, please come. Okay, yes, go ahead.
Okay, thanks so much, Dr. Joshua Olabi. I'm also Olabi Manuel by name, medical student in Babcock University. So I've learned that part of neuroscience, a scope of neuroscience is, of course, the clinics, okay, it talks of psychiatry, neurologists and all of that. We've learned that all of this can contribute to epidemiologic studies of neuroepidemiology. So I want to know if neurosurgery also can contribute to this. Thank you.
Okay. Just for emphasis sake, Josh, his question very specific is he wants to know if neurosurgery can also be a contributory factor to neuroepidemiology. I'll yield the floor.
Yes, sir. Can you hear me?
Absolutely.
Yes, the answer is absolutely yes. And that falls under clinical neuroepidemiology. So how will a neurosurgeon, for example, by having data, reliable data as to, for example, prevalence, for example, the pattern, of presentation, pattern of progression about specific disorders in such a way that the data becomes empirical and can be applied to, other contexts if they are well adapted. So that way, neurosurgery is contributing to neuroepidemiology. That takes us beyond just mere case report. We have this particular patient, he came in this particular way, and then we did this, and then this happened. Wonderful, a publication comes. By putting this data together, it makes a lot of difference. For example, at a point in time, we worked with a group of neurologists and neurosurgeons in East Africa, and we decided to look at the data, the clinical data for years, for example. And so whatever they had done in that clinic, which was a referral center, we could see the trend. We could see how many people came back. How did you intervene? What were the outcomes of the interventions? What could you do differently? By the time this data begin to inform best practices, then it becomes a data for neuroepidemiology beyond just case series and case reports. So the answer is absolutely yes. And there's, of course, many other ways to do that, but the answer is yes. Thank you.
Thank you. Thank you so very much. And let me re-echo this, that beyond the scope of what the presenter have spoken on, there are also other areas. What he has said is not exhaustible. In other words, there are more opportunities of things to rediscover and discover. There are emerging other psychoactive substances and drugs that are not documented. That are not documented. And I'm sure that even within the locality of where you come from, if you look inward and closely, you realize that there are other psychoactive substances that the people are getting the same quantity and depth of highness, you know, like going through this conventional one. So let me not be tempted to give another lecture. Any other question? You want me to call you by name to ask? All right. Mano Josh, I guess that you have stimulated minds and thoughts enough that they appear not to have questions as I thought. Thank you very much for your time and for the depth of what. Please, can we put our hands together for him?
Thank you very much.
Thank you very much. In the quantum of what you have made mention of, I heard various groups that you are looking at and synthesizing and analyzing emerging diseases. Psychoactive substances. Are you looking at that more from a developing country point of view or a global perspective?
Thank you. Our emphasis has been on the developing world because it's easy to track that in the developed world. I mean, for obvious reason, you can hardly do anything around here without it being tracked. But in the developing world, a lot of things pass through the cracks and you can hardly figure that out. So our focus, again, talking about return on investment, if we're going to put money into research, let's put it where it matters. So where we really have great, gray areas is actually the developing world. And that is why, and then as focused on, Africa. Yes. Thank you.
Thank you so much. The reason why I asked, I had a student who in the passing year, I never knew it existed, that a seasoning, if you remember a seasoning in Nigeria, generally called as Maggi, Maggi is being used together with lacasera, and it gives you a complete new feeling. It's amazing what the extent at which people go to find new feelings and excitement. And I think that I do not know whether these are, because sometimes you are caught in between the devil and the blue sea, such that people who do not know, when you bring it to beer, it's like, okay, you have opened a new option. So you begin to see now that when you go for occasions, weddings, people will not come with extra bag to carry rice. Rather, they'll be looking for lacasera bottles to carry, to excuse the place because of. So again, maybe that will be my last follow-up question. How do we draw the line in the context of what I said, being between the devil and the blue sea? When you have what I would call research findings that may not be too much public knowledge, it may be a local knowledge, let me use that word, but not public. And so being afraid of making it public, and then you have more subscribers as against keeping quiet. How do you place that balance?
Thank you, Dr. Dangana. That's again where that triangle comes in. The crosstalk between clinical neuroscience, experimental neuroscience, and classical neuroscience. So if a clinician begins to study a trend where people are not taking what we could have considered classical psychoactive agents like cocaine and what have you, but they come down with, manifestations that suggest that they have, been abusing something, then there's a need to probe that and document that. What have you been consuming? A colleague of mine in Leipzig yesterday, for example, who has also been in neuroscience and genetics, just posted that there is this psychedelics, from a mushroom that grows only on goat dung. And, I read it and I was laughing. But again, that's very interesting. So if people pick that up and they begin to consume it, it's probably what many people have not realized before, And then they come down with this. So the clinician begin to document it. And then the clinician connects with the public health person. And that is why, again, we must develop our public health in Nigeria. It's not just public health from the hospital. Public health is a field on its own that has a whole lot to contribute to the health system. So, and that is where we begin to push for policy, but without digressing. So the clinician now triangulates what is the pattern here? Where are these people coming down with these presentations or manifestations? And from there, they have report from the community, from the public. And then the experimental neuroscientists, like many of us, now begin to isolate those agents, now begin to experiment with them and realize, oh, this is what happened. When these drugs interact together, pharmacologists will talk about drug-drug interactions. Sometimes the behavior, the pattern of effect could radically change. And so this crosstalk now gives a full picture. And it again buttresses the fact that no matter how hardworking we are as individual scientists, if there's no crosstalk amongst all of us, we will keep contributing to knowledge. But then whether we're really going to be solving problem is left for us to see. Another very good example, for instance, was when we look at caffeine effect. Caffeine is in coffee, but when you isolate caffeine and you consume it at certain doses, it could be a different thing entirely. And I know that at least one of my research has documented the fact that caffeine exposure in model changes the morphology of astrocyte onto puberty. We stop at puberty, but I'm tempted to say for life. And that has implication. So what I'm therefore trying to say is there's a whole lot of things that we need to begin to look at, again, by having this crosstalk, triangulating, and make sure that at the end of the day, there's a full picture of whatever it is that we are studying, that our basic research has translational angle to it, that informs best practices, that informs our policies, that we are equip people to do the best of cutting edge research that they can do, and that there is return upon investment. I think I could stop here, but back then, I think a few years ago, if you just come to the table with a whole lot of technology-driven research, people applaud you. Now, if you are going for NIH and Wellcome Trust and what have you, if you can't prove that this is going to contribute to knowledge, nobody's going to put dollar on the table for you. And so people don't just want adventurers in research anymore. We just want to catch on doing research and publish beautiful papers. They will ask, what is the return on investment on this? Is this solving a problem? It does not mean that this must eventually solve problem, but it's saying that there is prospect for it. And that if that prospect is not identified or cannot be established, then we don't waste money, on such research. So again, triangulating, making sure that there's multidisciplinary approach and that we are able to crosstalk among groups of neuroscientists. Thank you.
Thank you. Thank you so very much. Thank you. We're very grateful. Often at times when you hear feedback of this, you get up, solution will start pouring. But it's a whole quantum of work. We already know that there is the health system, architecture. Let me use that word. The health architecture is already constrained. There is a growing population of those who want to go and join Joshua Wolabi, and then there are those who will have to hang around. So those who are hanging around, there is so much more to do as against all of that. So I think when we begin to think multi-sectoral, multi-approach, then we can begin to start the solution from somewhere. Thank you very much, Josh, for your time. And it's really, really, please, let's appreciate him some more.
Thank you very much. It's a honor and a privilege for me. Thank you.
Thank you. Ladies and gentlemen, I told you that that was going to be the last presentation. Thank you very much. And I'm sure that the time we have spent here today has been a rewarding one. If you have enjoyed yourself beyond the food you ate and other things, I just want you to focus just on the quality of programs you have accepted that you enjoyed. So if you have enjoyed the program thus far, can you please appreciate yourself for I'm aware that this evening there is going to be a farewell dinner, because by tomorrow morning, reality Check. We'll start. Those of you who are in school, who are teachers and have not finished marking, by tomorrow, you wake up to that reality. Those of you who are in school and have not finished an assignment, from tomorrow, your eye will open. If you have a quiz that you have begged and they shifted the quiz for your sake, as you sleep tonight after you eat, as you are traveling back, just be reading, because we have been stocked. Yes, sir. Please come forward. I hope you're not calling a speaker, because I've told them if you mention that, everybody will carry his bag and walk away. Thank you very much for your time, and I wish you a pleasant evening.
Yeah, thank you so much, Dr. Dangana, for that wonderful session. So at this point, I also want to say that from the desk of the LOC, we want to appreciate our patience, our doggedness, and we appreciate God that today has been so rewarding. Just some few announcements. As it has been said, we are going to be having dinner, and that is going to start by 7:30 p.m. But before then, as we go out of this auditorium, we want to encourage every one of us that the last but not the least phase of today's program is the poster presentation session, and that is just going to be done on a relaxed mode, so to say. Majority of the posters were displayed yesterday. We just have few, maybe like 3 or 4 that are new. So please take your time. Those that presented yesterday, we are also encouraging you to still stand by your poster so that some few people that came late can also interact with you. So please, as we are doing that, it's also an avenue for us to interact with one another, get contacts from the elders in the house, from our colleagues, and let's make friends that will help our career path. Let me just give a brief description of how to get to the dinner venue. It's actually in the male hall, Winslow Hall. So for those of us that are in the female hall, we can find our way there. Now, if you have visited Babcock shopping mall, it's very easy for you to locate. The road that is adjacent to this shopping mall, as you are coming, going from this axis, just go straight that route. Be watching towards your left. I think the first, second, and the third hall, that should be Winslow Hall. And that is where the dinner is going to be taking place by 7:30 p.m. tonight. Our elders, we may likely come and pick you up if you wish through a bus, and others, we can just take a walk to that place. Under 5-6 minutes, we should be there. Once again, thank you very much. Let's go to the poster session. Stand by your poster, even if you presented yesterday. And for the new ones, let's make ourselves available as people will be coming around to interact with us. Thank you, and God bless you.
I thought I was going to come back with Professor Olude.